Say the sea gets hotter and the drums be louder and the world seem colder, singing the same old tune. And the times get harder, worlds wait upon us, but it don't mean nothing, cause we're leaving soon. And so we cruise, baby, just you and me. And so we cruise, baby, to let me see. A hand on the wheel now and the other on your knee. Ain't worried about nothing, cause I feel you next to me. Graying with the gathering storm and the smoke hangs on the breeze. I know that I got you and you know that you got me. Say the sea gets hotter and the drums be louder and the world seem colder, singing the same old tune. And the times get harder, worlds wait upon us, but it don't mean nothing, cause we're leaving soon. And so we cruise, baby, just you and me. And so we cruise, baby, to let me see. So we cruise, baby, just you and me. And so we cruise, baby, to let me see. So we cruise, baby, just you and me. And so we shoot, shoot. Tell me, do you think of me like I think of you? Night after night, nothing to do, no one to fight. Now you come far away, far away. If you can, would you stay? Girl, you think you got it bad. Girl, you think you got it bad. Girl, you think you got it bad. Got out different ways with the same old payout. Had all the dreams with the same old outcome. Had it by a string and the same old breakdown. Worked it to the bone with the same old habit. Girl, you think you got it bad. Girl, you think you got it bad. Girl, you think you got it bad. Got out different ways with the same old payout. Worked it to the bone with the same old habit. You're acting like you're so bad. Never into it. You're acting like you're so bad. Never into it. Girl, you think you got it bad. Girl, you think you got it bad. Girl, you think you got it bad. got the red hair alone, gold hanging from her ears, melanin lady. She got a jig on strong. It's so good to be here with you. Somebody's gonna fall in love tonight. And I hope we make sweet love tonight. God created you in the heavens just for me, just for me. Been waiting so long for a woman like you to make me feel so right. You shine in the sea of people. Oh, this moment so real. Melanin lady, covered in all white lace. You're the baddest in the place. So good, shorty. Somebody's gonna fall in love tonight. Let's lose ourselves in this second night tonight. God created you in the heavens just for me, just for me. Been waiting so long for a woman like you to make me feel so right. God created you in the heavens just for me, just for me. Been waiting so long for a woman like you to make me feel so right.
Please take your seats. Our presentation will begin in 5 minutes. Thank you.
Please welcome to the stage Stat Senior Writer for Medicine and Editorial Director of Events, Matthew Herper.
Hello, everyone. I want to tell the people over there, I know you can't hear, so I'm going to talk really loud. Try and move you in. Everybody, yeah? Okay. Well, hi, everyone, and welcome. I'm thrilled on behalf of our whole organization to welcome you here to New York and to those of you attending virtually. I'm going to leave it to Rick Burke, our co-founder and executive editor, to tell you just exactly how much Stat is kicking butt in winning awards lately. But I do want to welcome you and emphasize that I think what we're offering you here are interviews and panels guided by many of the best healthcare journalists in the business. This is different from what you'll see at a lot of other conferences. I hope you appreciate that. And the thing about us being good is that we're extra willing to accept your help. So throughout the day, please, it's part of the fun. It makes us all smarter. Please raise your hands with your questions. Please speak up. If you're here in the room, it's because we're welcoming you here, and we want to hear what you have to say as well. My right hand, Catherine McPhail. Catherine McPhail! We'll be roving the audience with a mic, and so will I, to take your questions. There may be some other staffers. Make it a competition. Get the mic. We live in interesting times. That's not, by the way, a Chinese curse, as most of us have heard. But it's true. And every day it seems there is a new shocking development with the potential to change the way research and development is done. When we planned this, or started planning this, we were looking at a lot of long-term issues in R&D. But some of these short-term issues, billions of dollars apparently evaporating from the NIH budget, individual universities losing hundreds of millions of dollars in funding, universities unadmitting next year's class of grad students, FDA staffers showing up to return to in-person work and working in closets. The disruption to what we think of normal is profound, and we're going to be dealing with that today. Stat welcomes a diversity of opinions and debate, and there were invitations to Trump officials and MAHA-adjacent speakers. Those were declined. But let me give you a quick overview of today. Our practice is that I'm not going to list sponsored presentations here, but you should stay for those two. They're great. Starting off, Stat's Katie Palmer will have an interview with two tech execs about how to get GLP-1s to patients. I think this will be a bracing talk for a lot of people with pharma backgrounds in the space here. We'll follow with Jonathan Wozen's interview with gene therapy pioneer Jim Wilson. You probably read Jason Mass's story that, to quote My Chemical Romance, the CRISPR companies are not okay. Jason will talk with Kiran Musunuru, one of the hottest new figures in that field, about what's ahead and how things might get better. Nobody covers biotech better than my friend Adam Feuerstein. He'll lead execs from Akero and Madrigal in a discussion of how drugs for the liver disease MASH, which is caused by obesity, are finally having their moment. And you don't want to miss this. Before lunch, Helen Branswell will join Tom Frieden, the former head of the CDC, and Catherine Edwards, who literally wrote the textbook on vaccines, to talk about public health. I promise you this session is not to be missed. And then we'll have lunch, and it'll taste great. After lunch, Polk Award winner Lizzie Lawrence will run a discussion about the stakes for science in healthcare right now. I'll interview former FDA Commissioner Scott Gottlieb about a lot of things, including maybe eggs. Allison DeAngelis will take you deep into biotech financing and the effort to license drugs from China. And Rick will get the latest from our amazing DC team. A lot of the stories they'll talk about probably haven't happened yet. Then I'll have an interview with Pfizer's chief strategist, his first anywhere. And we'll have a session with Bill Nye, the science guy, who will talk about ataxia, a topic that is both personally and scientifically important to him. We'll finish with our live Stat Madness session, during which I will play the part of Simon Cowell from American Idol. And we'll close with a party celebrating Stat Madness, including our special cocktail and mocktail. some housekeeping. On the back of your name tag, you'll see the Wi-Fi info and a QR code to access our Summit mobile app hosted on Bizzabo. Download the app to find the most up-to-date agenda and speaker information and connect with other Summit attendees. Once you've downloaded Bizzabo onto your phone, you'll just need to input the email address you registered with. Since this is a biotech and health, not tech audience, our staff can help with this. If you hear something interesting or unexpected, share it on social. Be sure to use the hashtag StatBreakthrough and tag Stat News on your social platform of choice. You can tag me if you want to. And finally, a special thanks to our Stat Breakthrough Summit East sponsors who have helped to make this event possible. Our premium sponsors, Evidation, NYC EDC, Eli Lilly, Novartis, Samsung Bioepis, and Ultragenics. Our Stat Madness sponsors, Jobs Ohio and Cure, and our supporting sponsor, Sanofi. Thank you all for being here, and with that, I'll pass it to Stat's co-founder and executive editor, Rick Burke, for some opening words.
Hi, good to see everyone, and I'm told there's great energy in the room, and we're also hello to all the people around the world live streaming this event. I just found out Matt mentioned the cocktail event at the end as a way to keep you all here all day. I asked what it's in it. It's called a cranberry catalyst. And it has all kinds of sciencey ingredients as well as alcohol, but there's also a virgin option as well. So I hope everyone stays for all this. As Matt said, this is a really timely gathering because health and medicine is under such a state of disruption. And I really think that's too lame a word, disruption, but I don't know what to call it. So any thoughts? It's not disruption. What is it right now, science and medicine in the world? What's attack? What else? AI? What? What? Cataclysmic? Bloodbath? And we're not being, I don't want to be political here, but just the reality of what's happening to science right now. Alex Hogan, you're always good for description. What do you think? Not to put you on a spot. Yeah. All right, we'll go to cataclysmic. Does that fit? Does that suit? So I'll change my cataclysmic. So because of these cataclysmic times, in these cataclysmic times, we see it as Stat's mission to deliver the most trusted and authoritative coverage of any news organization covering these issues. And we're finding that readers are responding. We're seeing the highest rate of news subscribers in Stat's history. We haven't seen this kind of interest in Stat since when Helen Branswell there was the first reporter to really sound the alarms on COVID, and we had tens of millions of new readers. We're not seeing quite that level, but we're seeing people line up or sign up to read Stat in a way we haven't seen before. And by the way, we'll be 10 years old in November, so that's kind of a little context. Not only are readers flocking to read Stat, but they're engaging more. They're reading more stories. They're staying longer because this is really important stuff, and we're trying to get to the bottom of it as best we can. They're reading multiple stories. They're listening to our podcasts. They're reading our newsletters. And I just also want to put in a little plug for companies and organizations. We have special group rates, so more of the employees can read Stat. One FDA official told me he reads Stat to know what's going on in his own agency. A little aside that I probably shouldn't share, but we're journalists. We got to be transparent. So guess who pulled the plug on our group subscription? the FDA. So I just made the point that people at the FDA have told me they need to read stat, but now if you work there, there's no longer a group subscription, or I don't know if it's expired yet, but they're not renewing it. Just so you know, and anyone from the FDA watching or former FDA, we are offering special rates for employees who can't get stat at their agencies anymore, or who are worried about having a job but want to keep up with what's going on. As you'll be reminded on stage throughout the day, many of our reporters are the best in the business. You'll see reporters here from stat reporters from Boston, DC, New York, San Diego. These reporters are working day and night and weekends to cover every turn of what's going on with this cataclysmic, or whatever you want to, however you want to describe it, in Washington, the impact, the reverberations on health and medicine and life sciences. We're trying to write stories and report stories that are important to know what's going on, who's being affected, and it's hard work because things keep changing by the moment. Someone is fired 1 minute, then they're rehired, then this research project is pulled. I don't have to tell you how this is playing. But when I say how great, and when Matt says how great our reporters are, don't take our word for it. I just want to say that I'm proud to say we've been recognized with some of the most prestigious honors in American journalism. We've had 2 Pulitzer Prize finalists. We have a 3-time winner of the Polk Award, and most recently, recently for our healthcare colossus investigation, which chronicles how United Health Group built an unrivaled physician empire to boost its profits and expand its influence. Just last week, we were recognized in 7 categories for the highly competitive Cebu Best in Business Awards from the Society for Advancing Business Editing and Writing. For the 2nd year in a row, we were recognized for general excellence in business reporting. The citation reads, "Impactful, intelligent, deeply reported storytelling with rich context that makes you feel smarter after reading." And I have to say, before I leave, I have to do a particular shout out to our host, Matt Herper, because he was among the individual winners in this big Cebu award for his range of work, which, and the citation reads, "Matthew has a knack," and let me just say, they didn't even talk about his event assembling skills, which are like the best anywhere in the world. But in terms of his his actual stories that he does, Matthew has a knack for making readers smarter about one of the most impenetrable features of our modern economy, the business of healthcare and medicine. Across a range of engaging, well-written stories, his work imbues complex topics with essential context. And let me now just turn it back to Matt, who indeed, as the citation says, makes us all look smarter. So thank you, Matt. And I'm sorry if I've embarrassed you.
Not embarrassed, but we're running a couple minutes late. We need to make that up, so I'm going to talk real fast. You see ads for GLP-1s everywhere. They're not always from drug makers. Here is we're going to peer into the tech companies that prescribe these and other medicines online, and talk about how online prescription should happen. Here to kick off today's programming, 2 tech executives who are changing the way healthcare is delivered, to talk about their philosophies of getting drugs to patients. Please welcome Zach Reitano, CEO and co-founder of Ro, and Owen Tripp, CEO and co-founder of Included Health, and to moderate, Katie Palmer, health tech correspondent at Stat.
everybody. Hello to both of you. Thank you both so much for coming bright and early. So patients are facing incredible access issues in today's healthcare system. Primary care shortages, specialist referrals are really difficult to get, and everything costs way too much, it seems, no matter who you are. So both of you have built businesses that try to address those access challenges in different ways. They look very different, the companies that you've built. So I'd like for each of you to start, lay out for the room what it is that you do and sort of the thesis statement behind the companies and the technologies that you've built. Let's start with you, Owen.
Sure. Included Health is a personalized all-in-one healthcare company. We deliver benefits to members of group-sponsored health insurance plans, including over 30% of the Fortune 100. Our solution is to basically make it really simple for when members come, to provide everything from integrated on-demand primary care through behavioral health, and in some cases providing specialty care for the most rare diseases in the world. And we do this because our eye is on how we can create better outcomes for people, because it is those clinical outcomes that drive sustainability in group purchasing. So millions of visits per year. We're one of the largest, if not the largest, virtual care company now, and we are the largest navigation and advocacy company in terms of helping members better use the benefits that are entitled to them.
And you've been around how long?
We started the company in 2012. We founded it as a company called Grand Rounds, and in 2021 we changed it to Included Health.
Yeah. And Ro?
Awesome. Thanks for having me. At Ro, it's very simple. We provide this concept of goal-oriented healthcare. So patients come to us and they tell us what they want to achieve. So I want to lose weight, and we have comprehensive obesity programs. I want to have a child. We have fertility products and services. Better sex, better hair, better skin, whatever it may be, it's what they want, and we work backwards from there. The way that we deliver that care is we have vertically integrated the business. So we have a national digital doctor's office, built our EMR from the ground up. We own and operate our own CLIA certified and CAP accredited lab for at-home lab tests. And we operate pharmacies throughout the country. So it's that seamless integration between doctor's office, pharmacy, and labs. It's all abstracted away from the patient, again, through the lens of what they want to achieve. And over the last now 8 years, so not quite 12, we've helped millions of patients across the country, one in every single county in the US, and are hopefully just getting started.
Awesome. So these issues of cost and access have really come to a head with the approval of GLP-1s for obesity care in particular. How has this era of GLP-1s demand from patients and the cost management that is necessary from these large group purchasers crystallized or changed the way that you have built your business and technology?
Who'd you like to start with that?
Whoever wants to go first.
You want to start?
Sure. I love, and this is not going to come as a surprise, I love GLP-1s for so many different reasons. They saved my dad's life, so first and foremost, that's probably the biggest reason I love them. I also think that they will be the most important medical intervention of our lifetime and help us combat chronic disease. And third, I think they will shed light, which we'll talk about today, on a lot, if not all, of the flaws structurally that exist in our healthcare system. And I think they will do that for so many reasons. In terms of the way that they've shaped our business, they aren't, we've talked about GLP-1s, but the way that they came into the zeitgeist is through obesity, right? But they're not just obesity drugs, and I think that's well established now. They started as treatments for diabetes, obesity, sleep apnea, chronic kidney disease, heart disease. They're being studied on everything from addiction to Alzheimer's. So I think that the impact that they will have stems far beyond obesity, but we've never really had a type of medicine that I think will attack pretty much 80% of the reasons why people today are dying in the US, actually. So to me, that's where it'll start to shed light on some of the things that we'll talk about today, which are the structural flaws in our healthcare system. I think it will challenge and ask questions about what role does an employer play in determining a patient's individual access to healthcare. I think it will highlight, which we'll talk about as well, the bizarre pricing, list to net, why all those things exist. Because we talk a lot about this at Ro. GLP-1s are satisfying 5 criteria never before seen in medicine, right? So they are extremely effective, they're scalable, patients want them, providers want to prescribe them, and the majority of the US population is eligible for them, right? That has never happened before. And so when you have a need of 150 million people, finally there's a shared problem that everyone in the healthcare system is experiencing simultaneously, and I think that will be what leads to the spotlight. That will be what leads to the rewiring. That is what led to the, if you think about, there's analogies here to COVID, the fact that we all shared the same problem at the same time is what catalyzed so much change so quickly, versus, yes, we all experience different healthcare problems, but it's very rare that the majority of a population is experiencing a single healthcare problem at once. And so that is what I think will serve as the catalyst. And I think you've seen a lot of that catalyst towards that rewiring to be far more patient-centered, which we'll talk about. But that's how it's shifted our lens.
I'm also a fan of GLP-1s, not least which because it saved Z's dad's life. But I think sort of more broadly, and hopefully heartening to the people in this room, it has awakened newly a sense in the American population that we can create wonderful medications that have very rapid effect with a very trustworthy safety profile on something that actually really matters to people. And that makes me feel good. And it generates useful conversations in our primary care clinics around how to use these drugs. Where I will note concern, and I know we'll get into it, is that at least as of now, with all of the sort of publicly reported data and data that I'm happy to share from our own book, people don't stay on these medications. And partially it's because they don't know how to stay on them. They don't know how to manage the side effects. Managing the side effects on their own can actually be an expensive endeavor for the working Americans who try to take them. And we haven't gotten to that part of the curve yet, and we need to. And that's why you'll hear me talk a lot about an integrated approach, because it's only through an integrated approach that we can achieve the sorts of outcomes that we talk about that make these drugs really wonderful.
So what does a patient who is eligible for a GLP-1 look like when they enter included's funnel at this point?
Yeah, maybe
It's easier to imagine from Ro's perspective because that's direct-to-consumer pathway that's easy to visualize.
Yeah, so maybe I'll lay out some landscape for this because I think my sense is that Z and I will probably share a lot of facts about what we're seeing in our businesses that could seem sort of at odds with each other. I actually don't think they are, but the nuance here is important. So I think you said 80% of the American population is eligible for these things. Is that what you said?
About 60%.
60%.
Majority.
So what we see in our book, and just sort of, again, give you the sense, we're focused on the 150 million Americans who get their insurance through their employers or other group purchasers. That's the single largest segment of American healthcare. Not even close, actually. It's the largest. And you'll see reported data between 30 to 40% eligibility. The reality is within our book, most of that eligibility comes with some sort of string attached to it. You have to go through a step-based authorization. You have to sort of be eligible for specific sort of conditions or comorbidities. There could be, and Z may be able to lend insight here, there could be a sort of split between where it's been approved for diabetes, which has been the case for a very long time. We sometimes lose sight of that fact. And where it's been newly approved for obesity. But how I would summarize this, Katie, is that when people come in, our employers are really invested in the idea that if they can reduce obesity sustainably, you'll have all sorts of wonderful bang-on effects on musculoskeletal, on cardiovascular care. We talked about neurodegeneration, which is not really a big employer concern, but would obviously be a positive benefit as well. The problem is the fallout rate, and so the affordability challenge on these things, which is massive. And to give you a sense, if every employer covered this according to the actual obesity in their population, many of those employers would go bankrupt. Their response is to say, we're going to meter access to this thing, and we're going to make sure people stay on it.
When we're talking about the cost of these drugs, it seems like Included is addressing its client base, these large group purchasers. Ro is doing the cash pay model where it's talking to patients directly. If in either case, these drugs are going to cost somebody money. It seems like both of your approaches just like shift the nexus of that cost management a little bit. So how does that impact the technology that you're actually building to facilitate, make more efficient the care process here?
Yeah, I think there's a few things we want to touch on. There's the cost of the care delivery, and then there's the cost of the drugs. Mm-hmm. We don't obviously control the cost of the drugs, but I do think we can actually influence how they're priced by increasing access. In terms of the fallout rate, I want to hold both things to be true, which is patients will definitely achieve better outcomes if they can have an integrated approach, if they can have their handheld. We enable patients to message us 24/7, have sub 1 hour response times to side effects. They can report them in structured ways. All of those we do see lead to better outcomes. It is also challenging to look at the fallout right now when there's a national shortage of the drug and when every single employer pretty much requires a patient to go through an obstacle course to get the drug. People are shocked, well, why is it so hard for someone to stay on the medication? Well, pretty much every 6 months they're asked to jump through hoops, and they're asking their providers to jump through hoops, and it's not simple and straightforward, right, for anyone here who's interacted with the healthcare system. So, and the way that we have decided to solve that is by adding more and more restrictions. Oh, well, people aren't staying on it, let's add step therapy. People aren't staying on it, let's require 2 comorbidities. People aren't on it, let's add another app that they have to sign up with to make sure that they use it. People aren't staying on it, well, there's a national shortage. People aren't staying on it, let's raise the BMI requirements. And all of these things, there's a shortage of PCPs, they get 12 minutes with their doctor, the PCP walks in, and they're the ones that have to fight that fight on behalf of the patient. That's impossible. I can tell you that Netflix, 12 months after people have signed up for Netflix, there's about a 40% retention rate 12 months out. These drugs, when they are affordable, have a better retention rate than Netflix. So it's not, yes, could they be even better? Of course. But I think that I just don't want us to miss the forest for the trees here, which is like, they're really good, they're extremely effective, and when people can actually afford them and they're not deciding whether to go on vacation or get access to GLP-1s, they will stay on them. Can they achieve better outcomes with better care? Absolutely.
Can I just
Yeah,
respond?
no, I saw you smiling.
Well, I mean, I can't wait till the meeting where I pitch to employers that they should cover Netflix or that that GLP-1s are better than Netflix, which I know is not exactly what you're proposing, but let's just put this in context. The employer mindset on this is actually pretty straightforward. They're not arguing the efficacy of the drugs, which I think we could all sort of quickly agree on. They're just saying, are we going to obtain the benefit from the member who uses it? And the answer right now is empirically no, because people do not have persistence rate at 180 days or 1 year on these drugs. Now, I think the two of us actually feel like we have pretty good command of what the formula is. If you provide wraparound primary care, if you wrap around coaching and understanding on these symptoms, as well as some of the softer and I think still unaddressed sides of this, which is, how does this change how you relate to the rest of the world? How does it impact your behavioral health? If you provide that integrated all-in-one model, you get great outcomes. And we can share some of that data for sure. But in our primary care practice, we exceed the national persistence rates. But that's why these things need to go hand in hand. Simply giving somebody access to the tool and wishing them good luck does not yield good results. And employers are not going to underwrite that. They shouldn't. It's their job to manage the bottom line of their companies, and this is the single largest threat that's coming out of their health benefits line.
I want to check and see if there are any questions in the audience during any of our panels. We'd love to hear all of your questions. So if you've got one percolating, keep it in mind, raise your hand. Somebody will come around with a microphone. If our aim is to get patients to the drugs that they need with all of these wraparound services, the primary care nexus that they need, within the direct-to-consumer model, what are the pathways that we still need to build up to ensure that the patient doesn't end up in that silo without the extra support that they need?
I mean, I think we're doing that right now. We just want to continue to deliver better and better care. I think that there's a bunch of different things that we've touched on, right? There's like the who is actually paying for it, which depends like how wonky we want to get, because I think patients are paying for it through either their suppression of wage or through their healthcare benefit, like it's coming out of someone's dollars. And ultimately it's the patient, it's just how many times it's cut by someone else before that's the case. So there's the cost issue, then there's the care delivery issue, which I think we agree pretty much in every aspect that someone having access to better care will deliver better outcomes. To me, an interesting one is on the cost side is just the time horizon, actually, which is if we all sit here, like I think one of the really fascinating things is Lilly, who Ro has a unique integration with for their Zepbound vials. If you look at probably about 18 months ago, November 2023, they released Zepbound. And it was about $1,000. It was 20 to 40% more effective than Wegovy at a 20% lower price, depending on which studies you looked at. 9 months later, they released the vials, and then 5 months after that, around 6 months after that, they lowered the price of the vials even further. So it's $3.49 to start, $4.99 to continue. The reason I bring that up is because in 18 months, the largest pharmaceutical company in the world, with their most innovative drug probably over the last 50 years, dropped the price by 70% in 18 months. I think that was in large part driven by consumer demand. I don't think that that would have happened any other way. And so I do think you're seeing the demand of consumers drive that. But the really interesting thing that I mentioned earlier relating to shedding the light on the flaws of our healthcare system is the debate on the time horizon of cost and whether employers will cover it now or over the next 3, 4, 5 years. It's definitely something employers have to navigate. But as a society, I think, like, imagine just 7 years from now, 10 years from now, like, the price will be lower, it will be affordable, people will get access to it. The question to me is, how do we actually scale the care delivery required to deliver the level and quantity of care that people need? We have scaled drugs before as a society. The drugs get scaled, the prices come down. That's pretty inevitable. The next 10 years, I think one of the more interesting things will actually be we've never really had a single healthcare product that truly defines inequity. And I think this one will. There's like a clear product where, with all other aspects of wealth and even what wealthy people have access to in the healthcare system, it's kind of amorphous. It's like, oh, they have better access to better specialists, or they have faster care. People kind of don't even know what it actually feels like or what they actually have access to, versus in other aspects of life where, like, you're looking on Instagram, you see if someone has a Ferrari and you don't, right? But in the healthcare system, the inequity is sort of amorphous. It's life expectancy, it's level of chronic disease, it's access to these different parts of the healthcare system. With this, it will be like, do you have access to this world-changing medication or not? And so I think if we know if it's an inevitability that it will be affordable, I think that everything that we should be doing now is to say, as a society and across employers, across patients, across government, is how do we make sure that over the next 10 years, we don't actually have that massive, massive gap? Because if someone says that they don't think it's inevitable, let's have that discussion, but I feel like it just will be. So then on the care side, what we think a lot about is, okay, if we know that the drugs are going to come down in price, if we know that ultimately there will not be a shortage in the future, well, then it comes down to, how do you get 150 million people care when they currently can't make a doctor's appointment, or they currently have 2 minutes or 12 minutes or whatever it may be? You need, the only way we have ever done more with less as a society is with technology. We need to get leverage out of the labor force, and that's what we think a lot about at Roe, is we take a lot, I think the healthcare system is obsessed with quality, but it isn't obsessed with scaling that quality. Even the word scaling care, and it makes people's stomach churn, but it's because I don't think we really fully appreciate the cost of not helping the marginal person, right? It's like we're sitting here, people are screaming in a very loud room, and I think we just put on headphones, treat the person in front of us, and we're like, well, if we do this one really well, that's good. That's how we'll sleep at night. And I think it's not that we should eliminate this type of care, but we need to have a group of people, not everyone, obsess over the waiting line, which is massive, which is what we do at Roe.
When you talk about scaling that quality, you're talking about doing one thing very well, which is prescribing and providing supportive care for a limited subset of conditions with a formulary of drugs. Oh, and on your side, you're more like building the highways to connect the existing pieces of the healthcare system. So how is that challenge different?
Well, I have to start by saying I really actually liked his framing around inequity. I hadn't thought about that, and I agree. I mean, actually, just having proper access to a unit of primary care in this country is increasingly a struggle. And now, and it doesn't probably apply to most people in this room, so you need to imagine with me, imagine that you don't speak English and that you are working a shift that literally prohibits you from actually using your phone and contacting people. You have to build a healthcare system that is both flexible to those needs, deeply personalized in its care integrations, and able to deliver the whole of healthcare together. And I think that's critical in understanding certainly our strategy. We see primary care as needed to be integrated with specialty care and behavioral health, not just because that feels like the right thing, although it certainly is, but because it's actually the only way that you achieve the outcomes that can ultimately be underwritten to. Then, and only then, you have an American healthcare that is more performant, more aligned, and ultimately yielding better healthy days for its entire population. Let me describe to you one sort of optimistic use case for how this actually happens. I think employers, and I want to make sure that this is clear, I think employers actually want to underwrite these drugs. if the price comes down in the way that Z sort of depicted it might, and if people can stay on it and it remains integrated with the rest of their care, because it pays the line on so many things that they care about. Workplace safety, presenteeism, job performance, long-term healthcare costs. So people are waiting to do this. They just need to deliver it through a mechanism that's ultimately integrated, because if they don't do that, they're just throwing a bunch of money in the fan and seeing it blow out of their buildings. And most Fortune 500 employers, indeed most employers in the United States, can't afford to do that.
I'm curious if you agree, going back just a minute, with Zach's assertion that the cost reductions that we've seen so far are primarily driven through consumer demand and not the needs of your large group purchasers.
So what do you mean? I'm not sure that I have seen all the data that sort of consumer demand alone lowers prices, and usually consumer demand actually can raise its price, at least as long as that price is protected. So I think I'd have to beg off on that, because I'm not as expert as he probably is in the pricing curves. But what I can tell you for sure is, as we have additional options, not just sort of different formulations, but actually different ways to take these drugs or inject these drugs or participate in this revolution, that will have also an effect on consumer experience as well. And I'm really optimistic about that, generally speaking. But look, most consumers in this country do not understand anything close to what we are talking about. They don't really understand. They've heard GLP-1 maybe, but they really couldn't tell you anything about the profiles of these different drugs. And really, I know that a lot of the marketing is about eking out that 2, 3, 4, 5 points of additional potential weight loss. That really doesn't matter. Sorry to ruin anybody's day on that topic. What they actually care about is, can they afford it and do they have access to it? That's really the thing that matters. And the sort of remarkable thing, Z talked about all these sort of gates that are going up. People will still find ways to jump over those gates if they believe the medications can and will be successful. So that too is also optimistic. We just don't need to introduce that friction.
On the price versus efficacy thing, I think this is where in the consumer world, it's actually very straightforward, where it's like cost, quality, and convenience for people. And convenience is the form factor, it's the way that they can go get it, it's how it's delivered. But it's like, if someone, it's not just, hey, it's those options, right? If something is $1,000 and they can lose 20% of their body weight and they have to travel 50 miles, well, that's going to be less attractive than if it's 15% of their body weight, they're paying $25 and it's delivered to their door. So they're just weighing cost, quality, and convenience. The best technologies usually, over time, what they do is remove trade-offs where people don't have to choose and they get all three, right? And that is what I actually think you are seeing with, right now, what you're seeing with Zepbound. It's a cheaper, better drug. And so no one is trading off when they get it, which is when, if you look at market share in Q4 and in Q1, you're seeing massive swings there. But to Owen's point, the more competition we get, and actually the more similar future generations are, the better that actually is for patients. If someone's 20 versus 21, and someone's in this pen, and another person's in this pen, and it's like, this one has 4 things on the label, and this one has 6 things on the label, it doesn't matter. It's not going to matter. The only thing that's going to matter is that price is going to come down, because patients will have more options, and it will be actually harder. They will be more and more commoditized over time, which is sort of my point on inevitability, which is like, we're going to be here 10 years from now. It's going to be cheap and accessible and affordable. The problem is, we should be worried about what happens when tens of millions of people get access to it and tens of millions of people don't, and we're going to, like, do we end up with an Elysium 10 years from now situation?
Can I mention 2 statistics that are probably not on people's radar, but if you're interested in this and you have a stake in it, these are the 2 that I think you actually should pay attention to. One is labor force participation rate in the country. I'll explain why that's important in a second. And 2, how tight the actual employment market is. And those things are related, but not exactly the same. Why? Because if you are my customer, you care deeply about getting the best talent in the United States. And increasingly, benefits are going to be about what the pharmacy plan is and not what the medical plan is. Historically, we've always chosen about medical network access. Now people are going to be like, is this drug actually covered by my employer or is it not? That actually is probably the leading indicator of what he's talking about, of where consumers are going to vote with their feet. I'm not sure it's going to be at the level of detail about which drug, but maybe we come back.
Versus not.
We can come back and sort of revisit that. But this is actually going to be a real marker for people about whether they can go somewhere and take their consumer dollars in participation to achieve the results. And we are actually seeing that in this most recent open enrollment.
We certainly can revisit that, but probably in a future story instead, because our time is up. Thank you both for kicking us off with this great conversation. And again, thanks for being here.
having us.
For this sponsored conversation, please welcome to the stage Jillian Woollett, Vice President, Head Regulatory Strategy at Samsung Bioepis, and to moderate, Stat Senior Writer for Medicine and Editorial Director of Events, Matthew Herper.
And thanks to all of you. It was a great first session. So here's how the world is supposed to work in the drug business. You're supposed to invent a new medicine. If you're lucky, it sells like crazy. You make billions and billions of dollars, and then the patent expires, and lots of cheap competitors come in, and it goes generic, and now my Lipitor is so cheap that I don't even know what it costs. That's not what's happening in biosimilars, so we're here to talk about that. And when we were talking before, Jillian, you've been in this space for a long time, and we would have thought that we would have had people competing for taking on the great big biologic brands of today, all the best-selling drugs of the moment. But there was a recent report from IQVIA, who would know, saying that for branded biologic drugs in 2023, all the big protein drugs that are currently sold, around 90% don't have biosimilars in development. No one's even trying to unseat the kings. So there's not even a specter of competition for a lot of these. Why is that? What's going on?
So there's a bunch of things. The most obvious is it costs about 100 times the average generic to make a biosimilar. So generic, you talk 1 to 3 million. Biosimilar, it's costing you 100 to 300 million just to make the product in the first place. So you need significant resources, and historically you need to know how to run clinical trials and things that you don't in generics. The bigger issue is once you're approved, do you get market share? And do you get market share commensurate with the price reduction? So we do know how to make the products better to counter this so-called biosimilars void, but revising regulatory processes is not easy. And so we could probably half the time, half the cost. Need to do that. Second half is we've got to have a market that values the cheaper product. That's not the nature of the US system.
Why is that? So how does that, because generally when I go to buy something, I value the cheaper product, right? Usually if I'm buying a shirt, I'll buy the one, if they look the same, I'll buy the cheaper one. Why is the US system paying more for insisting on buying the expensive shirt?
The first point is they don't just look the same. The biosimilars have been evaluated by the same regulators to the same standards of quality, safety, and efficacy as the originators. So functionally, in terms of clinical outcomes, they are the same. So there is no compromise, but the incentives are misaligned in the US. It's who's making the decision and on what basis, and I'm sure everybody's heard about PBM reform. And part of it is that the majority of that list price is not going to the person making the product. So the system is incenting the higher cost product based on rebates and fees. So the biosimilars are forcing down the prices of the originators to some degree, but they're not getting market share. So for us, Samsung is known as a very solid business. We're business people, right? We're not traditionally known as a healthcare company. You have to look at it in business terms and say, where are those incentives? And now in the US, they're not there.
So a pharmacy benefit manager's job is to keep the price down, to keep the price of drugs down. That's literally what managing a pharmacy benefit is supposed to mean. Why isn't it happening here?
Because it's supposed to mean.
Okay. But so what is the role of biosimilars in innovation?
So biosimilars, the traditional US model, what I bought into as choosing to become a citizen, is competition. Is competition innovation. Look at the statute from 2010. Biologics Price Competition and Innovation Act. They're the driver that says, let's get the new and better products. And also, as we look to combinations, say, in oncology, and you're using multiple biologics, you need the headroom in the healthcare dollar of the cheaper biosimilar to enable that next gen of innovative products. So biosimilars are essential to innovation in the biologic space if we are to be able to afford that next generation of biologics.
So there's a virtuous cycle here that's not happening.
Virtuous cycle.
So what hurdles can we affect to change this? How do we start getting, and not just for the best-selling drugs in the world, which happen to be biologics and treat rheumatoid arthritis and multiple forms of cancer, but for all of those other products that we don't want to keep paying $1 billion a year for smaller products either, if there can be competition. How do we get what we have in traditional drugs with biologics?
The first step is probably some level of transparency. We need to know where the money's going. So PBMs are part of that, but all the middlemen are part of that. And so
the wholesaler,
the wholesaler, the specialty pharmacy, I mean, less than 10% of the list is actually going to the manufacturer.
Less than, wait, repeat that.
So less than 10% is actually going to the manufacturer and their partner of the biosimilar in general. These are hand-waving numbers. I'm a scientist. I don't like hand-waving numbers. But this is why the incentives aren't there. So if you look at us in the space, we have as many biosimilars as Amgen. We have twice as many in the US as Amgen does. But we have to be able to compete on the value of those products by people knowing what they're paying for. And the US is only 4% of the world's population. We are 60% of the biologics market. So the US has to work. If the US doesn't, work, we're not going to have biosimilars worldwide. So when you talk about the void, that's a trend that's going down. A few years ago, it was 50% of the biologics facing loss of exclusivity within 10 years didn't have any biosimilars in development. Now you're citing a report with 90. So clearly, we're on a trend we don't want to be on. We need to therefore have transparency, know we've got a competitive free market. Things like the Inflation Reduction Act, price controls, none of those are the solution if you have competition. Meanwhile, I would, from a scientific perspective, like to reduce those development times from 7 to 10 years, reduce those costs from 300 million, so you have more people in the space making the competition.
How much of those costs are scientific, ability to make the drugs faster or run necessary clinical trials, and how much are regulatory?
Again, these are real vague numbers. About a half is probably the clinical trials. And if we can bring it back to the analytics, which is how we've made manufacturing changes to originator biologics since '96, then we can be much more efficient.
I don't think people will get what you just said.
So back in '96, we faced a problem that biologics were beginning to emerge as a significant part of the market, but we needed to scale up manufacturing, we needed to replace equipment, whatever. And FDA faced the problem of how are you showing it's the same product you're making if it's in a different facility? So we developed the concept of comparability that says if I measure this, in the future after a manufacturing change, will you, FDA, agree it's the same product? And those so-called critical quality attributes became part of global harmonized so-called Q5E. This science is not new. So we established it in the 90s. For biosimilars, we've sort of done a bit of overkill with what we call these limited confirmatory clinical studies, but they're telling you nothing new. And that becomes questionable ethically because you're experimenting on human beings and it's not giving you new information. So if we make the regulatory environment consistent for all biologics, just as I asserted it was for quality now, we're more efficient. And if we're more efficient, more people can get the investment dollars to think about competing. But all of this is dependent on a reimbursement environment that values the cheaper product by giving it market share.
And what will it take to signal to companies to stay or get back in the market? Because I remember about 10 years ago, there were massive investments to get into this market. If people get out, they're not going to want to reinvest to get back in.
I think they'll reinvest if the numbers add up. Everybody's running it product by product because you've got oncology that was initially doing quite well. You've got rheumatology. It was made to be an issue with switching patients. So different therapeutic areas are responding differently. Different healthcare providers have different concerns. So I think people are running the numbers, but if originators have the premium pricing and there's a market, we, like everybody else, will look to originator products. It's actually remarkably amoral. It's business. It's economics.
All right. Well, thank you very much.
Please welcome to the stage Dr. James Wilson, President and CEO of Gemma Bio and Chairperson of Franklin Biolabs. And to moderate, Stat West Coast Biotech and Life Sciences reporter Jonathan Wosen.
Welcome to Stat. Jim, thank you for being here as well. Looking forward to this conversation. You've got, I think we were just talking a moment ago, 40 years of experience in gene therapy. You ran the oldest and largest gene therapy program in academia at the University of Pennsylvania, and you've been involved in starting a number of companies, including Gemma Bio, which I'm sure we'll talk about in a moment. So you know probably better than pretty much anybody that the space is in a bit of a rough spot right now. There are a lot of therapies that aren't reaching patients or profiting the companies that develop them. There's no shortage of examples. I think we've talked about the fact that experts have called this a great abandonment that's happened in the space over the past 3 to 4 years. What's your analysis of what's going on there? What's wrong with the current model?
Yeah, and by the way, thank you, Stat, for having me here at really an important time in biomedical research. When I think about that question, I think about it through 40 years. so maybe a different perspective. And so what we've experienced in gene therapy are cycles. And the first was the first episode of irrational exuberance prior to .com. And then there was, it sort of blew up. And for a decade, we couldn't even use the word gene therapy. We called it regenerative medicine. And then, like often in these emerging technologies, there was a more sober view of where the field was. But then at the end of the recession and then subsequently, there was a lot of money that came into the space. And I would say probably round 2 of irrational exuberance. And then there's always a correction, right? But I think what we're seeing now is more than a correction, and I'll coin a term, irrational pessimism. So what I think happened was in this more recent frothy time when there was a lot of money that came into gene therapy, rare diseases were the focus, 150 AV gene therapy companies, that in that swing that occurs, the correction and then maybe the overcorrection, that patients who could have benefited from those programs are now left with really no answer. And so they call it the great abandonment. I don't think that's fair. I think it's the evolution of an emerging technology where we go through these cycles. Now, if that's true, then we will be headed, which I think is an overcorrection. I think we're going to be heading into a mode where, a period where we'll see reinvestment and progress in the field.
So just wonder, drilling down into what's been happening lately, with all the examples of companies pulling back on programs or, shutting down altogether, sort of look at what happened with Bluebird Bio with their sickle cell gene therapy. I mean, what do you, is it a matter of not understanding where the unmet clinical need is? Is it, is it the classic issue of diseases being too rare to turn a profit? I mean, I guess what are some of the specifics that you would point to?
So despite the fact that I sort of lumped gene therapy into the natural evolution of biotech. I think as this has sorted itself out, there are some unique aspects of this therapeutic platform, and because of these unique aspects, really creating some challenges. So for example, I never would have thought that we would have been in a situation where there were FDA-approved products that were priced several million per dose, and no one's prescribing them. No one. The hemophilia A and the hemophilia B programs. What was an interesting experiment, sort of, about the same time the Sarepta product was approved, possibly less effective than the hemophilia products. But I think what this taught me was unmet need, and that genetic medicines, especially gene therapy and genome editing, while we have an enormous experience in terms of the safety profile, that there still are concerns about long-term safety. So my advice is to continue to focus on diseases of significant unmet need. If I could then transition to rare, there's a lot of disease of unmet need. In the non-rare space, there's enormous competition, but in the rare space, there isn't. So I do think that there are certain opportunities. Let me mention sort of my view of Orchard and Bluebird.
Sure.
I made a decision at the very beginning of my career that I wanted to focus on in vivo, so we didn't have to manipulate cells ex vivo. But the model that is being considered from a commercial standpoint are centers of excellence, because you can't do an ex vivo gene therapy with manipulated cells at any hospital that can do bone marrow transplantation. I think that's the way that this is starting to emerge. And that creates some challenges, in particular for families that may have to travel for their treatment, in which they're being displaced for a period of time. So there's some logistical issues, like we're seeing with not only Bluebird, but the Vertex CRISPR, on how do we then lay out a commercial model that's feasible and practical for families, because when you do commit to that product, it's essentially a bone marrow transplant and all the consequences, inpatient and then intensive outpatient follow-up.
So you just coined a new term for us a moment ago, irrational pessimism. I think you've talked in the past about companies stampeding out of this space. You're jumping in, right? You started Gemma last year. So, help me understand, why do you think, maybe tell us about what Gemma is, but why do you think you're set up to succeed where others have struggled in recent years?
So Gemma Bio is a company that we formed. I'd formed 8 other companies where I remained at Penn. Right. This was the time when we decided it was time to leave the academy and then bring our team and our intellectual property with us. When we started that journey, I really had no idea whether it was a good idea or whether it was feasible. It just seemed that it was something that someone or we should consider. And as I thought about the challenges and why products had failed, transformative effects, unmet need, the problem with the diseases that I felt were most tractable is individually they were rare. Yeah. So our strategy is to think about gene therapy and genome editing not as individual rare diseases, but as platforms. So is it possible that we could aggregate programs so that the only thing you change is the gene of interest, this gene for that disease? And if there's enough similarities, we will realize economies of scale, and we could, with more certainty, understand what the probability of success is. And then convince health authorities that this will lead to a more expedited process for getting these drugs approved. but then unmet need, and focus on unmet need. That'll drive pricing. That'll drive receptivity, and then in the aggregate, if you look at the sales of that platform, that would be sufficient to justify investment in the company and justify the development of a commercial enterprise to provide access to these products. And I do think there are some advantages to the quote unquote one and done treatments from a commercial standpoint, because you go to your hospital, you go to the center of excellence, you get your treatment, and you're done. And sales and marketing, I'm not exactly sure what relevance that has, like it normally is in very competitive situations. And so I do think the commercial model is going to be simpler, but different.
I think one thing you mentioned that was kind of interesting leading up to this conversation is you guys have been looking at opportunities in middle-income countries. Maybe you could say a bit more about how you're thinking about taking a global approach to treating rare diseases with gene therapy. You guys had an agreement with Brazil's health ministry, and wondering if that's sort of a template for similar agreements you want to strike in different parts of the world.
So, so one thing that is similar between 40 years in the academy and 5 months as a CEO is all you do is try to raise money. That's all you do. And you have to find ways to secure capital. And it's a hustle either way. And in a university, you diversify the ways in which you try to raise money. But it became clear to us, if we were going to succeed in this enterprise, it was not likely that we could rely on a traditional venture capital investment, capital market strategy to resource the company.
Because?
Just because of their receptivity of investors at this point in time. I will say one other difference, if I could.
Sure.
In the academy, you kind of know what the rules are about what you need to do to get money. On this side, the rules change. They seem to change almost monthly. But in that regard, what we saw as we announced this effort is the coming together of stakeholders internationally that want to be part of a solution for providing access to these therapies. And as a result of that, are willing, through private-public partnerships or some other creative mechanisms, to work collectively to develop these products as cheaply as we can, and then provide access to these products in a way that is affordable to middle-income countries. So we were approached by the Ministry of Health in Brazil about 1.5 years ago when they heard that we were thinking about starting a company, about a partnership where they would provide to us $100 million of non-dilutive capital and the full support of the ministry, which is Anvisa, the health authority, CMED that prices, Conitec that reimburses. It's all sort of integrated. And in return, we transfer our manufacturing technology to their manufacturing enterprise and provide a license under the public health system in Brazil. 211 million people in Brazil, and we're working to find a way to expand that throughout Latin America. And we have baked into that agreement what we would receive through reimbursement. We're also looking currently in the Middle East for a similar kind of strategy. But it's not only ways to find money, it's strategic, because if we have these strategic collaborations in which there's risk sharing, like investment, it also facilitates our ability to market those products in those regions.
Oh, please.
This microphone. Oh, thank you.
Hi, my name is Stephanie Henry. I'm a neuroscientist and freelance writer. My question in the context of gene therapy and getting access to a broader patient population, it seems like we have sort of this dichotomy where we're having these higher scale single dose therapeutics like Luxturna, for example, where you're close to $1 million dosage, but you get really great patient compliance. But then on the other hand, we can opt for repeated dosing, like in a lipid nanoparticle model. But then we're struggling with compliance, but we might get better funding from health insurance representatives or in a cash pay model. So where do you think we have to shift our energies to advocate to increase access? Do we focus on repeated dosing and shifting in pay for access, or do we focus on changing the tech or being able to emphasize the utility of compliance with a single dose mechanism?
It's a really good question. I'm on the single dose side of the equation, and I think there are two ways that we need to address it to provide broader access. One is we need to reduce the cost of developing the product. We need higher probability of success. I looked at the data from phase 1 to BLA for all biologics, for oncology, for CAR T. It's about 7.5%. For rare disease gene therapy, it's almost 20% probability of success. And if you believe in platforms, you can increase that probability of success. We have to work with all stakeholders to drive down the cost of developing these products, which will then make it more tractable for us to commercialize those products, and then the price point can be less as well. And then there are creative ways or potentially ways to be reimbursed, whether it's annuity-based or risk-based or reinsurance mechanisms. But I think it all starts with bringing the cost of the product, of the drug development down, so you have fewer failures. You know, even innovating in potency of the products, you can decrease the cost per dose from 200,000 per dose to 20,000 per dose. There's room for a lot of innovation yet in this space that I think is going to help us address that.
You know, one thing I do want to ask you about, you know, we touched on all the experience that you bring to this role over 40 years being involved in the various ups and downs in gene therapy, from irrational exuberance to the present day. You know, you've taken some criticism in the past in terms of management style. You know, we had done some reporting back in 2022 when you were at Penn running the gene therapy program that you know, essentially talking to folks who said that you could be a bit abrasive at times. And I only bring that up to ask if those concerns left any sort of impression on you that shaped how you're approaching running GEMA and the sort of culture and team that you're building there.
Well, it's the same team. We provided offers to that staff to join GEMA. 97% took the offer. Over the last 6 months, we've had 2 people leave. Yeah. With that said, this is hard work that we do, and it puts enormous pressure under people. Yeah. Not only the actual work, but the mission of what we're doing. It's almost, it's hard to go home when you think you're that close. Yeah. And through those experiences, I've learned how important it is to try to introduce as much balance as we possibly can. And I've carried those lessons for us into GEMA Bio. And in GEMA Bio, we're not operating under the umbrella of a university. We're under the umbrella of our leadership team. Right. So it has given us an opportunity to do things differently, okay. And I think do things better.
Okay. So, you know, looking 5, 6 years out, you know, what do you hope GEMA can accomplish, and how does that fit into where you see the future of gene therapy and genetic medicine?
Well, what worries me in the 5 to 6 plus range is what we're going to see is more healthcare disparity. Yeah. And when there were no solutions, there was no disparity. Right. But once there are solutions, whether it's you're too rare or you're too poor, and so I hope we can bring a number of these products in the platform forward commercially. But as we roll that out, that there's a mechanism to provide access in middle-income countries. And maybe some of these countries will take the lead, like Brazil is, and moving products forward and accessible to patients.
Okay. What's been your favorite part about making this transition out of academia and into industry?
favorite part. 24 was a tough year.
Yeah.
And extracting a group that had been together, some of us for decades, out of the university with our intellectual property, the equipment, and the space was challenging. I think what's exciting to me and to our team is we now have an international stage, which we did not have in Philadelphia at a university. And you asked me about travel. I travel a lot.
Yeah.
But rare diseases and the unmet need bring people together independent of their culture or where they live. And I think our access to the Ministry of Health in Brazil or a government in Abu Dhabi or other areas has really been, to me, quite motivating and quite exciting to be kind of a catalyst to bring these stakeholders together. And the burden of genetic disease is disproportionately in countries outside of the United States.
Yeah. Okay. Well, it'll be interesting to stay in touch and sort of track how this all moves forward. So thank you for taking time to talk to us about it. Thanks to the audience for being part of the conversation too.
Thank you.
For this sponsored conversation, please welcome to the stage Eli Lilly and Company Senior Vice President, Clinical Capabilities, Janelle Sabo, and to moderate, STAT Editorial Director, Laura Chang.
Thank you for joining us, Janelle. In the classic version of the clinical trial, there's one lead investigator at a single site, which every subject has to visit on a certain schedule. But at Eli Lilly, you've been developing a less centralized approach. Can you talk about some drawbacks of the classical clinical trial and some advantages of this newer type?
Yeah. For those of you that are not aware, traditional clinical development has been done at academic centers or clinical sites where the patients are expected to come to the site for their entire visit. In today's environment, that's not very conducive to inclusivity or to making trials broadly accessible, because the reality is most patients live 80% of their lives within 25 miles of their home, typically most of that within 8 miles of home. So if you want clinical trials to be more accessible, we need to put them closer to the patient and more in the community. And so at Lilly, for more than almost a decade, we've been working in trying to put our clinical trials more into a community-based model. We don't like the term decentralized clinical studies. It gives this impression that it's like telemedicine and everything's done away from the site. We really think it's this idea of using community providers, community doctors, and partnering them with some of the best investigators across the globe and being able to really make it much more accessible for everybody to be involved in a clinical study in their daily life.
So can you give an example of some of the people who have been left out of the more traditional approach?
So we often think about when we look at the diseases that Lilly is in, a lot of them are chronically prevalent diseases. We also have a number of diseases in our pediatric populations and also the elderly. These patients can't easily take time off of work. If they're in Alzheimer's, they need at least one care provider to likely be able to assist them to their doctor visits. That care provider is probably of working age, and therefore you're asking them to take time off of work. Sometimes these visits are 8 to 10 hours long, which means they need to miss an entire day of work at least once a month. And we know that that's not going to be conducive to people saying yes to clinical studies. And so we really have focused on how, for each of our disease states, do we take really the clinical trials to where the patients are across the globe by looking at prevalence rates and looking at individual needs in different geographies or cultures.
That's great. So now, instead of this one site, you might have, like, each trial might have how many sites involving how many partners?
It varies. So in some countries and in some geographies, what we'll find is a hub and spoke model. This works really well. I was actually just in Australia And in Australia, this hub and spoke model has really worked to involve even the Aboriginal population as well as the highly rural in the outback area. So here, what we see is really still one investigator working with multiple community providers. In the United States, that might look a little bit different. It might still mean that they have an investigator in a town near them, but some of their activities in their clinical study can actually be done in their hometown and much closer to home, whether that's an MRI or it's their blood draws or maybe it's picking up their clinical trial medication. We think there's a real opportunity to at least bring more of the activities closer to home that makes sense.
And so what are some of the innovations that have enabled you to run these more complex and distributed trials?
Technology, I actually really loved the conversations earlier. I think technology is really important to scaling high quality care. I actually think our first group of speakers are correct about that. I think the second piece is really thinking about how do we actually make clinical trials more an option for care, particularly in rare and genetic diseases, where there aren't a lot of options available. And so how do we make it more a part of the healthcare system, I think becomes really important as well. The last thing I would say is really working with regulators to adjust what I would call the less modernized frameworks of how they expect clinical trials to be done to make it more accessible to community providers has been another important aspect.
Yeah, so let's jump to that. So what on the regulatory front do you think needs to be done to make community trials even more effective?
Yeah, I'll speak about the United States, given that's where we are today. But the FDA actually, not too long ago, released a final guidance for what I'll call community-based trials. It's actually really excellent, but it has a couple of gaps from a modernization standpoint that we're working with not only the FDA, but other regulators, which is being more clear about the roles and responsibilities of investigators and community-based providers in particular is one gap that today leaves people with many interpretations, which is not very efficient or effective. There are a couple of others, but I'll sort of stop there. That's one example.
Okay. In terms of the industry too, are there things that you would like to see happen in the industry too?
Yeah, I find that right now the industry is at an interesting crossroads. I think traditionally providers and vendors have really been trying to provide services that are much more like traditional clinical trials, and I find some of the best innovation is actually coming out of, I'll call it adjacent industries, that are helping us to think of new ways to really take community-based research into the environment. And that can be anything from how we ship medicine and the shippers we use so we can easily get to patients' homes with investigational material. It could be lab providers who are providing new ways of doing labs in the communities and in clinics that make it more accessible. If we look at MRIs and imaging across the United States, think of how many of those are now in your community, typically around the corner from you, or there's probably one in your zip code. We can now use those. You don't have to go to a major center to get that done. And a lot of times they're open until 10 o'clock at night. And so really working with networks in communities to think about how would we do a trial here has been really important for us. And then using technology to sort of knit it together has been really important. Our focus has been on the patient. So we actually measure patient burden for every single clinical trial that we do, and we give it a patient burden score. We also give it an investigator burden score. And so what we try to do is look at how can we bring down the burden for any given patient in our clinical trials in each of the geographies we're conducting it, as well as optimize the time for our investigators so they can spend more time in care. And that's something we're really committed to. And so solutions are not international per se. I think local solutions are really important to also stay culturally sensitive and geographic appropriate for what works in that healthcare system.
Mm-hmm. When you're working with so many different partners, how do you ensure that they all are working in the way that Eli Lilly needs it to be?
Yeah, we have an oversight process that we have along with an entire quality system to really make sure that we maintain the right standards and that we're auditing and inspecting and qualifying every site, every vendor in every location that we're using. And then we have a field team that's located in 70 markets across the globe that actually are boots on the ground that really help us. And then with some of our important strategic partners, our stretch can go further beyond those markets. So we really have tried to make sure that we have not only central oversight of quality and the management of this network that we use, but really also how do we have boots on the ground that are Lilly employees that are really making a major difference to make sure that in the moment it's as we would anticipate as well.
So with all these different techniques, do you have a favorite example of how this kind of trial has changed the development of a certain Lilly product?
You asked me the question back in the green room, like, do we have any drugs we've put on the market that we've done this? So I would actually say most of the drugs you've seen from Eli Lilly and Company for the last 3 to 5 years were all developed this way, actually starting in phase 1B all the way forward. So the reality is we know it works. We know that regulators can inspect it. We can pass our inspections and audits from regulatory agencies across the globe. And what it's really allowed us to do is to involve more patients where they are. I think my most favorite recent example is actually in Alzheimer's, where what we know in Alzheimer's is that particularly in the mild to moderate, where it's like, I call it, the recent term I learned is worried well, but they still need a care provider. You now have somebody who's relatively well, they don't feel too sick yet. You have a care provider who's with them as their care partner. They certainly don't feel sick. And now you want them to participate in a clinical study. One, how do you find them and actually get the right imaging and blood work done to know that you found the right patient? So screening, mass amounts of screening becomes really important. And number 2 is then how do you make that super accessible to them and something that is approachable for them, given that many of them are still working, actually. They haven't yet even retired. And this is actually how we do a lot of our Alzheimer's research today is actually through community-based delivery. And I think the important part about community-based trials is we're in the community doing testing and screening, and patients keep their results. So whether you choose to do a trial with us or not, you're going to learn about your health. You're going to learn about your disease state and maybe other disease states, because occasionally we find that they have other things they didn't know they had. And that informs them, and we just think that makes healthcare better for everybody, regardless of whether you choose to participate with us or not. And we think that has a lot of health equity for the communities that we participate in as well.
Well, thank you for leading us through all those wonderful changes. Thank you for your thoughts. Thanks for joining us. I think that's all the time we have. Thank you.
So, some of you may have seen an article from stat reporter Jason Mast called "The CRISPR Companies Are Not Okay." And if you didn't see it, you should really check it out. For that story, he conducted over 75 interviews about the downturn in funding in the gene editing industry. And today we are going to hear from one of the scientists in that story about the potential of CRISPR in medicine in the future of the field. So for this session, please welcome to the stage Dr. Kiran Musunuru, Professor of Cardiovascular Medicine, Genetics, and Pediatrics at the Perelman School of Medicine at the University of Pennsylvania, and to moderate, Jason Mast, General Assignment Reporter at STAT.
Kiran, thank you so much for being here. So I don't know if we have any people who are invested in CRISPR companies in the audience right now, but if you are, I'm sorry. Last couple of years, I guess, we've seen valuations plummeting. We've seen layoffs, widespread research programs cut. Even as there's been some really remarkable successes, the sickle cell treatment, like among others. You've been in the field since the early days of CRISPR, leading this field. You've co-founded one company. You're, I'm sure, working on others. You're doing research to build new medicines. Tell me a bit about what is happening in the field. Why are we seeing this, I don't know, deflation or this mass layoff, I guess? I don't know.
Sure. Well, let me begin by thanking you, Jason, thanking STAT for inviting me to be here. It's a pleasure and a privilege. And to get your question, I'm really mindful of what Dr. Wilson just said a little while ago, the terms that he introduced: irrational exuberance and irrational pessimism. And we're very much in the second phase now. And you know, in each case, it overshot where reality should be. And so 5 years ago, we were in a situation where even if you had a fun new tool that looked good in cells. You may not even have mouse data, but you could garner enough enthusiasm and excitement that, hey, maybe this is the next big gene editing platform, that you could actually raise a substantially large Series A and start a company and hire a lot of employees and actually get going. And then reality hits and macroeconomic situation changes and interest rates go up and it becomes much harder to bring in new capital, and these are all expensive endeavors. It takes time. It takes many years, and it takes tens, if not hundreds of millions of dollars to actually make it to the clinic and have that first milestone. And when money is expensive, you simply cannot sustain it. And so we're seeing a lot of companies that just were not built to actually make it to the clinical trials of the clinic in a reasonable time frame, just run out of funds and not be able to raise more funds. And now we're in a situation where there are meritorious ideas, perhaps, but it's much harder to raise money for those meritorious ideas. And so there's going to be a delay. And the hope is that in the next few years, things will rebalance and we'll get to the right place. We won't overshoot the other direction, but it'll be right where it should be.
Yeah. You talked about sort of interest rates, changing microenvironment, overenthusiasm, companies that, and then investors lose patience, priorities shift. Have we also seen any sort of scientific challenges? I remember like early days of CRISPR, even a few years ago, people talked about muscle diseases, eye diseases, neuron diseases, all these conditions that we were going to treat with gene editing. And mostly that's kind of all gone away, and everyone's kind of working on a few very small handful of conditions in the liver where we can treat sort of get to relatively easily. Has that, what changed? What caused that?
Yeah, well, so let me point out first that what has changed in the last 5 years. 5 years ago, we didn't have anything in the clinic. Now, 5 years later, and this is really remarkable if you think about it, if you take a step back and not really worry about what's happening to the, on a day-to-day, month-to-month, year-to-year basis. We have remarkable therapies, right? We all appreciate sickle cell, that's ex vivo, so I don't focus as much personally on that. But even in vivo context, we have Intellia with transthyretin amyloidosis. 4 years ago now, showed what to me was stunning data. It worked. There was no ambiguity.
It's a heart condition that affects 5,000 people around the world.
And it's not a rare, it's not an ultra rare, it's not that sort of, it's a relatively common disease as diseases go. And basically on the first try, and the very first few patients they treated, you saw dramatic reductions in transthyretin levels in the blood, which we know are going to be linked to improved clinical outcomes. Again, Intellia, hereditary angioedema, their second target. They've actually done a randomized clinical trial that shows substantially improved outcomes, and you can't beat that sort of evidence. Did they cure every patient? Perhaps not, but maybe we're setting expectations too high when we expect a CRISPR therapy is going to be a magic cure-all and not what we should be viewing it as, which is an improvement in human health. You have Verve, and by way of disclosure, I was a co-founder and continue to be an advisor to Verve, but with hypercholesterolemia, which drives atherosclerotic cardiovascular disease with base editing in the clinic, and showing, again, that the technology works. So we have these very...
You can stop a leading cause of heart disease.
Exactly. And so we have these examples where the technology clearly works, right? And so there's a disconnect between the technology, in my opinion, and the financial, economic context. But I do take your point. That was your question, right? We can get to the liver. I think it's not a small thing that we've been able to do some things in the liver. Let's not, understate that. But, at the end of the day, there are challenges delivering to other organs in the body, right? And so how do you most effectively get gene editing tools that we know will work? How do you get them effectively and safely into muscles? So you could actually target the heart muscle directly, which as an adult cardiologist is very near and dear to my heart. I just saw myself walking into that pun and I apologize. Or muscular dystrophies, if you're talking about the skeletal muscles. How do you get to the central nervous system without having to inject directly into the site that you want to target? And those are challenges that are being very, very proactively worked on, and progress is being made both with viral vectors as well as non-viral vectors. So we will get there, but there needs to be patience. And part of the disconnect is, to put it bluntly, lack of patience on the part of investors. Yeah. They don't want to wait for 5, 10, 15 years for all this to unfold. They want something to get into the clinic very quickly. And that's just not the reality of the situation. Yeah. Maybe for some things, like getting into the liver, now that we've de-risked the technology, we've shown some success, maybe there is the opportunity to do things much more quickly and get to the clinic much more quickly. But everything else, there's going to have to be some patience. And we will get there, I promise you that.
Your work is largely focused on the liver, at least at the moment, where yes, everyone is basically working on the liver if you're working in CRISPR right now. But there's many diseases that are not being treated with this. There's many conditions that affect the liver or that derive in the liver. Why aren't people going after that? Some of these very rare, serious conditions, and what are you doing about that? What is your vision?
Yeah, and so some of this will parallel what Dr. Wilson was talking about earlier. So it comes down to the economics. So at least in the traditional model of drug development, and particularly as it applies to gene editing, it's very much about find that one opportunity or a couple of opportunities, deploy your technology, do all the preclinical development, make what I would think of as, the optimal drug. So with all the bells and whistles, try to push the technology to the utmost, make it the most effective, most safe drug you can possibly imagine. And then you're also running into the problem of this being an experimental technology that has not had, much experience in the clinic, right? Anytime you're starting with something new, the regulatory bar is going to be higher, as it should be. And so what ends up happening is it literally takes hundreds of millions of dollars just to get to the clinic. Forget about doing phase 1, 2, 3 clinical trials and getting a BLA and all of that, just to get to the
Just
clinic.
the mouse work, just the lab work.
Just all of the work. Hundreds of millions of dollars. But now that the first examples have proven out, I think things will change where instead of taking several $100 million, maybe it becomes $20 million. But that doesn't fully solve the problem, of course, right? So if you have a patient with a particular rare condition or ultra rare condition, or maybe they have a unique variant, it's unique to them, maybe their family, that's causing some ultra rare disease, so what we would consider an N of 1 scenario, obviously doing it for $20 million isn't going to work either. So this is where you really need to take full advantage of gene editing, and in particular CRISPR, to deploy it as a platform. And that's, again, something Dr. Wilson mentioned earlier. And the beauty of CRISPR is that the editor can be the same, and all you're changing is the guide RNA, the portion of the therapy that's basically the GPS that tells CRISPR where to go in the genome. And CRISPR doesn't care, I'm anthropomorphizing, but it doesn't care. It doesn't know what gene you're pointing it at. It doesn't care whether you're treating alpha-1 antitrypsin deficiency or hypercholesterolemia or a urea cycle disorder or organic acidemia or PKU. It doesn't care. It's going to go where you tell it to go. And all you are changing is that GPS component, the guide RNA, and the overall delivery vehicle, the actual drug that is put into the bloodstream and is where the toxicology arises, because it's the interaction of that drug with the body that's really going to cause any safety issues, if there are any safety issues. That's going to be basically the same. And you can give it once, you can give it twice, you can give it for a few different disease indications. Eventually, you're going to know, hey, you know exactly what happens when you put that drug in.
You're saying that you can go build one gene editor for one liver condition, for one ultra-rare mutation, and then take the same thing, tweak one small part, and with very minimal work, go to the next ultra-rare variant, next ultra-rare variant. And suddenly, instead of $20 million to treat one disease, you have $20 million to treat 100 or whatever
Exactly.
the number is.
Exactly. I think it does lend itself to that scalability. Now, part of the challenge is a regulatory challenge. It's just a new way of thinking. And so working with the FDA, working with other regulatory agencies, there has to be buy-in on all sides that this is a viable model, that this makes sense. But all the language that has been coming from the FDA recently has been very supportive of the idea of deploying gene editing as a platform, particularly if you're using non-viral vehicles like lipid nanoparticles. And so we've seen success in the clinic. I mentioned a few examples earlier, but there's no reason you can't take that same exact lipid nanoparticle, the same messenger RNA, maybe tweaking a little bit, changing the edit a little bit, and then all you're doing is you're just swapping in and out guide RNAs, and then the cost to develop the therapy comes way, way, way down.
And you're doing this yourself right now. You are making custom gene editing treatments for patients right now, yes?
We're trying to, yes. And so, to give you some context there, the companies, quite understandably, are looking for the sweet spot. They're looking for maybe rare diseases, but more common rare diseases, if that's not an oxymoron, where there's enough of a total addressable market to make sense to deploy hundreds of millions or maybe soon tens of millions of dollars to make that sort of optimized drug for that one population, right? One drug for a relatively large group of people, several thousand, several tens of thousands, and to them it makes economic sense. No company, at least at the current time, is actually going to focus on rare, ultra rare, N of 1 type scenario. So it's left us on the academic side to do it. And so the approach we've been taking And there's a question raised earlier during Dr. Wilson's talk about access, and I don't pretend I have any sort of answer to access, right? That's an economic question that's way beyond my expertise. But I would note that in this space, the ultra N of 1 customized medicine space, the stories that you hear are about motivated families with children who are affected by very, very severe, grievous genetic disorders. And they raise money from the community, from their friends, from whoever they can, and they raise millions of dollars, and that is enough to actually be able to deploy a customized drug. And it's been mostly with antisense oligonucleotides so far. But gene editing has the same potential, if not more potential. But I worry about that model because you're putting all the burden on the patient or the patient's family to make it happen. And
not everyone can raise $3 million.
That doesn't sit well with me. So our approach has been really patient-centric. And so we've been focusing on a set of diseases, ultra-rare diseases, urea cycle disorders, and the related organic acidemias. And I'm at the University of Pennsylvania.
These are very rare conditions that often cause ammonia to build up in a young child.
Exactly.
Hurts the brain, and you really need to treat immediately to have an impact.
Exactly. They're sick on the day they're born. They're already suffering irreversible brain damage, and they basically stay in the hospital or cycle in and out, and they get decline over time. And they can get a liver transplant to correct the metabolic issue, but they typically can't get that till they're a year old or older because they have to be large enough to actually accommodate the transplant safely. And so I'm at the University of Pennsylvania, but I'm also affiliated with the Children's Hospital of Philadelphia, CHOP. And so what we decided to do, my team, a couple of years ago, is to ask, okay, who are the patients with these diseases coming to CHOP? Let's track every one of them, each and every one of them as they come. It doesn't matter where they came from, what their circumstances are. They have one of these diseases, and it's about a dozen different disorders, not one disease. And they all have a gene, and they'll have a set of mutations, typically 2 mutations. These are recessive disorders. And regardless of what the variant is, regardless of what the gene is, regardless of what flavor of disease, we know that if we can fix that in the liver, then we should be able to make a difference. Maybe not cure them, I shy away from that term, but at least make their long-term outlooks much, much better, maybe to the point where they won't need a liver transplant, right? So instead of a death sentence, there is at least some prospect of having a reasonably good life. And so as each has come in, we've been sort of working on the editology, if I may use that word. And so the first patient who came in, that was about 2 years ago, we said, okay, they have this particular disease, they have this particular variant. How quickly can we solve it? How quickly can we come up with a reasonably good gene editing solution? Not a perfect one, because that can take years, but at least one that works well enough in cells in a dish and maybe in a mouse model that we're fairly confident that if we were to deploy it in a human patient, it would actually work well. And I'll be honest, we didn't really know what we were doing because no one had ever done this on, with the clock ticking. And so it took us more than a year to figure that out. Not really compatible with the time frame to actually help the patient, or at least, be able to forestall having to undergo a liver transplant. But then we learned. And so the second one, right, we get more experience and we solve it and we figure out where the pain points are. We get quicker and quicker. We just get more sophisticated in our editing techniques, our editology. And so, the third one, the fourth one, we learn more and more. By the time
How many are you on so far?
Yeah, so we're up to number 7, number 8 at this point. And, with number 7, almost to our astonishment, we realized that we were able to solve it within a month of the patient's birth. And that was so quick, we realized, wow, if we can actually solve it this quickly, that's the big first hurdle, finding a bespoke editing solution for the patient. If we can actually get it done in a month, then what's the next step? Well, the next steps are really twofold. It's manufacturing. How do you actually get that drug made in a way that's, a reasonable quality and safety standards?
Treat the patient, I assume, right?
And then you have to get regulatory approval, so you can't overlook that. So twin challenges, but potentially surmountable ones. And so then after we finished that one and figured out, okay, in a month, then we made a customized mouse model, not knowing whether, the FDA would want to see that or not. It's not something we'd be able to do for every patient.
Uncharted territory.
Uncharted territory, but, we're just trying. We're making cell models, we're making mouse models, we're doing off-target editing analyses, we're experimenting with manufacturing, just trying to see where we're going. And what we have figured out is that we think we can get this down to 6 months, at least initially, and then as we get better and better, this really streamlined. So that is a time frame that is compatible with actually making a substantial difference in these kids' lives. And so we're very excited.
I talk to people around the field about this, in companies, outside of companies. Everyone kind of agrees this is where you need to go. You need to get to a place where the amount of work and money that required to get a gene editing treatment into the clinic is vastly lower than it is right now, so you can get to all these rare patients. And where they differ is on the timeline. Some people think, I think like, as you're arguing, we can do this right now. And some people think this is perhaps understating how hard or how much work is needed, and that lowering the regulatory bar, can you end up in a safety event that like sets back the whole field, hurts a patient? How do you think about those trade-offs or that challenge?
Yeah, so I would make a couple of points. One, we cannot let perfect be the enemy of good, right? You cannot make the perfect drug. That's not feasible here. It's going to be too expensive and it's going to take too long. You just have to come up with something that works well enough that you're confident will work in the patient. And again, it was brought up earlier, you can do repeat dosing with lipid nanoparticles. And so you don't have to get it all in one shot. You can give the first dose and maybe get some improvement and then give a second dose sometime later. So you can scale, you can dose to effect. So you have some flexibility here. But the point is that you don't need to make the perfect drug. You just need to make something that is good enough. The other point I would make is that we need to think of these rare, ultra-rare, devastating metabolic disorders more like cancer. Like none of the cancer drugs that I know of work 100% of the time, right? There are always failures, and we accept that, we understand that. These are not cures. These are treatments that hopefully will help a lot of patients who receive them, but it's not going to help 100% of the patients.
And the risk tolerance is high.
And the risk tolerance is much higher. And why are we not treating these kids or maybe adults with devastating genetic conditions with the same lens, right? And so if you shift your thinking in that way, you don't need a perfect drug, nor can you expect perfect outcomes, or should you expect perfect outcomes. Maybe we're actually not so far away from being able to do this.
I hate to end on a negative note, but I would be remiss in not asking, and this is government-funded research mostly that you're doing. You are at Penn. NIH has been cutting indirect costs, for example, which is a significant portion, I think, especially for Penn. And yesterday there was an announcement about, apparently, no one knows exactly what's going to happen, but 175 million, I think, in cut dollars for Penn by the Trump administration. How does that affect your work? Does it put it in jeopardy?
It won't help, that's for sure, right? And so, to follow up on a point you were making, no company is really going to take the risks that are inherent in doing what we're doing on the academic side. It's really on the academic side that we have to be the tip of the spear. We have to be the ones who say, you know what, we're going to take that patient in front of us, whoever shows up in the emergency room at CHOP, and we're going to deal with them on their terms and try to fix their condition, and do that with every patient who comes in. No company is going to do that, at least not until we've de-risked it and show we have to be the tip of the spear. And the only way we can do it is with public funding, with federal funding. So the NIH is absolutely instrumental to what we're doing. We would not be able to even think about what we're doing without strong, strong support from the National Institutes of Health, like without question. And so if that pipeline is cut, or the flow is decreased, or it's in any way compromised, that is going to affect what we do. Now, whether that $175 million will directly impact my team's work because the funding will get canceled directly, and then we won't be able to help make life-saving personalized therapies for devastatingly ill babies, that's a possibility. But even if it doesn't directly affect the work that we are already doing, it will affect the infrastructure that makes it possible for us to do, right? So the funding we're getting, make the drug, do this, do the off-target analysis, that's one thing. But then the infrastructure we have at Penn, at CHOP, that's funded by indirect costs and other sources of funding.
Lab people who work with the FDA.
Like without all of that, we can't do it. Like I spoke about the importance of being able to work out a regulatory strategy, work with the FDA, streamline the process. You need people who have that expertise on the team. And that's not something that's funded out of traditional grants. That's the sort of thing that has to come out of indirect funds. And you take that away, then it makes it much, much, much harder for my team to do what it's doing. And you're just pushing back. You're hurting everyone when you do that. It pushes back the prospect of breakthrough, life-saving treatments and potentially cures for everyone in this country. And I can't imagine that's really the intended effect of what is going on right now.
Thank you so much, Kieran. Really appreciate this.
And with that, we are at our first coffee break. So right back outside of this room where you found your breakfast this morning, there will be snacks, coffee, and tea. I'm very excited to have some hot tea. There are also some smaller stations with the same items right outside of the back doors behind you. And also, please check out our sponsor and stat booths that are at the perimeter of the room in the back. We will resume programming here at 10:50. Thanks so much.
Right, let's see if my mic's on. Can I be heard? Is this thing on? I'm gonna keep getting louder. All right, I am going to, I'm speaking now because I have something to say before we actually start, which is, I've seen people crowding around the back. I know it sounds really embarrassing to come up here and sit at the head of the class, but there are a whole bunch of seats up here and they're the best seats. They really are. So we are going to get started again in a minute, and if people could filter in, and for anyone who didn't have a seat, there's a little area here that's kind of been a dead zone. We'd love for people to go ahead and come up and find some seats. Again, we're going to be getting started in a minute, and would love to fill in seats in the front. Seeing some cool people arrive, that's neat. So, just filtering in, want to start on time. We know the coffee break got caught a little short. It'll all still be there if you want to sneak over and get more caffeine. But, all right. So, welcome back. I hope you enjoyed some of the refreshments. I hope you got your choice of caffeinated or uncaffeinated beverage. I'm told hydration is more important than caffeine, but I don't practice that myself. Up next, absolutely one of, if not the top reporters in the biotech space, certainly at the intersection of biotech and Wall Street. If you want to know what's happening next, Adam Forstein's usually a good person to ask, and up next he's going to take you on a biotech roller coaster ride to talk about a disease that's been the next big thing for years, and to talk about why it finally is the next big thing. So please welcome to the stage Carol Huntsman, the Chief Commercial Officer of Madrigal Pharmaceuticals, Kitty Yale, Chief Development Officer at Akero Therapeutics, and to moderate, Stat Senior Writer for Biotech, Adam Forstein.
Hey, everybody. All right. Carol, Kitty, thanks for joining us today. And, so we titled this session, Finally MASH's Moment, because MASH is having this moment, a good moment. I feel like I'm writing a lot of stories about biotech in crisis these days, but this is a good one. But then finally meaning it's taken a long time. And we were joking backstage, it has taken so long that the disease actually changed its name during the time when these drugs were being developed. And obviously now we have the first one approved. But let's sort of set the stage and let's start. And maybe, Kitty, you're the drug developer on stage. So let's kind of start talking about MASH. Just give everyone here a sort of a thumbnail sketch of what we're talking about when it comes to MASH.
So, I think MASH is a metabolic disease, and it's a metabolic disease that evolves over many years from, starting really with excess calories, moving into metabolic syndrome, diabetes, dyslipidemia, that eventually leads to accumulation of fat in the liver. The fat really starts a sort of inflammatory cascade. The liver lays down collagen to protect the hepatocytes. It gets stiffer over time. You end up getting to the point where it goes from what we call MASLD to MASH, and then eventually you get cirrhosis of the liver, where really, at one point cirrhosis, if you look at the definition of cirrhosis, it's irreversible fibrosis of the liver, which is obviously...
And fibrosis, we're meaning scarring of the liver, right? Yeah.
Yes. Mm-hmm. And then ultimately these patients end up in end-stage liver disease.
And I know this is a disease that's often associated, as you mentioned, with obesity.
Yep.
Colloquially we call it fatty liver disease, or associated with fatty liver. But why is it not only an obesity? I think people sort of commingle those things, right? Why is it not just a disease of obesity?
Do you know, I think it is because the metabolic dysfunction comes in multiple forms. And so especially, you know, if you look at it as a global disease, there's many countries in the world that has very high prevalence of MASH with very lean patient population. So lean MASH it's really the same disease, which ultimately ends up with dysfunction in the liver, but not necessarily associated with obesity.
Now, Carol, you are the Chief Commercial Officer of Madrigal. You are in charge of selling the first. Yes. So congratulations. It's about actually, it was a year ago.
A year,
March
2020 last year.
Right,
yes.
of having the first drug approved. You know, the disease has kind of been around for like 40 years, and maybe like the last 10 plus years is kind of, I think, more on the radar screen of people. Tell us a little bit about Rezdiffra and you know, what it's been like in these initial days of selling this drug.
Thank you. No, so very exciting time because, as you said, it's been a long journey. Over the past 14 years, there have been more than 20 products and companies that have failed in terms of getting registration for a product and bringing a solution for patients to market. So super excited that Rezdiffra is the first, and now there's a solution for patients. Rezdiffra is a once-a-day pill that's very well tolerated, that reduces both the fat in the liver as well as the scarring or the fibrosis in the liver, and has been studied and is approved for F2, F3 patients. These are patients that are at risk. They're 1 to 2 steps away from cirrhosis, meaning could be liver transplant, could be hepatic cancer, could be death. So these are patients who are really at risk and need intervention, and now they have some.
And tell us, because you mentioned F2, F3, and it gets kind of into the weeds of, you know, and I had to learn a lot of this when I was writing about this. But like generally, like give us sort of a sketch of like a patient that is that you're treating these days. Like what like symptom wise and like in the sort of stage of the disease, where are they?
Well, that's the very interesting thing. Oftentimes, these patients don't have any real symptoms of the disease. These patients are moderate to advanced in terms of their progression of disease, but they don't necessarily know it. They might have an abnormal liver function test during a checkup. They may have some pain in their left quadrant and might go into the emergency room, might have some imaging and be found there, but often these patients don't have any symptoms at all.
And you came from Sanofi, right?
I came from Sanofi.
And actually, Bill Siebold, the CEO of Madrigal, also came from some of you. I think you told me you were his first.
I was his first hire. I had the opportunity to work directly with Bill for 11 years and was running North America and across the therapeutic areas and the country lead at Sanofi before I left.
So Bill's probably famous for being in charge of selling Dupixent, which is obviously a blockbuster medication for Sanofi and Regeneron. He came over, he brought you over. Talk to us a little bit about the challenges of selling, you know, kind of the first drug in a new disease class.
Well, I've had the opportunity to do about 25 launches in that 11 years in Sanofi. 6 of them were first in class, Dupixent being one of them, the first in class for atopic dermatitis. But there are a lot of questions, right? There were questions about whether the FDA was going to require biopsy, whether payers were going to require biopsy. And then there's a lot of education that needs to happen, a lot of, you could call it wiring the system or infrastructure building in terms of getting providers ready to treat those patients who exist, who are out there with moderate to advanced MASH. But it can be so rewarding because these are patients who can suffer devastating effects, and now they actually have something effective that can keep them from the liver transplant, from the hepatic cancer. So very exciting.
You know, getting back to the obesity question and how this sort of interplays, I mean, go back to Kitty. Talk to me a little bit about staging here, like how, because I think one of the issues that have come up and one of the challenges is like how you stage patients.
Yep.
You know, in the clinical trials, you use liver biopsies,
Yep.
which obviously is an invasive procedure, but there's also non-invasive ways.
Yeah. So going to Carol's point, I do think we're still in the era where a number of patients are absolutely shocked that they can get liver disease that's not alcohol related. And so, you know, you meet patients who really have no idea that, you know, their choice of diet and their lifestyle can actually lead to liver disease. And so we're still living in that era where these patients are walking around really having no concern for their liver. And so when they present, often, as Carol said, the patients will really have no direct symptoms, and suddenly what will happen, they'll go to their primary care physician, and suddenly their liver enzymes are at an abnormal level. And so what generally happens after their liver enzymes are raised, and that's really a marker of sort of liver injury. The first thing that normally happens is they do a FibroScan, which is basically an ultrasound of the liver. And basically it measures the liver stiffness. And so they sort of do those two tests and really often then realize that this patient is looking like they probably have liver disease. There are also some blood biomarkers that can be run that again can help stage the patients. And they'll run the patient through an algorithm and really then decide, will this patient receive a biopsy or not, or whether. And I think it's interesting. We're in this unique situation where the regulators are really requiring us to use biopsies. And I mean, a liver biopsy is not the most pleasant experience to put these patients through. It involves a very long needle, and these patients are.
I don't want to think about long needles into
Yeah.
the liver. Yeah.
On the larger side, so it's a complicated procedure too. But then in the real world, what we're really allowed to use what these are called as non-invasive tests, and so really what I think is happening now is there's a triaging of patients through the non-invasive tests. If they're coming into a clinical trial, the patients will ultimately end up being biopsied. And if they're going on to Rezdiffra, they're being followed non-invasively. I don't know about you, Carol, but I feel that the non-invasive space is really, moving on really quite rapidly right now. And I think, for a long time, there's been drugs that have been studied in MASH that maybe weren't as efficacious as others, and they were struggling to really define patients as a responder or a non-responder non-invasively. But as we're generating more data, I think that's becoming clearer. And I think, none of them really, unfortunately, have a sensitivity that they can really be used on their own. And I think what you're seeing is we're looking at overlapping tests. And really, I think that could be a way of the future where, we look at overlapping non-invasive tests to really stage the patients. And I mean, and I think it's the staging of the patients is going to be really important. In the future, we hope there will be multiple drugs to treat these patients. And then the patients, based on the other comorbidities and their staging of liver disease, will take the right treatment.
So I should note, two companies developing drugs for MASH, two different mechanisms. Kitty, I want to give you a chance to talk about EFX, which is the drug that Akero is developing, and obviously had some really pretty amazing data just recently in the most severe patient population. So tell us a little bit about those patients, those specific patients, and kind of what the data showed with EFX.
So I've been working in this space for a long time, and I was actually at Gilead when Gilead released one of the first studies in 2019, really looking at the compensated cirrhotic patients, and really was one of the first big failures. And so the compensated cirrhotic patients are one of the most advanced treatment groups. And it's really got to the point where there's so much scarring in the liver that the liver is really starting to not to be able to function properly. And if you read the definition of cirrhosis, it's really thought, and many doctors are trained this way, it's a level of scarring that's irreversible, and that once that you have cirrhosis, you really are unable to come out. And also, it's a very unfortunate diagnosis. It's akin to really an oncology or a cancer diagnosis, because the mortality rate for those patients, there's about 50% of the patients will unfortunately die within 10 years of their cirrhosis diagnosis. And so, it's really, a state of a disease where these patients really, when you get that diagnosis, there was really no hope for the patients in terms of, really they felt they were going to follow this cascade of, liver disease into end-stage liver disease and probably require a liver transplant. So efruxifermin's it's an FGF21. It's a hormone that regulates metabolism, particularly glucose and lipid metabolism, and really has a multi-pronged mechanism. It's very effective, as we said. It's a direct-acting anti-fibrotic drug. So we've seen, levels of fibrosis drop in what I would call relatively short periods of time. It also defats the liver. It also sets off a cellular pathway that, protects the hepatocytes. And really, by those two mechanisms, you're removing the assault on the liver. And so it's like the fundamental driver of NASH. And so what you actually see is we're seeing both MASH resolution, so the fat removed from the liver, and the direct-acting antifibrotic. And so it was great to be able to show after 2 years of treatment, for the first time really ever, that we've seen reversal of cirrhosis for this patient population.
And that was the significance there was, again, like you said, that you don't normally see reversal, right? No. You might see, we talk about a lot of progressive diseases where the best you can show is slowing of the progression of the disease, and we declare that a victory. This is one where we actually saw reversal.
Yes.
Just getting back to you, there's a school of thought, and there are people out there who sort of doubt MASH, and they say this is just, these are people who suffer from obesity, and just give them a GLP-1 and they'll be fine. Talk to me about that in the context of selling a drug for MASH, and like how, what is the role that GLP-1s play in this disease in terms of marketing and the treatment of them, and kind of what have you seen in the year that you've been selling Rezdiffra?
Well, first I will say I agree with Kitty that multiple ways of working or mechanisms are needed in this space. But today, for Rezdiffra, about 50% of the patients have been on a GLP-1, and about 25% today are used concomitantly. So GLP-1 for one of the comorbid conditions like diabetes or obesity, and then Rezdiffra for MASH.
So they're on a GLP-1, they're losing weight, but the damage that's being done in the liver is not being addressed by the GLP-1s.
Well, I think what we need to think about is time is liver, and these patients are really at risk. They're 1 to 2 steps away from cirrhosis. They are the catastrophic types of events, mortality that you see in cirrhotic patients, and even death. So they need to be treated with a liver-directed therapy. Rezdiffra is a thyroid beta agonist that works to reduce fat and also scarring in the liver in F2, F3. That's what it was approved for. And these patients, they need to have access. So I think the way that providers are thinking about it is they have different ways of working. There's a place for both. And from my perspective, the most important thing is patience. And we need to make sure that there are multiple effective therapies that patients can access.
So you're finding insurers who will willingly, are they reimbursing for both at the same time? But I know that was one of the questions coming in. Maybe the patient would try, a doctor would prescribe a GLP-1. If it didn't work, or they went off for side effects or whatever, they would then go on to a more liver-directed therapy. But are you actually finding that you are getting reimbursement at the same time?
So I think the reimbursement question was very significant coming into launch, meaning would they require a biopsy, and how quickly would they cover these patients? 80% of commercial patients were covered 3 months early. And why? Because I think the payers understood the need, the significant need that exists. And because GLP-1s have an indication for comorbidities, we've seen, as I said, about 25% of the patients actually happen to be on both today. And many providers do look at the different mechanisms and want to see that.
And is there like a sweet spot in terms of the kind of MASH patient you think that you're targeting?
So for us, it's really F2, F3 patients today. We see Rezdiffra because of its really optimal real-world profile in terms of once-a-day pill, very well tolerated. We see that 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 uptake from physicians is quite good. I think today it's F2, F3. We also have a study in compensated cirrhotic patients that's fully enrolled. And so we see that we'll have results from that study in 2027. And we see that Rezdiffra, because of its profile, including the significant efficacy, it can really be a foundational therapy for F2 through F4C.
So these days, Madrigal is judged quarter to quarter by your sales. And so far, you've done really well. The sales have been phenomenal. What can you tell us about Q1? Q1's about to end. How's Q1 looking?
Q1, well, I can tell you that we're continuing to execute very well, and we're seeing, steadily increasing providers, prescribers, and patients on therapy. So, and that's despite the typical Q1
You don't want to
dynamics.
pre-announce sales?
I don't want to
here?
pre-announce sales here today.
I tried.
Nope, can't do that.
Investors, I tried. I tried to get her to pre-announce sales. Okay.
Yes. But we feel very confident about where we are in terms of our trajectory and how we compare to some of the best launches out there in specialty medicine over the past 10 years.
So Kitty, tell us, we talked about the results that you had in the cirrhotic patients. That was a phase 2b study.
Yep.
You have a full phase 3 program underway. Give us a little thumbnail sketch of that phase 3 program, and when can we expect data?
So we have it's a 3-study phase 3 program. So, I think probably appropriately the regulators are really wanting us to generate a large amount of data across the patient population before you can get a drug approved. So the 3 studies are running globally at the moment. We have one study in, as Carol mentioned, the non-cirrhotic patients. Those are fibrosis stage 2 and 3. And then we have one study in the compensated cirrhotic. And then we have one study that we're really just looking at non-invasive methods of monitoring disease response. So really thinking it as a sort of a real-world setting. So we're of the 3 studies, the real-world study is fully enrolled. We announced that in January. And so we'll have that data the first half of next year. The F2, F3 studies coming right behind. We'll have it in the first half of 2027. And we're just a little bit further behind on the 3rd study. We haven't actually committed to when we're going to have that available. I'd like to get all of our global sites enrolled. It's funny when we commit something to the street, we like to, under promise and over deliver.
Of course.
And so it's always that balance of making sure you don't commit to something until you've got enough data to support it. So
I know there was talk. Well, one, I wanted to ask you about the impact that the mid-stage study has had on enrollment and what that's at. But also, like there was some talk or speculation or maybe desire that those results would help spur conversations with the FDA about maybe accelerating timelines.
Yeah, so I think if everybody always asks you, like, how do you make clinical trials go faster? And everybody, and obviously a great team and will make a difference, but really data drives enrollment into studies. And so, you're always fortunate when you're working in a drug which has differentiated itself in the data, and that's what will drive enrollment forward. And so we are seeing, I actually have just been traveling a lot recently, and I've been out sharing the data, and it's very nice because just because we released the data here in the US, sometimes you think that means like the world knows. And so I've been over in Europe, and it's been very nice when I've gone to speak to people and I'm like, oh, let me share the data, and they're like, oh, you don't need to share us the data, like we know the data. So it's good news does travel fast. But yeah, we're in this unique situation in MASH where the regulatory guidance is a little bit bizarre, and again goes against maybe what some people would think is common sense, that there's what we call an accelerated approval pathway available for the non-cirrhotic patients, but for the cirrhotic patients. And basically maybe what that accelerated pathway basically says is that the FDA are willing to accept a biopsy endpoint as a surrogate to long-term clinical outcomes. But they haven't quite accepted that yet in the cirrhotic patient population.
That's sort of counterintuitive, right? Because these are the most advanced. You usually think the FDA is more lenient, flexible with the most advanced
Yes,
patients.
the patients at most risk.
Yeah.
Yes, and so there's guidance out there, and so we are going to be having conversations with the agency regarding the guidance and updating the guidance. I mean, and that can be done. There's great examples. When I was at Gilead in hepatitis C, we changed guidance with data. The question really is whether the phase 2b data would be enough to change guidance. What we have been allowed to do is include biopsies in our phase 3 cirrhotic study, and it may be that it actually will need to be that large body of data that would be enough to change guidance. And so we're really hopeful that we could go down that path. But really, the regulators are pretty standard in how they respond to things. They don't negotiate ahead of time. You know, you bring the data, you discuss the data, and that's the point where things will change. It's not going to be ahead of the data.
You brought up accelerated approval and the versus outcomes, and I think that's an interesting topic. And you still have I mean, you're approved based on, you know, MASH resolution, lowering fibrosis, reducing fibrosis, but that's essentially accelerated approval. You still have to do an outcome study to show. Tell us a little bit about what outcomes means in this patient population.
It basically means that we need to do, you know, a biopsy-driven event. A biopsy at the end, but really looking at the mortality, the liver-related events. So reduced progression and mortality-driven events like transplant, ascites, HCC. So really looking to see if you're treating those patients, what is the impact on their outcome at the end of the 4-year period. We are hoping that with the F4C study that we have ongoing, and that's going to read out in 2027, that would also give us full approval if there's an impact on outcomes there.
And timing on outcome study, what's a broad range of timing in terms of when we might see
the
outcomes data?
F4C study, that's 2027.
Okay. And what, assuming that if it's positive, what impact that has on the market? Like what will having outcomes data do you think, to sort of with acceptance or from a market standpoint?
So in the F2, F3, we believe that we're capturing those patients, and we will capture those patients. With the outcome study, it's a little bit confusing with the names of our studies. Our F4C study is an outcome study, but if with F4C, that has an opportunity to actually double the market for Rezdiffra, meaning there's a smaller number of cirrhotic patients or patients that have a well-compensated cirrhosis, but we believe that there'll be better penetration with those patients because they're so close to those liver mortality events.
I want to, if there's any, I don't know if there's any questions in the audience. We have about 5 minutes left, so I've got a few more questions, but if there's anybody in the audience that has, I see a hand. Wait, we're going to get a mic up to you.
Hello. I just wanted to ask a question about your role as women in leadership. So sorry to bring it to another.
Please, go ahead.
Just wondering, you know, you've been in this industry for decades, and can you talk about your experience of women in the space? I just keep thinking of the places that I go to see the biotechs that are growing, the small ones, the big ones, and the disparity in women in the scientific community. I'm sorry, but
No,
really curious. It's perfect people to ask.
That's a great question, and I think that's one of the reasons why I wanted to come to Madrigal, actually, because of the story of Becky Taub as the founder of our company, who actually developed resmetirom from very early on and drove that forward as really a pioneer, you know, that brought Rezdiffra to us today, which can, you know, really set a new standard of care in MASH. But also it's timely because just yesterday there is the liver meeting that's going on right now in San Antonio, and we had an ad board of all women providers, sort of the thought leaders, et cetera, and we were talking about the evolution of you know, women in leadership roles, but also the specific needs of women with MASH. Super, super exciting time because we are seeing a real change in that. And I think it's evidenced even in our leadership in our company. Not only is our founder, and head of R&D a woman, but also our general counsel, our CFO, myself. So I think very diverse. And Kitty, you have a very impressive career.
Yeah,
and
Kitty, tell. I
role,
mean,
so
it's interesting.
please.
You mentioned you come from Gilead, so tell
Yeah,
us.
so I was at Gilead for 20 years, and then, but I left Gilead a little bit. We had a similar story. So I left Gilead with our CEO. So he was employee number 3, and I was number 4. And so, I mean, it is interesting when you meet a lot of women that have reached the C-suite, that it's interesting that often there is a male ally that you have worked with in your career where you end up having just a really good combination of skill sets, and then that you end up working, and Carol and I just met, but we very quickly had that conversation that we both have those sort of relationships with our CEOs where we actually know them so well, we don't actually have to communicate. And basically
we do, but we don't have to.
Yeah. we do too. But it's been interesting. And then what happens is when you get a female in leadership, it's interesting what happens around about the organization. And so I can say honestly, I am the only female on our leadership team, but we tried so hard in terms of we had 2 different females that we were on the verge of hiring for our chief medical officer, and just we weren't able to do so. But my organization, in terms of all of my VPs, there's a very high proportion of females, and I think that it's allows us to develop teams. I mean, it's interesting. I think this year at JP Morgan there was the whole biotech sisterhood that has evolved over the last few years, where it's female CEOs. But what actually is really beautiful about that group, I mean, I know some of them are ex-Gileads, and so I know them from there. But they're constantly out there thinking of other women. So I got, I'm looking for a board position, and I got an email from one of them like, oh, I saw this position. I thought immediately of you, and they're connecting women, and just because they would like to broaden the role for all of us. And so it's actually very powerful right now and incredibly supportive.
I totally agree. I mean, I have never in my 5 years in industry had a woman for a boss. But things are very different now, but I am very intentional about creating opportunities for women, mentoring women, and making sure that they do have a seat at the table.
Yes.
But exciting times.
Well, with that, we'll end it. Carol, Kitty, thanks for joining us today.
Thanks
Thanks
very much.
everyone. Thanks for your questions.
Thank you.
Please welcome to the stage Novartis U.S. Chief Customer Engagement Officer, Des Creary. And to moderate this sponsored conversation, STAT co-founder and executive editor, Rick Burke.
Hi there. I'm very excited to ask Des a bunch of probing questions. But before I do that, before we start our session, I just want to say, I just went on a Seema Kumar at Cure just took me on a tour of this building, and many of you probably know, but it's just really fascinating the kind of the research that's going on right above us and below us. It's just people doing it's exactly what we're talking about here, and real live research. So it got me kind of excited about the reason everyone is here. Now, Des, and what you do at Novartis is really interesting. It's the Chief Customer Engagement Officer. But what strikes me what really caught my attention is your history, because you were at GSK for years, you have a pharma background, but then you went to Microsoft to be their America's lead in healthcare. And I'm just curious about that detour. It seems like a cool detour, but why you did that, how you ended up at Novartis, and how that informs your role now.
Sure. Let's start with saying I appreciate you saying it's very interesting.
No, it's like, it's not the usual boring, like, it's just like, boring. Sorry, but you know.
Thank you. I think my decision, first of all, I did grow up in pharma and was very rooted in being focused on the patient, and had an opportunity to go into technology, which for me was about understanding a different culture, understanding how to develop and deliver innovation. And I learned a lot there. I believe, my time there helped me get closer to the industry I grew up in, better understand and appreciate the healthcare ecosystem, how it works, what are some of the barriers, and what can we do to work together to overcome those barriers to get to better patient care. And though as a pharma industry, our North Star, I think we have this in common as a community, our North Star is the patient. What I learned in technology at Microsoft was to balance that with what it means to be customer-centric. And if you get them both right, not only do you get to better patient outcomes, but you also create a situation where our customers are better cared for, which then also leads to better patient care.
Is there a speed at which technology operates that you've found is different from healthcare? Is there a sort of the approach of how you get things done, or is there?
There are a lot of differences. I think speed is one of them, but I also believe it depends. So if you work in technology and you work in a regulated industry, you'll be a bit slower than a deregulated industry.
Right.
But one of the biggest takeaways is innovation isn't just about a product. So that's a trap you can fall into when thinking about technology, that you believe innovation is about an iPhone or about a cloud service, and actually the innovation's in the people and the ways of working and how quickly people can make decisions. Are they agile in terms of change? Is their learning agility high? And so I think those things really do separate companies that innovate and ones that are followers, whether you're in a technology company or not.
And speaking of innovation, you were drawn to Novartis because of the CEO, or how much did that factor into?
Sure. Our CEO, Vas, was a huge factor in me of leaving Microsoft and coming to Novartis. That was a huge factor, also alongside of innovation and the vision to deliver innovation through Novartis into the community. So a couple examples of that. I think last week we announced as an organization our NFL partnership, which is first of its kind with the pharma industry and the NFL. And our first order of business there is early detection in breast cancer. And so for me, that is a great representation of why I came here. And the second is...
Can you stop for a second? So what NFL, what was your thinking about? Is it reaching a certain audience you wouldn't normally reach, or the scale of it? Like that's an interesting synergy there.
Sure. I mean, I think there are many audiences you can reach on the NFL's platform. For us, the first order of business, again, is breast cancer and early detection of breast cancer. And we had a Super Bowl ad earlier this year that was the focus of that ran before we made the announcement. And so we'll have other opportunities to reach a larger audience. Last year alone, globally, 296 million people received Novartis medications. We want to expand access to our medications, and part of that is making sure you get to audiences that you might not be able to get to otherwise.
Can you talk about some of the therapies that you're pushing at Novartis? And
sure, that takes me to innovation point two, which is radioligand therapy. And for me, this is precision medicine at its finest, and also a good collision of my two worlds, of technology and healthcare. Radioligand therapy, in short, for decades we've been treating cancer with radiation. That's not new. What is new and what makes radioligand therapy, in my opinion, cutting edge, state of the art, is that you're targeting radiation into cancer cells with minimizing damage to otherwise healthy tissue. And that is new. And we're the only organization that is doing that at scale. And so this is another reason I came to Novartis, is to be able to be a part in innovating and renewing everybody's belief in an amazing industry.
That radioligand therapy is fascinating, and we don't like. I asked a bunch of my colleagues if they could tell me about it, and the one person who's written a lot about it is I don't know if she's here in the audience, Allison DeAngelis, and it's turned out there's a huge interest in our readers in that therapy. Can you talk a little more about how Novartis has ended up sort of taking the lead on this, and sort of what your hopes are for the future with that therapy?
Yeah, absolutely. So for us, it starts with research, development, and then commercialization. And I would say the innovation in the medicine, the platform is one hand of it. The other hand is innovation and commercialization. And so what it requires to get this medication in the hands of a patient, or even get a customer set up to administer, requires the very best of us. And so if you think about logistics and cold chain supply, that is a very new muscle for us, and it's a difficult hurdle. But we believe in doing difficult things on behalf of our customers and patients. I will spend more time on commercialization. So after we've developed the medication, the platform, now it becomes about how do you understand your customer well enough to get them organized to administer this medicine. That requires orchestration on the customer's behalf. That is a new muscle for them. So the customer needs to orchestrate around the patient to administer, and our Novartis field teams and otherwise need to work differently together as well. So field reimbursement teams, our customer engagement teams, our medical teams, our market access teams, and more work in concert to make sure our customers understand the therapy, understand access, understand the barriers, and can get it to the patient.
That sounds exciting yet really daunting to me when you talk about it. It's not like the NFL piece I get, and that's like everyone can understand that breast cancer and but this is, you have to explain the therapy, you have to convince people that they need this, you have to identify the people. This is like a big process that was complicated, years long, right?
Absolutely. I think that's a good point. Part of what is unique about the therapy is you not only need to educate on the medicine, but you need to do some convincing and educating on the technology. But that is the responsibility and the privilege of being first. I'm pleased with what we've been able to do so far. In terms of the future, we believe this is a large part of the future of medicine. Precision medicine, targeted therapies that do the job that the medicine is meant to do without harming the rest of the body is where you want to be. And so if I rewind 5 years, what we thought, the way we thought about precision medicine and AI and technology seemed very far off. We're living a lot of that today. And RLT is radioligand therapy is a great example of that.
In our closing moments, given that you're so focused on the consumers, what do you want consumers to know about Novartis that they might not know?
Our mission is reimagining medicine. Our mission is not to do the easy things, but to conquer the hard things. We are very much believers in creating access to medicines. I said earlier that 296 million patients last year received Novartis medication. That was up on prior year because we believe in doing the hard things. And so though we are anchored in, along with our other colleagues in the industry, putting the patient as a North Star, we believe in being a partner to our customers, to the actual providers who are sourcing through what to do with EMR, clinician burnout, nursing shortages, and they have a mountain of hurdles on their own. We want to come in and be a resource, and we very much believe we're hitting that sweet spot. Best is yet to come, for sure.
Great. Well, Des, thank you so much. Really appreciate it.
Thank you.
All right. Sorry, I jumped the gun here. I don't actually need to use this. I'm just motioning with it.
But I do have it for when you guys have questions for this next session, which you very well may and should. And I really think this is such an exciting session. There's been no shortage of news about public health lately, from the CDC in crisis, the measles outbreak, and all of the public discussions about vaccines, vaccine efficacy, and vaccine safety. We have some amazing panelists to come talk about this. Please welcome to the stage Dr. Catherine Edwards, who is literally co-wrote the textbook on vaccines, just an expert we turn to again and again, and Dr. Tom Frieden, President and CEO of Resolve to Save Lives, also used to run the CDC. And to moderate, someone who needs absolutely no introduction in this room, my amazing colleague, STAT Senior Writer for Infectious Diseases, Helen Branswell. Come find me.
Hi, thanks everybody for coming, and thanks to the two of you in particular for sharing your time with us today. I guess I would start by saying we're in really unprecedented times. There's a large measles outbreak that has spread over at least 3 states so far, and the Secretary of Health and Human Services is stressing how to treat measles rather than telling people how to prevent their kids from getting them. The man who was nominated for your former post, Dr. Frieden, saw his nomination withdrawn last week, and then he wrote an article, or released a statement to the New York Times in which he defended Andrew Wakefield. Studies into vaccine hesitancy have been defunded by the NIH, agencies are being gutted, and the Advisory Committee on Immunization Practices, which was supposed to meet at the end of February, still hasn't met. So we've got a lot to talk about here. Dr. Edwards, I know earlier in your career you cared for children who were sick with diseases that kids don't get these days or don't need to get because vaccines prevent them. How worried are you about what you're hearing and seeing out of HHS?
I'm extremely worried. In the 50 years since I've been a pediatrician, the diseases that are present now are very different from those when I began. When I began, Haemophilus, bacterial meningitis was very common. At any one time, 2 or 3 patients were in the hospital with meningitis, pneumococcal meningitis, pneumococcal pneumonia. We also had a lot of infections from children that would have varicella, and 200 children would die every year of varicella. Polio was around when I was a child, and my classmate's brother got polio and had anoxic brain damage. So I've seen these diseases, and certainly measles is of concern. We know that these diseases are an airplane ride away, and we know that if we let down our guard, they are going to come back. I tell people sometimes that we've had such great vaccines that we're like the Maytag repairman. We have such good products that people don't know the diseases, and that is a problem. They don't understand that these are serious, and I've stood by the bedside of children that are dying of vaccine-preventable diseases, and it is not fun. So I am very worried.
Thank you. Dr. Frieden, there's just been so much going on, as we were saying just in the hallway just now. It's absolutely dizzying. But there seems to be a clear thread running through many of the actions that we've seen and the statements we've heard so far coming out of HHS, and a lot of them relate to vaccination policy. What are your thoughts about the official messaging right now about the various measles outbreaks that are underway in the country, and what you're seeing and hearing from HHS about those, and particularly Secretary Kennedy's comments?
Well, Helen, exactly 35 years ago, I was a newly minted Epidemic Intelligence Service officer here in New York City assessing a large measles outbreak.
Oh, wow.
And we've seen kids severely ill, kids dying, kids with long-term consequences of neurological problems from measles. So when the head of HHS says, oh, it's a routine childhood disease, well, yes, it's a common childhood disease that still kills around the world approximately 100,000 kids every year. it's also the most infectious of the infectious diseases. So the federal government might pause, but the measles virus doesn't pause. Bacteria and viruses don't pause. They accelerate when we let down our guard. And it's quite predictable that reduced vaccination rates will result in increased measles spread. Measles is so infectious that if in this room one of us had measles and 3 of us weren't immune, probably all 3 would get it. That's how infectious measles is. No other infectious disease is routinely that infectious. But many other infectious diseases will come back if our defenses get weaker. As Dr. Edwards was saying, I became a physician and an infectious disease specialist in a time when diseases like Haemophilus and pneumococcus were killing and disabling lots of people. And these diseases have largely gone away because of vaccinations. And it is a precious but a fragile and vulnerable progress, because every day lots of kids are born. And if kids in the coming cohorts aren't vaccinated, we will be condemned to repeat the past.
I was wondering in particular about your thoughts about the way Secretary Kennedy has been messaging, you know, stressing that we know how to get kids through measles, stressing the importance of vitamin A replication, supplementation, as you have studied. How do you feel about that? It seems to me so uncommon that we're not hearing the, top leaders of the country's top health leaders stressing the importance of vaccination.
Although supportive treatment can be somewhat helpful for kids with measles, the bottom line is vaccination is how we prevent measles and how we prevent measles deaths. Without going into the details, that outbreak I assessed in New York City 35 years ago, because we were obtaining acute and convalescent serum from kids with suspected measles, we could study vitamin A levels in measles at the CDC vitamin A lab. And we showed that vitamin A is what's called an acute phase reactant, which means it goes down when you're very ill, when you have fever, and it comes back up when you get better. And this, along with other evidence, led to the American Academy of Pediatrics recommending vitamin A supplementation for kids under 2 with measles in the United States. That's already been a recommendation for many years globally, because we know vitamin A is important, especially in communities that get too little of it. In the US, less than 1% of kids have insufficient vitamin A. And even if you have more before you get sick, you're still going to have too little when you're sick. So it's not useful as a preventive measure. You can take too much, and that can be quite dangerous because it's what's called a fat-soluble vitamin, so you can have severe problems from taking too much of it. But yes, it has a role in treatment of young children with measles that reduces a little bit the severity of their illness. A whole lot better not to get measles by getting vaccinated.
Right. Dr. Edwards, we were talking before about the way Secretary Kennedy has been categorizing vaccination. It's saying effectively that it's an individual choice, it's up to the parents, and that we know that or that at least the CDC was asked to pull some advertising for its flu campaign, the Wild to Mild campaign, because the word was that HHS wants to focus on individual choice. And you have talked to parents about getting their children vaccinated. What's the danger of going that route versus a more active recommendation?
Well, certainly we always want parents to be informed about the vaccines. We want them to be informed about the disease. We want them to be informed about the vaccines and what the potential side effects of those vaccines would be. But we present the vaccines this is what your child should receive today. Do you have questions about it? This is very important for keeping your child healthy. I give this to my grandchildren are all vaccinated. My children are vaccinated. I got disease, so I didn't need to be vaccinated. But today, this is what your child should receive. if you present this as an option, you could say, well, today you can decide whether your child wants measles vaccine or not. It's your choice, and really, if you have questions, I'm happy to answer it. So you can understand the approach of saying this is what should be done, or you can do it. It's a little bit like telling your children or your grandchildren that they shouldn't do something, or if they want to do something, they can. It's a totally different approach. And if you look at the data, you will see that approaching people with you can decide what you want is very ineffective. It also conveys a message that this is not important, that it is not critical. And we know that if children are exposed to measles, they have 72 hours to get the vaccine or they get measles. So asking you to go to your doctor to talk about it is another delaying technique. So this is really important. The other thing that's so distressing is you probably know that this past week, all of the grants that addressed concerns or discussions or hesitancy or how to present and talk to parents, all of those were barred. Every grant that had the word hesitancy in it was withdrawn. Now, why is that? That's very uncomfortable and very unfortunate. So it's really the message, how you communicate, is critically important.
At the start of this conversation, I mentioned the fact that the ACIP, the Advisory Committee on Immunization Practices, was supposed to meet at the end of last month, and it has not yet met. I know, Dr. Edwards, you were on ACIP at a point, and you also, I think, chaired FDA's Advisory Committee, the Vaccines and Related Biological Products Advisory Committee, or VRBPAC. It was supposed to meet earlier this month. It did not. Across the administration now, departments are being asked to defend or explain why they need these advisory committees. The implication seems to be that it looks like the number of advisory committees could be dramatically cut. What do we lose if we lose these two bodies?
Well, these are critically important advisory bodies, and I'm sure that Tom will want to comment a little bit about ACIP too, but the ACIP is really the committee that makes recommendations to the head of CDC about which vaccines should be approved for the use in children in the public sector. That's half of the children in the country, and in general, the private sector goes along with it. So if CDC meetings are not held, this last meeting was going to talk about a new meningococcal vaccine. That discussion never happened because the meeting didn't happen. Was going to talk about a new chikungunya vaccine and also modifications to the RSV recommendations and the influenza. So if we don't have ACIP, we do not have recommendations for new vaccines for the CDC. The VRBPAC is an advisory committee. I was chair of that committee that goes over the safety of all the vaccines, and it's a committee that is full of very difficult questionnaires and asking about whether the vaccines are safe and whether they should be actually licensed. So the other thing that's so interesting is that we keep hearing that there's going to be a transparency that we've never seen before, but it seems to me that there's no transparency, that there's a lack of transparency. And these committees that are full of very smart people are being muzzled.
Dr. Frieden, I'd like your thoughts about ACIP as well, because you will recall that during his nomination process or confirmation process, Secretary Kennedy alleged that 97% of former and current ACIP members had conflicts of interest. And recently there was an addition to the website of ACIP that listed some of the members and some occasions on which they recused themselves for votes because of, they had done some work on a study or whatever, effectively portraying all of that as conflicts of interest. What do you think is going on there?
It's very clear there's an attempt to discredit basic facts because you've got here fact versus fiction. And the ACIP, which I oversaw for the 8 years I was CDC director, really is a best practice in public discussion. We had people coming from all over the world to monitor the sessions. It wasn't a widely known committee outside of public health or pediatrician circles, but the ACIP, every deliberation is public. Every presentation is public. All of the meetings are live streamed. None of the members are paid. All of the people on it represent important non-conflicted interests: parents, pediatricians, public health specialists. And the issues that the ACIP assesses are really complicated. Dr. Edwards mentioned some of them. They're hard. They're hard topics. At what age is it safest and most effective to give certain vaccinations? What is the latest data showing? Because data changes. One of the things about science, and I think one of the things that's frustrating for the public sometimes, is that science does change. And so we always have to say, based on what we know now, here is our best recommendation. That might change in terms of the number of doses, the size of the dose, the spacing of doses, and that's the way science works. It's not immutable truth. It's a process, and the process is open assessment of information and provision of that information. The issue of conflict of interest is very concerning to me because I saw that statement, 97% have severe conflicts of interest. Absolute nonsense, untrue. Where did it come from? We dug it up. Okay, there's a 2009 report about a 2007 audit of CDC, all of CDC's advisors, not just advisory groups, but anyone who was a consultant for CDC. And it was assessing this really horrific form. Somebody I saw say, it's actually worse than doing your taxes to do this form. It's really tedious. And it said, yes, 97% of people who submitted to all of the committees had some problem with that form. They forgot to initial a page, or they left something out, and by the time that report was written in 2009, the CDC had already begun addressing those. And in my time there, we also strengthened. It's really important that there is neither conflict of interest nor apparent conflict of interest. And so what was just posted on the CDC ACIP website was saying, okay, here are all of the disclosures. Disclosures are a good thing. They're not an indication that there's a problem. There's an indication that there's a safeguard against a problem. And there were things like, oh, this researcher worked on a Shigella vaccine for the same company that has another vaccine that's coming before ACIP, so they are recusing themselves from that discussion. That's the kind of open, transparent discussion and avoidance of conflict of interest that is best practice.
Right. I think one of the things that ACIP doesn't get enough credit for is, you know, because I've listened to a lot of ACIP meetings over the years, they bring up stuff that industry probably wouldn't necessarily enjoy them bringing up. I mean, it's not like they're rubber stamping vaccines, you know. If they think there's a problem, there was an issue with GBS and the RSV vaccines for seniors, Guillain-Barré syndrome, and if they see a flag, they raise it. It comes up at the meeting, and they talk about whether or not they need to revise policy, whether they need to take a more go slow approach, which they have with the RSV vaccines for seniors. I mean, this is not a body that is in the pocket of industry, right?
Absolutely. In fact, when I opened one meeting, I created some controversy by saying, let's be frank, the public sector buys half of the vaccines with an assured market. It educates clinicians on how to use them. It educates the public on the need for them. It transports them to more than 40,000 pediatricians' offices in the US. It indemnifies, other than any malpractice or significant manufacturing problem. And in response, frankly, manufacturers charge whatever they want. And that's a problem. And so when I think people have suspicions about, hey, there's something nefarious going on, well, there are some problems. There are some conflicts, but they're not in ACIP. They're in how we structure, how we purchase our medical products in this country, where taxpayers pay to develop the things through NIH research, and then they pay to buy them from the companies that benefited from that research. That's a problem, and we're one of the few countries that doesn't control that. And I get that our pharma friends won't like what I just said, but you know, that's my perspective.
Okay.
I think also
Yes,
that the ACIP provides an enormous amount of information on the website in forms of slides that are really good. And they have epidemiologic data, and I use them all the time to talk and to teach about vaccines. I also use the website, and that also has been concerning because, as you know, there have been a number of changes to the website at CDC. So now I'm always wondering, well, you know, is this changed or is this not? So that also is another issue that has been brought up with what's happening currently.
Helen, if I can just add on, I'm often asked, well, could ACIP be replaced by some university or private body? And I actually think no, because it has such an extraordinary depth and breadth of knowledge. The CDC scientists who have worked 20 years on an issue, who have no conflict of interest, will tell you, here's the really deep dive into this data, and then the ACIP considers it. I don't think there's any group that could do that.
So we're hearing that big cuts are coming at CDC, you know, and the agency is massive in terms of the tasks it's been tasked with by Congress and the things that it tries to do. I'd like you both to sort of give us a sense of some of, you know, 2 or 3 of the things that you really think that it cannot stop doing. And I know that's a very difficult task because there are lots of things it shouldn't stop doing.
Well, if I had to choose 3, I would start with global health security, because we're only as safe in this country as the world is. Diseases don't respect borders, don't need visas, don't stop at walls. And if outbreaks emerge and spread without being stopped anywhere in the world, we are less safe. So
isn't that difficult in this time when the CDC is not allowed to interact with the WHO?
it's a big problem. If you look at the measles and polio and tuberculosis networks, they rely on the CDC labs to support the World Health Organization. So by not talking to WHO, we're weakening the systems that allow us to track what's happening and respond rapidly. So the first is global health security. The second is surveillance and response. I really worry about data systems. You know, if you look at some of the global work, stopping what's called DHS, the demographic health surveys that USAID has funded. Now, there are some legitimate concerns about those systems in terms of their building of capacity in other countries, but they're unparalleled. There's no other data system that has so much and so rich data. If we don't know what's happening around the world, and if we don't respond quickly, and in the US, remember, about 2/3 of all CDC dollars goes out to state and local governments to do rapid detection, rapid response. And third, I'm really worried about the leading causes of death, because tobacco use, unhealthy diets, this has not been an area that has always had strong bipartisan support, to put it bluntly.
Right.
And there have been proposals to eliminate centers from CDC, to stop it from doing tobacco control, whereas, in fact, it is the main part of the federal government that tracks what's happening with tobacco use and supports comprehensive programs in state and local governments. So those are the 3 areas that I'd be most concerned about.
Dr. Edwards?
I think I'm also concerned about surveillance, from what's happening currently. I'm sort of a pertussis expert, and 2 of the leading people in pertussis at CDC are going to leave. They also tell me that they're not sure that they will be able to conduct the surveillance to monitor the amount of pertussis that occurs. That's very worrisome. We started with CDC funding a vaccine surveillance network 25 years ago that measured the impact of vaccine, called the New Vaccine Surveillance Network. Vanderbilt, who was part of that, was just told that they would no longer be able to be funded to do outpatient surveillance or emergency room surveillance. So we won't know what the impact of vaccines are. So if we don't know, we can't say that it's good. The next area that I'm worried about is vaccine safety. And I think that we hear all of these complaints about safety, but it was such an extensive system of vaccine safety that was put into place for COVID that exists, and I'm worried that that's going to not be supported or going to be somehow modified or changed, and that is also really, really important as well. And then finally, I'm very worried about the young people. I'm very worried about the young scientists at CDC, some of them who've been terminated because they haven't been there long enough, and to see what's being done. And you know, we all are going to be replaced, and we're going to be replaced by the young people, but if we don't have trained young people, then who is going to replace us? So I think those are areas that I'm very concerned about.
Can I just ask you a little bit more about the vaccine's effectiveness surveillance? Because you know, in some respects, one would think that Secretary Kennedy would be interested in that kind of data. It almost seems counterintuitive that they would stop collecting that. What is that about?
Well, I think there's an awful lot of counterintuitive things that are happening currently, and sometimes I wake up and I think, is this real? You know, so I think that's part of the goal. If you have no data, I mean, as you know, Venezuela has no infectious diseases now because their entire system has been eliminated. So if you have no surveillance for disease burden, then you don't know that you have the problem. And so I think this is really a severe problem.
Okay, so I'm going to ask one more question, and then I'm going to go to Matt for audience questions. One of the major advances we saw during the COVID pandemic was, putting the years of science into messenger RNA into use. We got vaccines far more quickly than anybody really thought was possible as a result of that. And those vaccines, one of the beauties of them is they can be updated very quickly and produced in mass quantities very quickly. If there is another pandemic anytime soon, we will need that capacity and that technology. And yet there is a kind of a demonization of mRNA going on now. And we've heard that grants that include mRNA are being flagged. People are being warned to take it out of their grant applications. Are you worried that we're facing a future where somebody's going to take this stuff off the table?
I'm very worried about it. I also feel that the whole mRNA with COVID got a very good and extensive assessment of safety. I was on the Pfizer DSMB, and that study had 20,000 people receiving the mRNA vaccine, and I went over hundreds of case report forms and looked at the cases. I also was funded by CDC to look at vaccine safety, so I've looked at many adverse events. The adverse events that were clearly shown to be present through CDC systems, the vaccine safety data link, are the myocarditis that was seen in the young boys. But other than that, there was no indication of Guillain-Barré. There was no indication of other adverse events. We followed the pregnancy histories very closely. There were no evidence of any congenital malformations, miscarriages. So other than having a sore arm and feeling bad a bit, it really is quite remarkably safe. And so I think it's really unfortunate that it has such a bad name. And I know that even at the NIH, they are being asked to list all of the things that are using mRNA, and that is not a good sign.
And I would just add, it's not just the next pandemic or better vaccines for flu or other things that are potential with mRNA, but anti-cancer vaccines. And it's a great technology. We were fortunate. It saved millions of lives. Like any technology, it needs to be rigorously studied.
Okay, Matt.
We're going to go long because there are so many questions in the room. We're going to start right here. Please identify yourself.
Thank you. Mylan Studer-Anders. Wondering about the vaccine communication issue, right? Vaccine, it's a very strong word, and the jab is very invasive. I'm wondering, have you thought about maybe, hey, it's not a vaccine schedule, but it's a health schedule And regarding the vaccine that's invasive, I know that some companies are trying different ways to deliver that vaccine. Maybe, vaccine now is a negative thing in people's heads now. I mean, there's anybody you ask, people have an opinion on vaccines. Maybe we should change the name of a vaccine.
Well, I think until recently, other than clean water, there's probably nothing that has had an impact on public health like vaccines. So I think that part of the issue is that it is being demonized. And I think that we do need ways to communicate better. We are looking at, and certainly I've been looking at studies with nasal vaccines and with cutaneous vaccines. I think we all are looking for ways to deliver vaccines other than by injections. And so I think that needs to be acknowledged, and I think we do need to find ways. I mean, I'm not very good at all these internet things, but certainly my young people are, and that's how you reach the people. So again, I think we need better communication. I think we need to listen to people and see what we have. But I also think that the community of the people that are vaccinated needs to speak up, because if your children are totally vaccinated and they live in this community where the rest of the people aren't, they are at greater risk for disease. So I think we also have to push back a little bit in saying that the norm and really what is good is vaccination. So yes, I'm open to communication, but if those grants get axed, we're not going to have new ways to fund them.
Hi, good morning. I'm Sophia. I've been a nurse for 30 years, most of that time in an adult emergency department. I pride myself on facts over feelings. But I have noticed in the past couple of months, I'm headed all the way in the feelings section of things. And even for my 2 elementary school children, I am now wondering about next flu season and whether or not the vaccines that they will take will be safe, whether they'll be available. And I'm starting to panic, and it's only March. That's the first point. The second thing that I'd like to say, the question, can public health stay healthy? I'm wondering about all the mental anguish that we're having and how that will be addressed, because I don't even hear anyone talking about that. A lot of us are under so much stress, even those of us who are living relatively comfortable lives. So I can't even imagine those who are feeling the pressures of just managing daily existence, food insecurity, and all that other stuff. We are just in uncharted waters, and I wonder how we'll stay healthy from a mental health perspective at baseline.
Amen.
Thank you very much for that, and thanks for your work in the emergency department for decades. I think vaccines, perhaps more than anything else, depend on trust. And what we're seeing is a breaking of trust. You heard Dr. Edsberg say she's not even sure she can trust what's on the CDC website anymore. That's really concerning. And I think part of the theme here is it's good to like natural stuff. But just remember, bubonic plague is natural. So there are really good things from science that keep us safe and healthy, whether it's clean water or vaccines or antibiotics when we need them. And getting that dynamic right is really important, and keeping balance in each of our own lives to keep that mental health vibrancy is so important. So thank you for the work you do.
Dr. Frieden believes in balance. He emailed me last week to suggest I write about something, and I wrote him back and said, I'm sorry, I'm on vacation. And he said, stop reading your email when you're on vacation. It's not good for you.
Yeah, Ron, sorry. Is this on?
Yes, it is. We can hear you.
Ron Cohen, biotech founder, CEO. It's been a very interesting panel, and you have all dived into many of the trees here, but the forest seems to be inescapable, which is that there is an effort right now in our country to dismantle over 200 years of progressive improvement and advancement in public health, starting with Edward Jenner in 1800 or so for the smallpox vaccine. what we are not grappling with here yet is what do we do about it as a community? Because this is, what you are describing is alarming. You know, we're treating it in a sense as an academic sort of analysis, which is what we're all trained to do. But really the big picture is this is alarming, and what does our community do about it?
I wish I had a snappy answer for you, but I don't. I think partly we need to keep focused on making the facts clear. Partly we need to get better at communicating, whether it's in social media or traditional media or in local communities. Partly it's being strategic about making sure that we don't lose the basic systems to track what's going on, so that we're not like Venezuela, where there's no illness anymore because the tracking systems have stopped. And if you look at what happened around the world during COVID, I think COVID broke a lot of things. There are countries around the world that hid millions of deaths. If you can hide millions of deaths, you can hide a lot of things. And we need, in public health, our core value, our core methodology is truth, and our core value is effectiveness at reaching the people who can benefit the most from the interventions. But I think we need to do a better job of telling the story, of what we're doing, how we're doing it, of being frank about what we know and what we don't know. And that's not going to be easy. It's not just the US. We're seeing these trends in countries around the world where there is you know, it's now a demerit to be an expert in something rather than a merit. And that's a real problem, because we need expertise, and we need to be able to distinguish snake oil from facts.
I think the other...
There's actually almost a response to that, but it already came in from Steve Dickman, who's the CEO of CBT Advisors. A bunch of people in the room know him. Reciting the book In COVID's Wake and saying that it seems that in some ways public health experts and officials during COVID did not follow the science, and how can they win back trust?
Well, I think the main thing we need to do coming out of COVID is a renaissance in public health, a robust primary health care system. It is pathetic just how bad our primary care system is in this country and around the world, and resilient communities, more able to withstand health shocks. I think you can criticize different things in the COVID response with validity. The CDC error on laboratory testing was awful, and it resulted in a lot of deaths. Some of the guidance was impractical, slow, inappropriate. Schools were closed too long, inappropriately. There were definitely mistakes. I don't think anyone anticipated how dramatically the virus would evolve over relatively short periods of time, so that what worked at one point didn't work at the next point. And this gets to the essence of public health. Public health works well when we have accurate systems to track what's happening in real time, to try a response, to assess whether that response works, and then to adjust it appropriately. And that wasn't done during COVID. The harms of COVID are massive. Easy to be, you know, Monday morning quarterback, some of the things we said at the time, but really moving forward, not many people want to hear about COVID. Yes. There's real fatigue with it, which is exactly what happened in 1920 after the 1918-1919 flu pandemic. But we do have to address the problems that we have now in ways that resonate with people.
I think also that the local, everyone likes pretty much their doctors. And I think capturing some of that community, making sure that the local doctors and that understand are supported and helped, I think that really goes a long way in terms of credibility. I see so many of my colleagues in conflict, however, because we are so intertwined with federal funding. I mean, I'm retired, so I don't get any money from the government anymore, I guess, except my Social Security, which maybe I better be quiet about that. but I think that's a real issue too. I mean, there was a discussion yesterday with the National Academy of Medicine, and some people are saying, well, why don't you say more from the National Academy? Well, you know, the National Academy has funding from the government as well. So it's really difficult to know how to articulate this in a way that is not going to damage things that are very important. So it's a hard time, that's for sure.
Stop, except I am allowing one more question, and I want Helen to answer it too.
Oh.
And I've been told that you're only one reason I'm allowing this, aside from that Catherine Ames is a friend of the event, is that you're each allowed one word.
Oh, okay.
Catherine Ames, patient advocate, CEO of Fierce Hope Patient Advocacy and Consulting. My question, I promised I would wrap this up for us, is a yes/no answer. Dr. Edwards, Dr. Frieden, and Helen, is in your combined century of experience, on behalf of young people who I work with, mostly adolescents ages 13 to 23, 25, can you give us a yes/no on if this truly is an unprecedented time in your career? You said you had a childhood friend who had polio. Is it a yes/no answer?
Yes.
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 Thank you.
Thank you very much, guys. We just ate into everybody's lunchtime, so go hurry up and eat. Thank you so much to all of you for being here. On the back of your name tag, you should have a little white sticker. If the sticker says R, go to the rooftop. If the sticker says 12, go to the 12th floor. If you and a friend are separated, that's okay. These are the guidelines to help break up the crowd. Pick whichever sounds better. Head up there. Go to the rooftop or the 12th floor. Get some food. We'll see you back here at 1:30 p.m.
Okay. That spelling is definitely not right. You said it's K-H-R-U. Through. And. Yeah. Where?
Chronicling? How do you say it?
London.
No.
Yes.
Please take your seats. Our presentation will begin in 5 minutes. Thank you. Please take your seats. Our presentation will begin in 5 minutes. Thank you.
Did everyone have a good lunch? I hope so. If everyone could please start to trickle back into the main space, we are going to start our next part of programming. I hope everyone tried the strawberry dessert because I think that was the best thing. But for those of you who are in the kitchen, it is now a great time to find a seat because we are going to move to our next section. We have a very, very good lineup for the remainder of the day. We are going to hear from former FDA Commissioner Scott Gottlieb. We are getting a first interview with Pfizer's Chief Strategy and Innovation Officer, Andrew Baum. And we are going to see none other than Bill Nye, the science guy. But first, we're going to talk about what is happening at the NIH and the FDA and what all of that means for science and research, which I know is a question on everybody's mind. With that, we are going to jump right back into the program, so everybody please do find a seat. Thank you so much.
Here for a sponsor spotlight, please welcome to the stage Melissa DuPont, Global Public Affairs Lead, Neurology Pipeline at Sanofi.
Hi, everyone. Thank you. Hopefully you enjoyed your lunch. I had to change my opening statement after hearing all of the talks this morning. And I really want to say that now, more than ever, it is important to listen, act, and lead with the patient community. That's going to be the only way to drive change and innovation. So I'm going to tell you a little bit about my background. I spent a bulk of my career in R&D. I ran large global clinical trials, multi-therapeutic areas across all different phases. I also worked on some external initiatives focused on clinical trial technology and patient experience. My sweet spot was always piloting a new technology in a clinical trial, rescuing a study that was failing enrollment, had high patient discontinuation. Why were we having these problems? In hindsight, clinical trials were being created in a vacuum. We built it, they'll come. Do we understand, and did we understand the patients we were serving? Did we understand their lifestyle? What kind of economic status was it for that patient population? And what kind of burden were we putting on the sites? So when there was a rule that was developed to work directly with the community, I raised my hand and I started in neurology. And there we got to listen and talk and bring the community on early in the development life cycle, so that way we were actually building and developing programs with people that had lived experience. For example, in MS, our multi-study, large phase 3 program was fully informed by the community, right down to some of our inclusion-exclusion criteria, where we were able to widen some of it and break down some barriers around the norms in MS research. Then we were entering rare neurology, a space with ALS and chronic inflammatory demyelinating polyneuropathy, or CIDP. ALS is a disease that the patient community is riddled with large unmet needs and many failed clinical trials. We had to bring them along from the beginning, and we knew this. They helped shape and design our phase 2 program. And while we didn't meet our primary endpoint, we were able to be transformative and deliver our data back directly to the community with the partnership of PAGs, because that's how powerful partnerships can be. And now in CIDP, another space that we're new and learning, we're learning from the community. We're listening, and we're building. We built the phase 2 program, we built the phase 3 program with their input and hand in hand with the community. And so I want to say that, by doing these things and taking these actions, not only did we change some criteria, make some visits better, we also were able to widen clinical trial access for our community and foster and push for more innovation. Now, as we focus on the neurology pipeline and we're bringing additional options to our communities that have significant unmet needs, listening is more important than ever, because we can bring to light what is unseen. And listening takes place in various forms, and that's some of the things I want you to walk away from this. It's not just direct conversations. We can literally meet and listen to the community where they're at. Social media, discussion forums, webinars, on the Hill, wherever they are, we can listen, and we can take those words, fears, challenges, and we can put them into action to help them. We can also translate those stories and bring not just awareness, but we can also take CIDP, MS, and put them onto key agendas, thinking beyond the medicines and focusing on the lives. And by listening, acting, and leading with our patient community, our partners, we can ensure that individuals with neurodegenerative diseases live for the moment rather than for their disease. I'm happy later if you want to deep dive and talk about more examples. This is my passion, and I'll see you after for the networking. Thank you.
Simon, the George H. Gardner Professor of Obstetrics and Gynecology, founding director of the Center for Health Equity Transformation at Northwestern University Feinberg School of Medicine. Jonathan Jackson, co-founder and research director of Crescent Consulting, and a returnee to the Stat Summit stage. Holly Fernandez Lynch, associate professor of medical ethics and law at the University of Pennsylvania, and to moderate, Stat FDA reporter Lizzie Lawrence.
post-lunch session. So chaotic, disheartening, cataclysmic. These are some words to describe the past few months for science and public health. You've seen the headlines. Some of you have felt this impact personally. But to quickly recap, first, government agencies scrubbed references to DEI. Then came the NIH cuts to indirect costs. Then the administration started leveraging funding for political reasons, canceling grants on studies to study diseases like cancer and diabetes, to punish institutions like Penn and Columbia. Meanwhile, morale is plummeting within public health agencies as Elon Musk and the US DOJ service starts aggressively trying to fire staff. We have 3 esteemed researchers with us here today to speak about unpack some of the consequences of these actions, answer your questions, so please ask questions, and start to think of a path forward. So we'll start broadly, big picture question about the future of scientific research in the US. What are you all most worried about right now? Who wants to take that one first?
I'm happy to start. Yeah, so I think we have to recognize that for all intents and purposes, we are looking at a lost generation of researchers. So we have rescinded offers for students, for some staff positions. We have changes in terminology. We have just a climate of uncertainty where there was relatively more certainty. So even if everything magically resolved today, there is going to be a lingering chilling effect. And of course, it's going to be the most marginalized communities that feel this disproportionately, because we saw those cuts, were the kind of the first and fastest to be announced. So I think we have to recognize that, again, even if everything goes away today, we are looking at probably a period of upwards of 10 years before we can fully recover our scientific workforce.
I agree with that. I mean, I think the question is, what am I not worried about? There's so many things to be concerned about right now. You know, as Jonathan mentioned, the loss of the scientific workforce, right? Trainees are dramatically affected. People who, were excited about entering science PhD programs have had offers rescinded. You know, there's austerity measures within universities, concerns about new faculty hires, a concern about brain drain, and that's just within the university setting. We haven't even talked about the firings within our scientific institutions, right? So you can't do science without the bureaucrats who help disseminate the funds and make sure the regulations are being followed. So I have deep concerns about who's going to be able to do the science and what's happening to the government workforce as well.
So in addition to the workforce and staff, we have grants being stopped, terminated. I got a letter last week. My grant is terminated because it is harmful to the health of Americans. That is a quote from the NIH on letterhead, and I am a highly funded NIH researcher over the last 2 decades. So again, I don't think I've harmed Americans, but ever, and that was never my intent because I'm also a clinician and I took the Hippocratic oath not to harm anyone, especially in the field of obstetrics and gynecology. But this is really serious. This is super harmful. And it's not about, I hear, 2 years. No, actually, the NIH director and the National Cancer Institute director are both appointed by the federal government. And that is a concern, a real concern, because 4 years is a lot of time to lose in the workforce and with labs literally shutting down. And this is like cancer, NCI is cancer. I mean, cancer is one of the top causes of death, right, along with cardiovascular disease. And those labs are shutting down. That is a concern. We are going to fall significantly behind.
Yeah. Melissa, I wonder if you could tell us a little bit more about the grant that was canceled and the reasoning that was given to you.
Yeah, so I'm not the only one who has received letters like this. Grants that are being shut down include grants that have the words diversity in them, faculty hires. So the whole NIH FIRST program cannot hire any new faculty. And that FIRST program was to help advance the careers and opportunities for more representation of scientists in our country. Right, more diverse scientists, because we know that diverse scientists and scientists that represent a variety of communities in our country actually do research in unique areas of our country that haven't had that experience before. We also know that, so I've had a clinical trial, a cancer clinical trial to try to improve representation in cancer clinical trials, because we know cancer clinical trials, when they're available to you, you get better care and better cancer outcomes. And so not everybody in this country has an equal opportunity to enroll in a cancer clinical trial if they are diagnosed with cancer. And 2 weeks ago, I had a cancer clinical trial protocol at the national level pulled. It was approved by the Institutional Review Board at the National Cancer Institute, and then it was disapproved the next day because the word gender was in it. And it had nothing to do with LGBTQ persons. So again, I was promptly told to change the science, meaning take out the word gender and replace it with sex, which is scientifically inaccurate. And as a scientist, I have an oath to myself and to the people, the public, that I'm doing good science, and I refuse to change words.
So Jonathan, I want to hear your thoughts on, I know we talked a little bit about what it means, the consequences of things like health equity, diversity in clinical trials being lumped in with this, becoming casualties in this war on DEI language. Maybe talk a little bit about how this happened so quickly and
I think before I acknowledge that it happened quickly, we have to admit that the system was pretty fragile, pretty performative beforehand. It was not great. And so, if you've got institutions that can scrub all mention of health equity over a weekend, then maybe it wasn't really embedded in those institutions to begin with. So we have to recognize that we hadn't done the work. There are probably a lot of people in this room that don't care about DEI. It doesn't affect your workflow, it doesn't affect your science. It may not affect your impact or your lives or your neighbors or your families. But what I want to remind you is the people who are impacted by DEI are going to be, they are the canaries in the coal mine. The way they come after folks like us is how they're going to come after folks like you in the days and the weeks and the months to come. So pay attention to how DEI is being weaponized, because this is the playbook. We have to pay attention to this. So we already have this chilling effect. We've heard from Dr. Simon about some of these things. I had a new grant start in November. We were gonna hire a postdoc for 2 or 3 years. We've had to pull that because we are not sure if the grant is going to last past our reporting period in May. So we were not sure about that. And there are knock-on effects. I do a lot of work that is focused on communities of color. And what we've seen initially is a full retreat of all of the researchers who cared about health equity. We know the famous health equity tourist phrase that was kind of coined and popularized by Stat. We see this full retreat of all of those performative individuals, and the communities are left hollow. They're left decimated. I had a community call just a couple of days ago, and there was just 12 shell-shocked faces in front of me, saying, the very idea of community is being eroded, it's being eradicated. So again, even if we weren't impacted, the communities that we are trying to help, the communities that we are trying to improve outcomes for, are also being hollowed out. And without any kind of ability to shine a light on that or to rectify that, we are still going to be left sorting through the damage for decades.
So I wanted to ask, I guess specifically with this ban on words like gender and this focus on language, how can researchers work around that? Should they work around it? Is it bad science to use inaccurate terminology, or are there ways to kind of, work around the restrictions that have been made?
Yeah, I was just going to say, part of the challenge that's happening here is the confusion and the disorientation, and the fact that people are really being driven to distraction instead of doing the science. We're in meetings about, can I say diversity here? Can I say transdisciplinary Or is it going to be caught up in a word search for trans, right? And so the science isn't getting done because we're working on just responding to something that's so illogical and unscientific. There are some, I think, examples where you can change the words and it's not going to affect the science. And then there are other circumstances where the word, like you're studying trans patients. You can't just change the words and still do the science. And so I think it's bad in both ways, right? If you can just change the words, it's still a waste of everyone's time to do it. And otherwise, like the genuine science is being decimated, right? It's problematic all around. I'm sure we all have had conversations. I constantly am saying to myself, we're living in the twilight zone. There's a list of banned words. There's a list that, they're just going to go through a word search and we can't do our science, right? It just is so illogical. And that's part of the challenge, right? How do you come up with a logical response to something that's not driven by something that makes sense? It's really intended to decimate science, intended to decimate universities, and it's having that effect.
But we can't let it do that. We can't. And the public really needs to weigh in, all of us, of all levels, because, you guys know about statins, right? A long time ago, statins were trialed just on white men. And we didn't know they were helpful for women. But indeed, cardiovascular disease impacts men and women, all people, right? And it's one of the leading killers on this planet. And statins have done a whirlwind of good for many people. But if we hide our science or try to change our science and not include everybody and represent everybody in our clinical trials, then we have no clue what medications are right for who and why, under what circumstances, doses, everything. So we can't let this happen.
Yeah, and I think we have an audience question.
Over here.
Yeah. Hi, I'm Melissa DuPont. I was just up a second ago. So huge, this is a major topic, but my question for you is, how do we shed the light? So what tools can we bring so we can have riots in the streets? Like, how do we arm the community to have these rational conversations with stuff that is very irrational? I think that's going to be a key, because I think we all know it here, but I think it's our community needs those tools.
I think we're starting to see some of this through good science communication and reporting, which is bringing to the public a recognition that this is not, ivory tower science that doesn't affect you. Like, these are cancer cures that are not going to be available, right? Your family is going to be affected. Your loved one is going to be affected. So, the general population, right? Like, my parents never needed to understand indirects before, right? But you can talk to your neighbors, your colleagues, right? Help them understand, what indirects pay for. Help them understand how university science supports industry science, right? We have built over decades this collaborative relationship where we innovate within universities through public funding, right? So that we generate these public goods that industry can then build upon, right? We need to bring that message to the public so they understand, just like the price of eggs, right? That's something we talk about all the time around these dinner table conversations. We need to start getting people to understand this is not just affecting scientists. This is going to have effect on, real people, real patients.
Well, and science and technology is important for all of our lives. You can bring it back to, instead of thinking about science as these Bunsen burners and these beakers in some mysterious lab in an ivory tower, but what about our cell phone? Our cell phone, for example, that pretty much most people use on the planet, is benefited from a ton of research. Not just research to develop the apps on the phone, but just the technology of the phone in and of itself. That took a lot of scientists. So I think, again, making the word science more connected to everyday life and every person, I think, is really important right now.
And I'll say that just to kind of answer the question, we need to make sure that science is humanized. Those of us who work in science, do your parents really know what you do? Do your kids? Certainly not your neighbors. So maybe just start with having a conversation. Maybe just say, there's a lot of stuff about science in the news. There's a lot of stuff about, maybe we want our kids to like choose if they have vaccines or not. Just have a conversation of how do you weigh evidence? How do you come to a decision? How can you curate knowledge and kind of enable access to it? So I think we have to recognize that this feels catastrophic to all of us, but there are 19 other catastrophes that are currently going on, and nobody cares. So we have to advocate for ourselves as people first, and then we can talk about, like the really cool science that we're all missing out on.
We have another question.
I'm not going to stand because I'm in the middle. Hello. Lisa Lewis. I work for Faster Cures at the Milken Institute, focused on all things representation and equity across the biomedical research ecosystem, including clinical trials. You answered part of my question, Jonathan, but I actually know a little bit about all of you on the panel. That's not creepy at all. I'm kind of a fangirly. But, Jonathan, as a neuroscientist, Holly as a rock star in bioethics, Melissa as a trailblazer in health equity for gynecological disorders, how can we do more of what Jonathan just said and make this human, right? Jonathan, you said that there are canaries in the coal mine and communities of color, those who are underserved, obviously will be affected first by all of this. But all of us in this room know someone who lives in a rural area. Right? We're in New York City. We have world-class medical centers here. What about people in Montana and Mississippi and Nevada? How can we let people know that all we want is access to research for humans, period?
So I'll just very quickly say, I grew up on a farm in Texas, so I really understand the need to reach rural residents. I go and visit my beloved grandma every year who lives in rural Mississippi. But I'll tell you what I told her when I saw her last Thanksgiving, which is my grandfather's dealing with a huge variety of cancers triggered by his former workplace. And so my grandma had questions. And if it turns out if you answer a couple of questions, you get to the science of this cancer. You get to the science of the disease. You get to basically say, this is what we know, this is what we don't, this is why we're trying to explore this new area. And so my grandmother, who has lived in rural Mississippi her entire life, is now a much more staunch science advocate because of a 15-minute conversation that I had. So, I think that we all have somebody in mind, maybe outside of the 5 boroughs even, I dread to think, but that we can have that kind of conversation with. It just starts with one conversation, one human connection.
Yeah. And Catherine, I think we have another question over there, and then you guys are doing the work for me. This is great.
And just to say, there is somebody over here who's got their hand raised.
That's who I was pointing to. Yeah.
Thank you so much. This is equity, making sure that the people that we don't see are being served, right?
My name is Johanna Bennett, and I'm with the healthcare team at Porter Novelli. I had 2 questions, but I'll ask the first one and let someone else ask a question. This is not an administration that's known for being terribly forgiving. Are you putting your jobs at risk by speaking out?
Hell yes.
Okay.
Absolutely. Somebody's going to find this talk and Google my name, and I might lose the grants that I have by the middle of next week. But but it's the job. We're scientists. If we sit back and just allow ourselves to shrink back in the face of darkness and denialism, then we don't deserve the jobs that we have. We are the ones who are pushing back the brink of ignorance. Yeah. If you can't do that, then you shouldn't be a scientist.
Absolutely, 150% agree. And I'm in academic medicine, and last time I checked my university, Northwestern, our values were the pursuit of knowledge in the service of truth. And whoa, you're censoring my websites, and I'm tenured professor, and you're censoring them on words that I received tenure for. So again, there's got to be that power, that collective voice coming from our academic institutions, from our public, from all levels, because this is a full throttle assault on health and science in this country right now.
So the big question on everyone's minds, which we've been talking about already, but is just what now? And part of that is communication and speaking about these things. But then there's this dramatic shift where science has been funded federally, and that money needs to come from somewhere, and it's very unpredictable right now. So I want to ask you, should industry step up to fill the void? Do we have, I know there's complications with that, conflicts of interest. Do we have the luxury of worrying about that if the alternative is no research? And how should we start to grapple with these questions?
All you, Holly. Yeah. Well, so look, I mean, yes, there are conflicts of interest related issues that I think we already have academic industry collaboration, and so we're capable of addressing those issues. I think the bigger challenge is that it's not going to be possible for industry to play the role of the government, right? What government-funded science supports are public goods, right? Basic science that benefits all, as I said before, right? That industry can then take up and develop into commercial products. So is industry going to step up and say, right, well, all of my competitors can benefit from this too, but sure, we'll pony up the money for that kind of academic research? I mean, I don't know. Are you planning to do that? And it's a big challenge, I think, to expect for-profit businesses to play the role of the government. It's an unreasonable thing to expect. The other challenge that comes along with that is that industry funding follows market demand. And that's another thing that the government can help accommodate, right? The government can say we have unmet needs in these areas, and maybe there's market failures, and maybe we need to prioritize in ways that, not in a derogatory way, but that industry is not prioritizing because that's not where the business interests lie, right? So the interests that the government is responsible for promoting for the public are not the same necessarily. I don't mean to suggest that they're diametrically opposed. It's a Venn diagram, but it's not a perfect overlay. And so I don't expect industry is coming to the rescue. What I would like to see is a more full-throated defense of university research that industry relies on, right? So the universities are not, I don't think, doing a good job coming to each other's defense. I don't think industry is doing a very good job coming to the defense of academia either.
Right. Yeah, so I mean, something else I wanted to ask you about was, and you mentioned this, Holly, is the gutting of federal health agencies and what that means for science. How are you all thinking about that, and is that affecting you right now? What are the impacts that are to come from that?
Yeah, I mean, we're having a brain drain in the government right now, especially in the health agencies. That's concerning. There are people who have been lived their entire career in the federal government, in the areas of health, and leaving. They're going to industry, so good. There's going to be talent somewhere, but that's concerning. But in addition, we have, I'm an obstetrician, right? I take care of patients. I was on call a few nights ago. We have the pregnancy monitoring systems. Those databases and other databases are being taken down. They were being taken down or edited even and stopped. We've stopped collecting data on births from like around a few months ago, right? It's going to go back up, right, I'm told, in April. But we have the highest maternal mortality rate of any industrialized nation on this planet. That is sick. It's wrong, right? And it blows my mind that we would like hide data, stop data that the public's tax dollars pay for, right? We need data in order to understand what's going on in anything, let alone in pregnancy. So it's a real concern.
Yeah.
I want to just pick up on this point. We haven't yet talked much about FDA, right? FDA needs people, right? It needs experts who are able to work with sponsors to help you figure out what's going to satisfy regulatory standards. It needs people to review applications quickly, resolve problems. Would you want to work at FDA right now? Would you want to work anywhere in the federal government right now? Already, it was a thankless job, right? You have academics like me criticizing you all the time, right? You have patients, you know, banging down your door. You have industry saying you're holding things up with red tape. You make way less than you make in industry, and now you've got DOJ coming for your jobs, right? It's just an unstable environment, which is incredibly bad for moving the science into patients' hands and clinicians' hands.
You know, I was asked the other day, well, where can patients or people go get accurate health information right now? Because now disinformation is rampant, right? And distrust is rampant. And I say dis on purpose because it is harmful. And it was really hard to answer that question. And if anybody knows me, it's not, I can talk. I can usually answer a question. And it was really hard because not everybody has access to a healthcare provider or access to a healthcare provider they can trust. And so where does the public get accurate health information right now? It's a great question.
Yeah, yeah. Go ahead, Jonathan.
Yeah, I guess I'm just trying to start thinking about synthesizing these things. We have to recognize that with the changes, with the losses that we're seeing on the federal level, we're seeing a loss of individuals who can help underpin the administrative component of doing science in the United States. We have a loss of independent scientists who are intramural that are helping lead the way on some of the most critical health questions of our time. And then I think most critically, we have a loss of vision and leadership because we are seeing departures from, from even the highest ranks at NIH and across like a lot of federal health and health agencies. So we have to reckon with the fact that where we had information that we could criticize, now we have nothing. It has gone dark. And I think the next step beyond that darkness is the push to kind of isolate. So again, same playbook that, that large forces have used against minoritized and marginalized groups, the same playbook. So there's that kind of that divestment. There is kind of the disruption of the status quo. And then there's that isolation component. So again, just you can set your clock to it. That's what's coming next for all of us.
Yeah. Another question?
Yes. Hi, my name is Julia Cohen. I'm a city research scientist with the New York City Department of Health.
Bless you.
And, it's very uncertain times. In my office, we're like, I was very uncomfortable because it's like, oh, we'll just keep going the way we're going until we hear something else. And I'm just like, oh, we're way past that. So I'm just, I'm struggling with, the idea that we're so dependent, at least in my bureau, I'm in disease control, we're so dependent on federal funding. And so I'm thinking, like, we need to think outside the box. I mean, I work in HIV, that's my area of specialty, and we had ACT UP. We had, we had people on the ground doing the work with, I mean, it's easy for me to say, I guess, because I get a salary, but which may go away because it's from the Center for Disease Control. But I wanted to just say that I had done a very small project. It was a health equity project. It started during the pandemic. And I worked with only 8 of my colleagues, and we have 6,000 people in Department of Health. But it was only 8 of us. We did it all with no funding. It was a federal grant, but there was no funding attached to it. And when I told my colleagues, they were like, oh, you're crazy. They said, we can't do a project with no funding. I said, we can do it. We can. Yes, we can. And we're still doing it. We're doing webinars. We're doing interventions with community. We're doing all kinds of things. I understand it's a very small scale, but I think we really need to think outside the box. This lesson we've learned is we cannot depend on federal funding, that we need to find other resources.
Absolutely.
So thank you.
Thank you.
Yes. Thank you.
Yeah.
Thank you.
Just, I agree with all the points you've made. I just have more of a hypothetical question, and it comes to courage. Where are all the billionaires? There are all these people. The United States has incredible wealth, and not that they should offset the cost, what the federal government should be doing, but why have they not spoken up? There are a lot of people who have always spoken up about a lot of issues. Where are they? What has happened? This city is filled with wealth, and go out to California, go out across the country. Where are they on this issue?
Well, I think there are some op-eds of CEOs funded. One of my professors from Yale School of Management, Professor Sonnenfeld, he definitely has been in the media with with a CEO voice and CEOs voices. But I agree, it's not enough. We need more. We really do. And thinking outside of the box, there still needs to be people having salaries. You know, I don't, like, personally, I'll be okay, right? If my entire research portfolio goes down the toilet, I'll be okay, but I worry about all the team and all the community organizations that partner with me and that rely on the funding. And that worries me a lot. So yes, I have to go to foundations, I have to pitch maybe VC, all these different things that I have to do that I didn't have to do before, and it's a new skill set. So again, I am very worried about that, and I would like to see more people step up. Yeah.
I just want to quickly say, I think there are two elements to that question. Number one, we're not visible. Like I said, there are dozens of other catastrophic events disrupting dozens of other industries right now. We just don't have the loudest voice in the room. Number two, I don't know if I'm allowed to say this, but many billionaires are on the side of the current federal administration because it's beneficial to them and it's beneficial to their companies. They're essentially apolitical. They'll support whichever party gets them the most tax breaks. So, you know, it might be it might be against their sort of self-interest to wade into this with any kind of oppositional force against the federal administration. Yeah, I mean, I guess it's courage versus, you know, the continued accumulation of something that you don't need, right?
Yeah.
So.
You also have to assume that they think anything is wrong with what's happening right now. I think that's to your point. You might agree with it.
Right. Another question?
Hi, my name is Jasmine Evans. I am a PhD student at Temple University. My work throughout undergrad and even my doctoral work has always been about like health disparities, now more specifically within AI in medicine. And as someone who is set to graduate very soon, everything that has been going on is a bit disheartening, not only for me, but even other like young professionals that are in the room today. If you guys could like speak specifically to that audience, to those peoples, what would you say, especially with those like in health equity?
So I get this question a lot, and I'll let you speak too. I have a lot of soon-to-graduate mentees from all levels, and this is an opportunity. You gotta take it that way. As much as it is a disaster and scary and harmful in so many ways, it's an opportunity, like Dr. Jackson said earlier, to really think about how we frame things, think about how we bridge and cross. Right, AI and health equity is so important right now. I actually have a grant whereby we're getting community members to inform AI algorithms to do better cancer care delivery, like navigation, patient navigation. So there is a way to frame your research that's so critical right now in a way that speaks across the aisle, across audiences. And it really comes down to what we said earlier around representation. Right? We need representation of voices in that AI algorithm because then otherwise the AI algorithm will not generate the magical things it does that really represents we the people. And so I could go on, but there's hope. Don't give up hope. Absolutely. We've been through a lot in this country over centuries. Right? And this is just an opportunity to really make it even more solidified what we do in the structures that be and the processes that be.
What I want to say is it's important to remember that these systems were never safe. Never. Especially around questions of health equity, especially for individuals that are intersectionally minoritized. These systems were never safe. So now it's almost like saying the quiet part out loud. We know that maybe you had prospects of going into some of these institutions that have now refused to kind of defend or support scientists who might look like you or pursue the things that you're interested in. So you have to keep that in mind. So I'll put my hand up and say, I lost my job a year ago. I was fired by my institution who used the same tactics that now the federal government is using against them. So we have to recognize that there is a certain value to mutual aid, recursive support. The same systems that keep minoritized communities afloat the world over, we can now start to take advantage of those as scientists to support ourselves and the communities that we live in.
Yeah, and we're out of time, but Holly, do you have any other hopeful words you want to end on?
No, I was just sitting here thinking you're a much better mentor than I am because, yes, of course, right? You want to reframe things as opportunities. You want to be hopeful, but this is wrong. I mean, this is so wrong. It's so bad. And even if you have concerns about DEI generally speaking, this hammer approach that's being used without any sense for who's being harmed and what we're losing. It's the way, we were talking about this before, like science is being held hostage, right? I'm at Penn, right? We're now being threatened with the loss of $175 million because we had a trans woman on our swim team, right? These two things are just so disconnected. So, yes, I want to be hopeful. I'm hoping that we can, move past this, but I also want to be realistic. Like, so much damage has already been done. We're going to see loss of a generation of scientists, loss of, just an incredible amount of science. And so, my hope is that it stops, not that we figure out how to fix something that's so incredibly wrong. And it's not hopeful, I'm sorry, but it's
okay.
just wrong.
It's important words to end on, and thank you all so much for joining us today.
Please welcome to the stage NYCEDC Senior Vice President for Life Sciences and Healthcare, Vic Parvaz. And to moderate this sponsored conversation, Stat First Opinion Editor, Tori Bosch.
I'm always here to just skip the stairs, take the difficult route. Well, Vic, thank you so much for being here with us today. So you've been in your role since October, I believe, right? Yeah. And it's a role in which you're now overseeing $1 billion life sci investment as part of a major initiative in New York City. How do you plan to spend that money?
Yeah, well, well, thanks, Tori, first for having this conversation. And I just want to acknowledge that it's not easy to be the opening act for Bill Nye the Science Guy. I just wanted to make sure that was acknowledged. Look, $1 billion can go a long way, but I sort of want to have this conversation and think about beyond the metrics. So let's talk about the metrics first, right? $1 billion. Right now, we have 42,000 jobs in the life sciences ecosystem in New York City itself, 150,000 if you count the greater New York City metro area. In New York City, that's over 1,500. The 42,000 is over 1,500 companies, right? So clearly, we have the infrastructure that's continuing to grow in terms of where these jobs live. We can also speak about it in terms of the talent pipeline and what we're graduating. So actually, the New York City metro area leads in life sciences graduates on an annual basis. So we're graduating approximately 2,700 life sciences graduates annually. So there's a lot of great metrics in terms of how we're doing today, right? I can even add into that, we've got 3.5 million square footage of lab space, right? And that includes dry lab and wet lab. We're intending to increase that with the $1 billion to 10 million square feet over the next 20 years or so. So there's the metrics, but I think we speak too often about the metrics. There's the qualitative piece, right? If I might indulge you in that. We're also investing, we're taking that money and we're investing in a program called BioBus, right? So this is a program that's existed in the Bronx for a number of years. In 2023, the EDC invested $700,000 to expand the program. And with that expansion, we're in year 2 of that. BioBus, which is literally a state-of-the-art mobile lab, is exposing STEM to underrepresented communities in the Bronx. And with the expansion now in year 2, we're going to hit almost 13,000 students. So, you know, you look at the top level metrics, which are so impressive in terms of numbers. What's interesting to me 5 months or so in my job is thinking about also the social impact. You can have both, right? You can have the financial impact. In my seat, I'm meant to grow the jobs and economic impact in New York City, but it's also nice to know that you can do that in conjunction with social impact.
So, you know, you said there's 42,000 jobs in the city alone, 1,500 companies leading in life sciences graduates. Why don't people already think about New York the way they might think about the Bay Area or Boston for life sciences workforce?
Yeah, I mean, I think it's a very fair question, and maybe I will answer the question, but you know, we're very proud that we were just noted as the applied AI capital of the world. So why do I bring that up to answer your question? Life sciences, healthcare, across industries, everyone's moving to accelerate based on the use of applied AI. And I think we're at a critical juncture here in New York City where we're not waiting for that to happen. Many of the big pharmas have already taken stride in that domain, but as we're building out this 3.5 million into 10 million square footage of lab space, we're building it deliberately with the idea that it's going to be wet lab and dry lab. We're looking at the tech companies that sit in New York City, and we don't think of them as silos, right? We're proactively bridging it. So, you know, next year when I'm sitting up here, if you'll have me, you know, what I'd like to talk about is the number of jobs that are bridged between tech and life sciences. So I think it's a very exciting time in New York City right now to look at that. There's also commitment. New York City has always been a life sciences leader. We have great stuff going on in West Harlem. We have institutions such as Cure Deerfield, where we're sitting today. We have the New York Stem Cell Foundation. We have Pfizer. We have Regeneron. We have Lilly. You know, we've the big names. We've got the startups. We've got active incubators and accelerators. So this has been happening for years. The commitment that New York City and New York State are showing and the voice in which they're showing it is through monetary commitment, right? So the $1.6 billion commitment, we're very proud to say that the New York City Council approved a new science campus in Kips Bay. So literally, you know, like a couple of blocks from where we're sitting today. That will go live in 2030. And so if I use my same sort of litmus test, there's the metrics, right? That's very impressive. It's going to be 2 million square footage of a science campus. It's going to house the office of the chief medical examiner with a forensic lab. It's going to have a life sciences dedicated public high school. But the impact that you'll have is this combined campus. You're pulling the community and you're also creating the future science leaders and innovators right here in New York City and fueling that into New York City. So, you know, a first opinion I recently published argued that it's a little bit of a volatile time, perhaps, in biotech to be a biotech employee. And the first opinion argued that it's a time in which biotech workers need more diverse skills, right? You can't only have one set of skills. What kind of work are you doing to help
train this workforce, whether they're mid-career or early career, or just kind of starting to toy with the idea of biotech?
No, it's something that's very close to my heart. I think people are going to get tired of me saying this phrase, but again, talking about this both financial and social impact, right? So the financial impact in terms of continuing to fuel great talent into the New York City ecosystem. One program that we're actively involved in is partnering with GenSpace. So they're at Sunset Park in Brooklyn. And a couple of years ago, we created a program called Break Into Biotech. So again, many of us in this room are fortunate to have been exposed to science at an early age, and even more fortunate to be able to have the financial means, in whatever way that was possible, to progress along that route and deliver on our passions. Many of us don't have that opportunity. We've recognized that at the EDC. So this program, Break Into Biotech, is for those a little shy of their mid-career, have minimal or no experience in the life sciences. And GenSpace, being the first community-based biolab, has partnered with us to create cohorts of about 15 students. So it's an intimate setting where they can work on either therapeutics or programs related to the sustainable bioeconomy. And with that, just off the heels of that, on Tuesday, we're also making sure that they're skilled in business skills, right? In these days, and certainly in the future, it's going to be nearly impossible to be in the lab and not have some business savviness. So we're recognizing that as well, and to arm them properly, we're fueling the science, hands-on lab experience in Sunset Park with business skills as well.
And what are you hearing from people who are doing your training programs?
I mean, that's the best part of my job, right? There's a lot going on in these dynamic times, but the thing that I take the most joy in is I would have never been able to do this if it wasn't for this program. Most recently, just last month, we celebrated right here at Cure, the EDC celebrated its 1,000th Life Sci intern. And the testimonies during that event were, I've never been happier. This wouldn't have been possible if it wasn't for the Life Sci intern program. And if I elevate us to the stats again, so that's the social impact and the emotional impact, and you're not only changing lives of individuals, you're changing lives of the individuals, their families, and in many respects, their communities. The stats for the Life Sci internship program, nearly 60% are women, nearly 70% are individuals of color. So when you look at the stats and you think about the social impact, and then also having a north of 50% retention rate of those interns staying in the New York City life sciences ecosystem, I mean, I don't know how you get a better story than that.
And how did you get into this?
Biobus. No, no, they wouldn't have me. No, my background is I went to pharmacy school, and so I had an inkling towards science. I will say, and this is a true statement, Bill Nye, the science guy, had a lot to do with it. So if you're out there, Bill, I did get into science because of you. But no, I think for me, it's what many of these students in the internship program at Break Into Bio tell us, right? Which is the ability to create and the ability to have impact. And for me, you know, I've had a variety of roles. I've been in the lab hands-on. I've also led a health sciences practice for a Big Four. Regardless of where I was in my career, the impact to patient has always been center, and I think that's something we need to hold on to in these times.
Well, we were promised an interpretive dance earlier, but I think we've run out of time.
We ran out of time.
Yes. Well, thank you so much.
Thanks, Tori.
Up next, please welcome to chat with me about the FDA and tariffs and eggs. Please welcome to the stat stage a man who needs no introduction but wears amazing suits, Scott Gottlieb. I mean, and really amazing suit. So. So you went out on a bit of a limb at the beginning of the year. I think it may have been the end of last year. I think I was supposed to be on vacation when you were on CNBC. But you raised some concerns about what the confirmation of Secretary Kennedy might mean in terms of vaccine uptake. Now, we've already talked a lot about vaccines today, and I don't know that you need to say a lot more, but I did want to check in. We have a measles outbreak with more than 300 confirmed cases with deaths. What have you thought about the communication so far?
Well, look, it wasn't just concerns about what the long-term implications would be in terms of vaccine uptake, although that was obviously the underlying concern of the issues I raised in that CNBC hit and in some subsequent hits, but it was also what I thought he would do in short course. And all that's been borne true. I said on that hit that he would take down the front-facing pages of the CDC website that encouraged people to get vaccinated. I said that I felt he was going to disband ACIP and reconstitute it with like-minded people. We now see movement towards that. I said that there may be some movement to try to influence the review process. I think we've seen that with the vaccine advisory committee that was canceled around the selection of the strains for the flu vaccine. You know, my biggest concern at the time, which we haven't seen yet, was that they would look to move the vaccines that are currently on the recommended schedule, things like MMR and DTaP and polio, onto another portion of the schedule that's sort of a softer recommendation where the vaccines aren't typically mandated as part of the school entrance requirements. So Gardasil, for example, is in this second category versus MMR and DTaP and polio. And I think that there is a possibility that they could look to move those vaccines.
And that does mean people don't get Gardasil.
Well, what it would Gardasil isn't a requirement for school entrance requirements, and I don't think it should be. I think that it's appropriately positioned on the schedule, but certainly polio should be. I think MMR should be, DTaP should be. So if you see those move into the category that's considered joint decision making, where you need an affirmative decision by the physician and the parent to get the vaccine, which would conform with what RFK said. He says, I don't want to take away the vaccines. I just want it to be a choice between the physician and the parent. And that sounds very sensible, but for the fact that if you don't require these vaccines as a part of the school entrance requirements, you're going to see vaccination rates fall dramatically. And you're going to start to see more distributed outbreaks. And I think that the challenge for states is that a lot of states, particularly red states, that are under pressure to weaken the vaccine mandates as a condition of going to school, right now really they hide behind, in some respect, the CDC recommendation. They say we can't do that because CDC recommends these. And I think if you took that away, a lot of state legislators would be under more pressure to weaken those mandates, and we would see vaccination rates really more precipitously fall. They're already declining, they'll continue to decline, but you'll see a sort of step change.
You would see the measles vaccines fall enough in a lot of places.
Well, if you see another 5 to 10% decline, you're going to see much more distributed measles outbreaks in this country. I think that could do it.
There's been a lot of other news. It's very useful to ask Scott in a lot of contexts. He has sat in the FDA commissioner's chair, obviously. You're on the board of Pfizer. I meant to disclose that as soon as you got out here because someone will note it if I don't. But you've also been a venture capitalist. You've worked in think tanks. You've done a lot. So there's a whole bunch of policy things that have come along. But one I really wanted to ask you about was Operation Stork Speed. The FDA's positions on infant formula have actually been surprisingly newsworthy the past few years. It was one of the biggest issues under Caliph's administration. But this new initiative, a set of actions and initiatives focused on infant formula, beginning a nutrient review process and increased testing for heavy metals and other contaminants. How big a difference do you think this makes in the infant formula market?
I don't think this gets at the underlying issues and the underlying tension, which is that Europe has a food standard when it comes to baby formula and infant formula, and they endure a lot more outbreaks than we do here in the U.S.
Outbreaks of formula-borne diseases.
Formula-borne diseases, right. And we have something that's more akin to a drug standard, where we not only require clinical trials for new formulations in this country, so you have to do a growth study where you compare a formula-fed infant to a breastfed infant. You also standardize it against a known formula if you want to have a new formulation, to be able to demonstrate that the baby thrives and grows just as well under the new formulation as they would under the existing formulation. And typically, you'd also randomize to a breastfed baby. And we also require drug-like GMPs on manufacturing. And we have stricter standards, and we have less outbreaks of illness. And so there's a tension between the two regimes. And you see people sort of giving this nod towards the European formulas on a presumption that somehow they're more healthful or contain more natural ingredients, but they're also less regulated. And I think if we want to adopt something more akin to what Europe allows, we should have that debate. This doesn't get at that underlying issue. And if we want to continue to maintain a drug standard here in the US, there are ways, I think, to make it more efficient for baby formula manufacturers to try to reformulate formulas, to try to get in some of the new ingredients that parents seem to want to seek out in these formulations, and make the process easier for manufacturers. Right now, there aren't the equivalent of PDUFA dates. There's no predictability. The inspectional force isn't well staffed, so getting a new plant inspected takes a lot of time. There's no requirements on FDA in terms of when they get back to sponsors.
But this is, again, the paradox that you might need something like a user fee to make that happen.
You might need a user fee, or you might need appropriations. This was an under-resourced part of FDA. Now, they increased the resources when we had the tragedy associated with the outbreak under the last administration. So there's been more resources put into that group, but mostly on the inspectional side. But if you look at what they did, it's an RFI, which is a request for information, which RFIs go out all the time. They don't necessarily translate, and they often don't translate into actual policy making. It looks like an extension of the personal importation policy. So, I think in some respects it was aptly named Project Stork Speed, because a stork travels 15 miles an hour versus Operation Warp Speed, which goes a lot faster.
I was setting up the joke myself,
but,
but
you see some of the dialogue coming out of this, and you'd think they discovered fire. This really isn't going to change the policy framework. And I think if it's a prelude to doing that, great. But hopefully this isn't the totality of the policy making.
We have, we will be taking questions. Catherine's out there, and I think Mario's out there too. But I have one more comment from RFK on Pulse that I want to ask you about, which is something you wrote about in Stat this morning, so you guys can all read about it more on statnews.com, where we all live. But one of the biggest political issues has been the price of groceries, in particular the price of eggs. The price of eggs is being driven up by avian influenza, by bird flu. And RFK had suggested that one solution might be letting the flu run through flocks to find out which chickens are resistant. What do you think would be do you think that would be a solution, and what would be a solution to trying to keep egg prices down?
Well, I wrote about it on your page today. Look, I think right now we're in a period where it's possible that the avian flu isn't going to come back in the same way it's been spreading in the past, although I think most people believe that it will, that this is endemic now in North America. And typically in the past when we saw these outbreaks of avian influenza, and I lived through some of them when I was at FDA, they kind of burned themselves out. This one has been persistent. Right now we're in a period where the wild birds really don't fly through the North American flyways in the wintertime. And so we've seen kind of a lull in outbreaks on farms, and it could be because of that, and that we will see a pickup as birds start to migrate again in the spring and transmit this onto poultry farms. Look, I think that what Secretary Rollins put out was a thoughtful, comprehensive plan. So there were a lot of good ideas in there, particularly around stepped-up biosecurity. I think the big debate is going to be whether or not you move to vaccinate hens. And there's a couple of problems with that. Setting aside just the logistical challenges, you do get pushback from anti-vax groups, even though we vaccinate hens for probably 5 different pathogens, at least 3 and maybe 5. So they're already being vaccinated, but you're seeing some pushback. But the other issue is a trade issue, and I think this is the one that's more paramount for the Department of Agriculture right now, which is that we export a lot of baby chicks. So we raise about 10 billion broilers in this country, about 300 million or almost 400 million egg-laying hens, but about 10 billion broilers. And some of those chicks get exported on FedEx and refrigerated planes under very carefully controlled conditions. And the problem with the vaccine is that while it would substantially reduce the likelihood of a hen contracting the virus, or not substantially, but it will reduce the likelihood, and it will reduce the likelihood of them succumbing to it and becoming ill, they could still get the infection and they can develop a subclinical infection. So what the Europeans worry about is that if we vaccinate all our chicks and we export them, we could also inadvertently be exporting chicks that are sick with the virus but just don't show manifestations of the illness and therefore won't be identified. And they'll be importing avian influenza. There are ways to control for that. France, for example, vaccinates its ducks, and they've been cleared to export them because countries, including the US, have confidence that they have enough biosecurity measures in place to make sure that they'll identify any sick ducks and also keep it from getting into the duck populations in the first place. So with good biosecurity, we could both vaccinate our chicks and still get clearance to export them. But that's been the sticking point, that the broiler industry is reluctant to vaccinate the chicks because they fear they will lose the export market. And so that's the tension right now. That would be one significant step. The other is that there's a whole category of eggs called breeder eggs that are basically eggs that are laid and incubated for purposes of breeding new chicks, but a lot of them don't hatch. If those eggs are handled correctly, where you pick them up quickly and you get them into a refrigerated setting quickly enough, where the odds of salmonella contaminating those eggs is sufficiently low, you could put those back into the food supply and either sell them as table eggs or more likely sell them as what we call breaker eggs, where they're broken and put in things like egg beaters, like eggs in cartons. And that would increase the egg supply. And the way this has been described to me by economists is that the egg market trades a lot like the oil market, where the last barrel of oil is what determines the price on the market. So it's what, we have 100 million barrels of oil a day, but if you take 1 million barrels off the market, suddenly the price shoots up. And it's similar to eggs. If you can get a couple of incremental millions of eggs onto the market, you're going to get a big price reduction. And so this may be one way to get more supply into the market. The concern by FDA, and I'll pause here, but the concern by FDA always was that, and this was the egg rule that we put in place back in the early 2000s when Mark McClellan and Les Crawford were there. But the concern was that the breeder eggs, typically table eggs are taken out and put in 45 degrees right away. You refrigerate them right away to reduce the chance of salmonella.
The breeder eggs are allowed to stay out longer.
They're allowed to stay in warm temperature because you're incubating them, and then they don't hatch. So, but if they could collect them more quickly, which is logistically challenging, and get them into cool temperatures when they're showing signs of not incubating, those could be used as table eggs.
Have a question out there?
Hi, Mike Koskowski. I work in healthcare communications. So I was at the JPM forum and I saw the CNBC piece, and I feel like you've got a real crystal ball on things. But I wanted to ask a little bit more about RFK Jr.'s communications approach, because I see like the Fox News interview that he gave a couple of weeks ago. He's recommending cod liver oil. There's a lot of sort of, this is about malnutrition when it comes to the measles outbreak. And I'm just curious, just taking a step back from the sort of details of this, he's promulgating a lot of theories from a communications standpoint. What's the counter? Like, how do we think about the risks from a communications standpoint of, I would call it junk science, being out there and being believed? And, how do you kind of counter that in an environment where that's not favored?
Look, it's very hard now. I mean, obviously, he has an audience that has been following his pronouncements for a very long time. They're going to still continue to follow what he says and be influenced by it. But now he's got all the stature of the office and has the ability to reach beyond his typical core audience and influence, I think, a larger swath of Americans, many of whom are not familiar with him and may not be familiar with some of his past pronouncements and, have, quite frankly, a skepticism of what he says like I do. I think on the issues around vitamin A and cod liver oil and I think steroids, where he was talking about those in the context as treatments for measles, that is extremely damaging, because the impression it leaves to consumers is that there's a treatment available for measles when in fact there is not. And for a lot of parents who might have been contemplating getting their kids vaccinated in the setting of an outbreak of measles, if you hear that treatments are available, that may tip the balance of the equation in your mind not to get vaccinated because you think that there's something available when in fact there isn't. And the same way a lot of people, forego vaccination for COVID or flu because they know Tamiflu is available and Paxlovid's available. And, those aren't entirely irrational positions. A treatment's available, you're otherwise at low risk. If I get sick, I'll take the treatment. That's not what's available in this case. You don't have an active therapeutic available, and yet that certainly, there's no question in my mind, that left that impression for some cohort of people. I don't know how many, perhaps a lot, and that's going to have an impact on people's willingness to get vaccinated. And this is a large outbreak. We don't have a good sense of how large it is. We know, and I've been told by officials, that a lot of people aren't presenting to get diagnosed because there's some trepidation in the community. We've seen 2 deaths already. If you believe, as we know, that the case fatality rate is about 1 in 1,000 for measles, maybe even a little bit lower than that. If you extrapolate from 2 deaths, you would surmise that there are thousands of cases of measles and not simply the several hundred that's been reported. So this may be a much larger outbreak than what we're detecting right now.
2 things I'd want to ask you, and we are running short on time. The first, how big a deal are tariffs for the pharmaceutical industry? If these tariffs actually come to pass, what's that going to mean for drug companies?
Look, I think it's quite significant. If you look at the trade deficit with Europe, about 40% of the trade deficit with Europe are pharmaceuticals, because so much of the manufacturing has been moved into Europe, particularly Ireland, because of the tax advantage. I think the companies would be more than willing to move their manufacturing back into the U.S., particularly if you can get more favorable tax policies here in the U.S., but that's a multi-year effort. Even if you identify the manufacturing space here in the U.S., certainly if you have to build it, but even if you can identify manufacturing space, bringing a new facility, bringing a product back to the U.S. and getting that facility online is a multi-year effort with the regulatory clearances. There's ways to create a federal task force where you can expedite some of that work if they're really earnest about trying to get the manufacturing back here. But the immediate hit to cash flows would be substantial. And I think what people are hoping is that the administration tries to come to some kind of agreement that if a company is working to bring the manufacturing back to the U.S., that the tariffs wouldn't go into immediate effect. Because frankly, putting those tariffs into immediate effect, it's going to provide an incentive to bring the manufacturing back to the U.S., but it's also going to hit cash flows and impede your ability to do it in some respects, not to mention the impact that it would have on Ireland's economy. So, hopefully there's some kind of more sort of deliberate, thoughtful policy put in place where you create incentives and structures that allow the manufacturing to come back here, even absent changes in tax policy, without, bludgeoning companies with an immediate hit to cash flows, because over the years the U.S. has created a set of policies that both allowed and in many cases incentivized that manufacturing to go overseas.
And now one of the bull cases for pharma and biotech companies from some investors is that we'll see really large-scale deregulation. Obviously, there are places where we could see reductions in barriers, but do you think the kind of up to and including getting rid of phase 3, do you think anything like that is plausible, having run the agency?
No. I think that there's ways you can reform specific aspects of the review process.
Like you did with cell and gene therapy.
Like we did with cell gene therapy. I mean, some would argue it was both regulatory and deregulatory, but I think there's things that you can do to reform how you approve gene therapy constructs based on organ system rather than therapeutic space. They've talked about changing the evidentiary standard around ultra-rare diseases to allow substantial clinical experience to be the basis of approval. So there's ways to get out targeted guidance that would reform specific areas. But the idea that you're going to do away with phase 3 trials, there's a lot of statutory obligations that the agency's under. You'd probably need a change in the law. Not to mention the fact that there seems to be this sort of odd dichotomy in policy right now where on the one hand, people are talking about extreme deregulation, like getting rid of phase 3. And on the other hand, people are saying that 60,000 patients randomized controlled outcome studies for vaccines aren't a sufficient requirement.
There should have been larger randomized trials for the boosters, right?
So I don't know how you square those two, that we're going to do away with clinical trials in this context, but we don't have enough clinical evidence for product approvals. I think at some point, the sort of incongruity in those two positions starts to become more manifest.
Oh, we'll see. But I do want to summarize this. We are about out of time, but I think I have a 2-word summary of everything you just said. It's complicated. Regulation is, and simple solutions often are the, simple, obvious, and wrong.
Durable reform that has a meaningful impact on people's lives takes time.
All right. Thank you very much.
Thanks a lot.
For our next session, here to discuss what happens when the right entrepreneur meets the right investor, please welcome to the stage E. Larson, President and CEO of Expedition Therapeutics, Inc., Dr. Ken Song, Chairman, President, and CEO of Candid Therapeutics, and to moderate, Stat Biotech Startups and Venture Capital reporter Allison DeAngelis.
Hello. Well, thank you both for being here. You two each have such a unique personal background and I'm excited to bring you together on stage, but to kind of like brief, real fast. So you are coming off of a turn as an executive at Lian Bio. You were there during the time period where the company was kind of assessing its future, ultimately opted to, kind of sell off assets and shut down. You had been at Turning Point Therapeutics, which was later sold to Bristol Myers Squibb. And now you've got this new company, Expedition Therapeutics, that's licensing drug candidates from China. And Ken, you're coming at it from a little bit of a different point of view. You've just come off of this like rocket-powered biotech story. You took your radiopharma company, Raise Bio, public before selling it to BMS. And now you also have a new startup. You both have new startups. And there's a theme here. They're both licensing drugs from China. Yours is in the autoimmune field, having licensed some oncology assets from China. You two are also, kind of old friends and neighbors through your time living in California and also you being on Raise's board. Before you two just completely take over this chat and ignore me, I wanted to ask you. You just flew in from Shanghai. You're in the U.S. for a trip. There's a lot of curiosity right now, I think, in the biotech industry about what the Chinese ecosystem is like. Can you, start off by telling us what is happening on the ground? What is the Shanghai ecosystem like?
Yeah, thanks, Allison. It's been a really interesting experience for me. I've been boots on the ground in China for about 3 years now. And having visited a lot of labs, talked to a lot of companies and investors there, what I have seen has just, it's astonishing. It's really blown me away. The productivity, the speed, the scale at which things are happening, that growth trajectory is truly incredible. And I think that that's something that maybe a lot of people who haven't been to China are not really paying attention to. But it's certainly something I think you need to pay attention to because what's happening in China is no longer just about developing generic medicines or even Me Too. There's really been true innovation that's happening there. I'd like to advise people to think if it doesn't matter what therapeutic area, if it's a known target or known biology, just assume there's one in China. And if there's not one in China, there's probably 4 in China already.
So, I'm hearing more like 5 or 6 in China.
and that's not an exaggeration. So it's just something that we now need to keep in mind when we're thinking about investments and the strategic path forward, that's got to be part of your risk calculus.
Well, and it seems like you two have both really not even just taken that into account, but really steered towards this market, through each of your new companies. Ken, jump in here. Like, what brought kind of the Chinese biotech market into your orbit? What made you want to get involved with starting Candid Therapeutics, your new company?
Yeah, so at Candid, we've licensed in now 3 programs. All of them sort of originated in China. And that's really just coincidence. So it wasn't like when we started Candid, we're going to specifically go out and search only for assets based in China. We had this thesis around, can we find these bispecific antibodies that have been developed in oncology and repurpose them for autoimmune disease? And so we took a global look. We were agnostic in terms of where these would come from. But things in China are done just at a much larger scale, right? And I would say, I agree with Yi, over the last 5 or so years, it's moved literally from 10 years ago, China would have a lot of Me Too's. Like literally, they were undifferentiated. They were just like, we could be 3rd in class or 4th in class as a large enough market. But I think they've migrated now towards, hey, we're still working on validated biology or validated mechanisms, but we're making them better. And there's this question around like, is it still copycatting or whatever? Is it innovation? I mean, I don't really know what innovation necessarily means and like where you cut that off in terms of what's innovative versus what's not. I mean, if you look at some of the largest drug classes, so a lot of us are probably taking cholesterol-lowering medications and we're on different statins. The first statins that were introduced, pravastatin or simvastatin, they all targeted the same enzyme, HMG-CoA reductase. But then Pfizer came out years later, like they were 4th or 5th to market with Lipitor, which became the market leader. And it was like very chemically similar to all the other statins, but it had a better clinical profile in terms of a deeper reduction in cholesterol, et cetera, stuff like that. And then there was Crestor after that. And so was that innovation? I mean, I would say yes, because they became the market leader and made the drugs better. And I think that's what we're seeing a lot more in China in the last 5 years, is there's a large number of programs where I think people there realize we can't just be a Me Too, we can actually make these things better. More potent or a more convenient dosing schedule, et cetera. And those types of, I would say, improvements or discovery is happening globally, right? Even within pharma internally. But yeah, there's just a lot more of it happening just because there's a lot more investment, but there's just a lot more people that are doing things. And so just by sheer numbers, you're going to see more stuff there, is what I would say.
Well, yeah, that's something that we had talked about off stage because your Expedition Therapeutics is, if I am surmising it correctly, like the whole idea is to scour the ecosystem, find, hopefully some of the best assets, if not the best drug candidates out there, license them, and then, kind of bring them to the US and develop them, which is, we've been seeing that happening. You've got a little bit of a different business structure, but, what do you think the future looks like in terms of drug development in China and, how it will impact US biotech?
Yeah, so I think, unlike Ken, Expedition was formed specifically to look at the Chinese market for IP, for global development. But that's not to say that we are only focused on China in that sense. Anything you source needs to be competitive globally. So we think about the competitive environment. We're trying to find best-in-class molecules, best-in-disease molecules that can compete effectively. But as Ken noted, there's just such volume there, so much to look at, so much richness in China that we can really exclusively focus on that. Because I'll comment on one thing. A lot of people ask, can you take a trip to China for a week, go get a deal done, right? And I would have to say that is very difficult to do. In China, a lot of things are done relationship-based. There's a lot of opacity. There's no comprehensive database you can search if you're looking for a particular profile. You have to actually talk to a lot of companies, get under CDA. Their websites are not up to date. They often have a lot more in their portfolio than you realize. And so I think you have to do that work and build up that relationship in order to understand what it is you're potentially in licensing and making sure you do have something that is differentiated and competitive on a global basis.
I think just one thing that's interesting is, Yi and I are both Asian, but like I was born in Michigan. I was raised, like, I don't really speak any other language besides English. And I'm Korean by background, not Chinese. And so, I mean, on one hand, it's like, so like me going to find stuff in China, like, is really not that different than anyone else in this room. Like, I mean, I consider myself I'm American first. I was born in Michigan, like Midwest. You can't get any more like middle America than that. But you know what's interesting though is I think having grown up, from first generation parents, I do think that maybe one of the slight advantages though is, because I compare this to like my non-Asian work colleagues. I think I do have a greater appreciation of just Asian culture. And there are certain ways of dealing with, I would say, people in Asia, whether they be Japan or China or Korea or whatever. And those cultural norms are different than what they are in the States. And also the pace and reading people, and like you said, like if they're telling you something, like they may not be telling you the full picture. And then you're just kind of like, okay, how do I get there? Not just because you sign a CDA, that doesn't mean all of a sudden they open up the vault of information that's there. They really have to trust you. And I do think you do need to build a relationship. I do think being Asian does help a little bit, though. I'm not gonna lie. Because they just like look at you and they're like, oh, you kind of look like us.
They don't care that you're Korean American.
Well, the funny thing is actually, apparently, like my last name is Song, and you could be Chinese or Korean. And so they've kind of just adopted me as a Chinese American. Like literally, like there's this panel called WeChat, like this messaging platform, and there's these articles like I have a Chinese name apparently in China. So I was like, oh, that's interesting. So I tell people I'm Korean. They're like, oh, okay. They're like, oh, really? They're like surprised. And then we just
Ken
continue
is
on.
very popular in China.
Well, before, I mean, we could very easily get into, Ken's whole experiences abroad, which sounds like it could be its own panel. But I did want to ask, because I feel like I've been having a lot of conversations with people, here in US biotech that, have almost like an existential feeling of, hearing about so many drug candidates coming out of China. And at a time where, I'm sure everybody in this audience can agree, because we've been having this conversation all day, there's so many kind of threats to what US science and the future of US scientific innovation is going to be. Where do you land on, this trend that we're seeing, and if it's a threat to kind of the US biotech ecosystem, or whether it's a friend?
So I'll go first. I think threat is the wrong word to think about it. But I do think you have to acknowledge that this is happening, that this trend, this is the new reality, and we can't get complacent. I think there are things in the US where we are still far ahead of what China is doing. The talent pool here, the experience, the clinical development, the strategic leadership, the capital markets, the robustness of all that, those are all other ingredients that are needed to have successful drugs. We're still far ahead, but we can't remain complacent. So I think that what's happening in China, in the best way of looking at this, should make everybody better. We need to adapt and evolve, understand what's happening, and think about where are our strategic advantages, what should we be doing to stay ahead. And ultimately, I do think that will produce better medicines more efficiently and benefit patients at the end of the day. So I don't think of it as a threat, but not a friend, but not a threat. I think that we can all become better because of this.
Yeah, I think threat is definitely the wrong word. I mean, I just sort of think about it like there's always, our sector in drug development, there's always things that are changing, right? And I think if people want things to stay the same and there's something that's new that's being introduced, then people like to use the word, oh, this is a threat, rather than people embracing, well, this is just the new reality of what it is now. So I mean, I think about like, even just simple things like when people are sequencing the human genome, right? Like, was that a threat, right? Or when you have these privatized research institutions in the US. So a lot of times, a lot of basic science research is happening in the public universities, but now you privatize research institutions that get benefactors' money. Is that a threat? I mean, that's a threat to academia? I was like, no, that's just another vehicle by which research is being done, and perhaps in a much more focused and translational way. The whole emergence of biotech and venture capitalists, is that a threat to the industry for biopharma? I mean, No, that just was a new opportunity that presented itself. I mean, you could say this anywhere, even going back to history, like was the printing press, was that a bad thing, right? Was the Industrial Revolution, was that a bad thing? Was the internet a threat, right? So I don't think we should look at it this way. I mean, I sort of look at this just as, this is just the state of affairs. There is now a shift, I think, in the way of how the industry at large needs to think about discovery. Discovery around targets that are known or molecules that are already out there in early development. Because if you want to just say, oh, the old venture capital model, I used to be a venture capitalist, the old venture capital model of, oh, let me go find a bunch of super talented chemists, right? Here's a known drug target. I'm just going to give them a bunch of money. They're going to do discovery, and I'm going to fund that, and that's going to create a lot of value. I personally think that investment thesis is completely broken, right? Because you don't need to do that anymore, right? Because now, if there's a drug target that's already known, highly likely that that chemical matter already exists, right, across a spectrum from early discovery even into the clinic. And it's more efficient to go and search and find those assets that you can in-license and then develop them rather than sort of starting off anew. I mean, AI, that's another one. Is AI a threat? I mean, are we, like, but you can either be scared of it and then you're gonna be left behind, or you can embrace it and then sort of take advantage of it. You know, I had an interesting conversation with a very senior pharma person a few months ago, and I said, you know, it's interesting, like if I was running pharma, I don't want this to be controversial, but I was like, if I was running pharma, I would think to myself, if we have like a list of 20 targets we're working on, you know, and the model is let's have our internal discovery team doing early discovery, I don't know that that's the best use of the dollars and time. It's almost like maybe you repurpose your pharma team to go find and first search, are there already existing chemical matter out there? It could be a hit or a lead or whatever, and you could save yourself a year or 18 months. And I would say the senior pharma executive, who I'll leave unnamed, did not disagree with that view.
Well, isn't like pharma haven't they already taken that kind of stance? I mean, most, you know, new drugs that are developed.
But they're doing both. They're in licensing, but they're still keeping their R&D budgets the same for early discovery. I mean, again, I'm not a pharma person. but I'm just saying that I'm big on efficiency. But it's just like, look, if there's an alternative way and there's limited budget, then you're gonna have to trade off somewhere. But again, that's just a reflection of just where things are today. Not for novel biology, et cetera, you still need that. But for things that are what most of our industry does, which is work on known targets, there are more efficient ways to probably jump from discovery to development. And if you're not buying into that, then you're just probably, your cost of capital that you're using to do that early discovery is probably so high. And I think the number of investors that are interested in funding those initiatives is quickly dwindling.
I want to put a pin in that really quickly because we have an audience question. I actually think we might have more than one.
We do.
Hi, Lonnie Levin. I'm at Weill Cornell Medicine, so I'm an academic scientist trying to do drug discovery. It sounds like it's out of date. Our tech transfer office didn't want us to nationalize our patents in China because their sort of party line is that China doesn't respect them. Is that out of date? And if it is, can I quote you guys to let them nationalize my patent in China as well?
I'm not an IP attorney. I think that is pretty, I mean, that's an older view. I would say that there is, I mean, look, you have global patent protection, et cetera. And I think for us, I'll just tell you, as we build out our patent portfolios, we look at sort of the major markets. And definitely you got US, EU, China is up there. I think it would be a mistake not to file the patents in China, just given the size of the opportunity. And also a lot of pharmaceutical companies and strategics are looking, if they acquire a product, at what the opportunity set is in China, because most pharmas have invested and built infrastructure in China and have commercialization there. So not to have patent protection there, I think, would be a mistake.
So I'll help you a little bit more even. So at Lian, we sold our Chinese rights to our lead asset back to BMS for $300 million for China market only. So that IP, I think, is important and respected.
I think we have one more question over here.
Sure. Kathy Edwards. What do the Chinese scientists think about the craziness that's happening in this country now? Are they talking about it? Are they cheering? Are they depressed, or what's happening?
Yeah, are you hearing about that at all from your staff, in China?
Yeah, I think broadly speaking, I think the Chinese population are paying attention to what's happening here in the U.S., mostly to try to understand how it might impact their work. And I wouldn't say, I don't want to get into the details of it, but I do think that they feel that the U.S. Innovation Center is still here, but they sense an opportunity to really try and catch up. So I'll just leave it at that.
Okay.
Very testy if you start asking geopolitical questions.
Well, then here's maybe more of a fun question. So as I mentioned, the two of you work together at Raise Bio, which was, one of the first in the class companies in the radiopharmaceutical space, which is a space that I didn't talk about 3 years ago.
Yeah.
How did that acquisition come about? Because Ken, you were CEO at a time. The company IPO'd in December, and then IPO'd in September, and then December BMS announces it's going to buy you. How did that happen? Did you have any inkling of that?
No, it's interesting. When we started Raise in 2020, very few people cared about it. There was sort of this legacy from 15 years ago with Bexar and Zevalin, and those were sort of commercial failures, which I think created a whole black cloud in the modality. Novartis had done some work in it, which I think garnered some interest from folks, but largely it was a dead field. But it sort of made sense, the modality made sense, it worked, it had some pretty clear advantages in oncology patients. And so as we're building Raise, and we're able to get investors, which I think are sort of early indicators of where the market could go towards, interested. We started talking to strategics, but I'll tell you, they were very much it was so new to them, and most pharmaceutical companies did not have anyone internal that knew how to diligence it or lead it. So when we went public in September of 23, which was not a great environment, we went public with no strategic interest. I wouldn't think we were dual track. We were not. We were one track. We're going to go public. The IPO went really well, and then it wasn't until a couple months later where then we started getting some inbounds around, hey, is there any partnering interest, et cetera. I have my own views on partnering, which I'm generally against, because I think that's the fastest path to destroying value. I'll just leave that there as a soundbite. But anyways,
I'm not sure I'm going to let you leave that there, but finish your thought.
But we ended up, even going into middle December, we didn't really have any sense that anyone was interested in acquiring the company. And I remember I had a board call, a week and a half before Christmas, because we were doing some discussions, but I said, hey, look, nothing's really happening. Everyone enjoy your Christmas. We got some important meetings at JP Morgan, and we'll update everyone in the first quarter.
Was that the case, as you recall it?
Yes,
it was Friday morning. It was 7 a.m. It was a super short call because there's nothing to say.
It was a very short board call.
And then I had 2 calls later that morning, literally back to back. And then in both those calls, we got unsolicited, surprising bids to take the company out. And so then I had a board meeting the next day on Saturday because I was like, oh, remember I said nothing's happening? We actually have to have a board discussion. And then over the span of literally just about a week period, it was a crazy process where a third pharma came in to the picture. There was a lot of back and forth. And then I was at the airport Christmas Eve, thinking again, we had a little bit of time to run through the process leading up to JP Morgan, when we got sort of a final bid, unsolicited again, because they kind of broke out of the process from BMS, but they wanted to sign the deal within 24 hours, which was Christmas morning. So then I left the airport. I did not get on the plane. I went back into the office, got my team in, and then got another board call going on the Christmas Eve. So it was a flurry of activity, and then 5 a.m. on Christmas morning, we signed the deal.
So that is
How
all
did that
accurate.
go over?
I have to say, Ken is being a little bit modest because when I was at Goldman, I've worked with some great M&A bankers, and I would say Ken is one of the best M&A bankers I've ever met. So he really did orchestrate a lot of this. It's one of the fastest deals that's ever happened, I think, from standing start to signing. And I think that's a testament to what he built at Raise, but also sort of how he managed the whole process as well.
Okay, we've got another audience question over here.
Hi, thank you. My name is Diego Garzón. I'm with Apertura Gene Therapy. I just wanted to ask something. Can you well, first of all, maybe I just say, obviously I think competition better than threats because it's going to make everyone better. Can you mention the statin example? And obviously it seems like the China's breakthroughs are really closing the delta on that. And you mentioned examples of where the US may have advantages. I'm curious if you could highlight maybe some of those areas that you're seeing China has those advantages and things that the US can take from it. And I'm also kind of just curious about whether that is also with registrational trials, because ultimately that's where the biggest capital is in our drug development process.
So maybe I can start on that last point. You talked about registrational trials. I think both Ken and I are looking at earlier stage assets, not sort of registration ready. And in part, that's because of this disparity or arbitrage that you're referring to, because there is much more availability of capital here for us to run those pivotal trials. And so that's why it makes a lot of sense for us to look for this IP. It's very inexpensive for these Chinese biotech to take, from discovery into early clinic, very fast, very cheap. But then as you move through the clinic, it becomes much more expensive. And there's not a lot of ways to keep that cost down. It is what it is. You have to run those global studies. And so that capital is readily available in the US. So that's why I think this model of in-licensing out of China, developing it globally makes a lot of sense. And I think it does benefit patients globally, but it's almost like a partnership because China's getting that ball rolling and we're kind of taking it across the finish line.
Yeah, and I'll just add, I think the strengths of what you see in the US and where I think China still may not be as caught up, at least on a broader scale, is still on the clinical development strategy. I mean, a lot of drugs die in the clinic, not because they're meant to die, but because the wrong trial was run or it was wrongly powered, et cetera. And I think the level of clinical development sort of strategy and sophistication is probably higher sort of in the US. It's there in China, but it's much more difficult to find. And I know sort of we're wrapping up here. I was just looking at the title, entrepreneur, investment, is it magic? I mean, I think what it is I do think, because I used to be an investor as well, and I've done several different companies, I do think the entrepreneur is critical. I think because the right entrepreneur will understand how to pivot and how to move around and try to get to the true answer. So I think it is true, you want the entrepreneur, you do need the money. I think you also need to be working in the right area. And I also think the other thing that's important that I've learned from my career is you've got to be willing to adapt to real-time information. So just because you spend a lot of time, et cetera, don't look at that as sort of like, oh, just because we've done it, we got to keep on going there.
A little sunk cost fallacy.
Yeah, exactly. That's a huge thing in our industry. And it's just like being unemotional about completely changing directions or pivoting based upon what's there. And then people say that, which is true. And then you just have to be like, it's timing and luck, ultimately. Those are two of the probably biggest factors that ultimately determines whether or not you're successful or not. The timing you can kind of control because you can predict what modalities you think may or may not be sort of the early part of the inflection curve. But obviously luck is luck. So.
Well, I told you backstage that you two were going to make me obsolete. You took up you took the wrapping up the session job from me. Please, help me thank my panelists for this really engaging talk. Okay.
For this sponsored conversation, please welcome to the stage Ultragenyx founder, president, and chief executive officer, Dr. Amol Kakis, and to moderate, STAT senior writer for medicine and editorial director of events, Matthew Herper.
So, a lot of titles, but what it doesn't say is that you are really a pioneer in the field of rare disease drugs, going back before Ultragenyx to BioMarin. Maybe just very quickly give the audience a quick a quick summary of that experience.
Yeah, sure, happy to. I started my academic career as an MD, PhD medical geneticist at UCLA, and I got into a project to develop an enzyme therapy for a rare disease. And I thought I'd finish that project, hand it off to a company, and go on to the next project.
And just to hit the themes I know we want to hit. So that means that in this disease, the whole thing that was causing all sorts of problems for these kids was that there was some enzyme that's missing. If you could make it, they should get better.
MPS I patients are missing one enzyme, and we can make it for them. We had cloned the genes that occurred in the early 1990s. If we made the enzyme, could we give it back? And we showed in animal models you could do that, and I thought, I didn't know much about the industry. I assumed, yeah, we'll just make the enzyme, show it works, hand it off to a company, and what I discovered is no one cared. And the tragic reality is I figured we've spent 30 years of NIH science to figure out what's wrong. We now know what to do, and we're going to write papers and leave them on the shelf, and we're not going to do anything with it when the kids need it. There's a fundamental injustice in that. It's like, why are we doing all this if we're not going to do something? So that was the problem. While I was near giving up, a family, the Dant family, found me. They had their son Ryan had MPS I, and then they said, look, you're our only hope. Our son's 5 years old. This treatment, this enzyme, is the only thing that could save him. Can you save him? And that's when it turned from, abstract basic science into the life of this kid. And then the question is, are you going to walk away from this kid, right? Not walk away from a project, but you're going to walk away from this kid. And honestly, I didn't have the courage to say I'm going to walk away from this because my academic career didn't allow it. So I decided we're just going to have to try to do this, and I have no idea what I'm doing and how to get there, but that's what we started.
So what's the difference then between being a CEO and being an academic?
Well, as a professor, you have sort of a narrow set of what you have to do and controls, and it's a small box you're in. And there's also limits to what you can achieve. When you're CEO, it's now wide open, and you have so many more responsibilities, so more people and stakeholders than you do that are immediate. Patients, of course, but investors, board members, employees whose career and lives depend on you. So the stress level is much greater, and you're trying to save kids at the same time. And so it is definitely, and also it's high profile.
Yeah. And
when something doesn't work in the lab before, you just had to do another experiment. When it doesn't work in a trial, you have a press release and cataclysm.
And you're public, so you have a stock price. But so you created Ultragenics with a specific thought around that issue that we just described, around you have kids with diseases where you think having an enzyme replace it. Describe the thesis for people here.
Well, I'd spent 11 years at BioMarin developing drugs and learning about the biotech industry and how to operate. And I left the company and was working on a foundation, write a book, and I thought I was done. But then all these families came to me and said, we have these ultra-rare diseases, we're trying to develop treatments, can you help us? And I realized that the work wasn't done, so we decided to form a company, and the thesis around Ultragenics was, can we form a company that will focus on ultra-rare genetic diseases where the science is very clear, the biology is just ready to be translated. There's people who were like me out in academic labs that had the answer that no one cared about. And can we pick those things up, put them in the clinic, and develop them more aggressively, more rapidly, and get them through? And that was the thesis. I seed funded it myself. So in 2010, I was me and my secretary and a couple $1 million.
And so usually people, or a lot of people argue that they don't like biomarkers as a way of approving drugs because they think that they want to see clinical data, they want to see clinical evidence. You actually think about this in rare diseases being the opposite, that in some diseases the biomarker is the better evidence.
No, I definitely agree that in many rare diseases where you know exactly the genetic cause, and there's a material that builds up that's toxic, that toxic material that builds up is a biomarker, but it's also the cause of disease. And if the treatment is replacing or fixing the underlying defect, that those are highly predictive situations, but we treat them like any other disease and we demand so much of them. And so in Ultragenics, one of the goals was to push forth on using biomarkers for rapid approval. We started that with MPS 7. the Mepsevii product for MPS 7. That disease affects 200 in the world. And Bill Sly, the scientist that did the work, it's been sitting fallow for 20 years. But I wanted to make that a test case. Okay, we're going to treat a disease with only 200 people, 20 people in the US affected. But can we use a biomarker, get it approved, demonstrate it works, and do the work? And we had still difficulty with the FDA at that time, right? And in Europe, it was accepted. We did get the product developed in 3 years, got it approved. But in the US, they were looking at the clinical data. They wouldn't accept the biomarker. So that case didn't get us open the door. But what is the difference? In this situation, the biomarker is not a random thing. It's not a blood test that doesn't mean anything. It is the toxic material. It is the cause of disease. And I think we need to think about designing treatments to affect the cause of disease rather than to think about treatments affecting the outcome of disease. The cause of disease is what you really want to fix. The outcome of disease are clinical things that happen many years later and are a combination effect of the disease plus a number of other variables. And the way I try to describe this, Matt, is like in a car example. It's like if you have the brakes broken on your car. Well, if your brakes are broken on your car, you will have crashes. But how you crash or how often may depend on who you're driving, if you're driving in Manhattan or if you're driving out in the Iowa corn farm, right? The risk of crash depends on a lot of other factors. But if you have brakes that are not working well, you're going to crash. But you can measure the brakes accurately, but measuring crashes is variable. It's inconsistent. It's hard to measure.
A medical example that I think was helpful was the example of HIV.
Yeah, in HIV, you measure the viral load. That's the disease cause. If you get rid of the disease cause, the outcomes will improve. If you try to measure the outcomes, like in HIV, if you talk about opportunistic infection, hospitalization, and death, it takes years for those things to develop, and they're highly variable with different secondary outcomes. You never figure out the way. So what I want to be clear about is the biomarker-based approval for HIV is not just speeding the approval of drug a little bit. That's what accelerated approval means to people. It is a transformation of treatment for the underlying cause, meaning drugs that really get at the virus, so that we end up developing a treatment that is actually fundamental. If we develop only clinical endpoint driven, we focus on outcomes, you would develop immune stimulants, which would make their immune system a little better for 12 weeks and maybe help them with an infection, but they're going to die anyways. Does this sound familiar? Are a lot of the drugs we're developing helping your symptoms late and chronic and never fixing them? This is the paradigm of depending on clinical outcomes. If we start looking at disease-caused biomarkers, we're now going to develop drugs that actually fix underlying problems, and we'll fix the clinical outcomes in a much more profound way, just like accelerated approval work for HIV.
Do you think that your barriers in doing that, in creating a paradigm that is something like an HIV for at least some rare diseases where you really understand what's wrong? That's obviously not all of them, right? Do you think the barriers are more regulatory or science-based?
I think it's primarily regulatory, and it's a little bit political too, because there's been so much written negatively about accelerated approval as being drugs that are experimental and not really proven. But the truth is that biomarker-based drugs can be more potent, more specific, more effective than drugs that just affect downstream disease issues. So the regulatory issue has been that the level of evidence required to allow or qualify a biomarker is so great that for many ultra-rare diseases, you can never meet the standard. And therefore, for the last decade, we haven't had accelerated approvals for neurologic genetic diseases, even though the biochemistry is very clear. The science is sound. It's not about science. But in the last couple of years, through a number of efforts, we were able to pull together a coalition of companies and academics around one particular disease area, MPS, brain disease, and held a Reagan-Udall conference, which is the FDA's Regulatory Science Foundation, and showed the world with that compilation of data that heparan sulfate, this material that accumulates in Sanfilippo and other MPS brain diseases, can predict outcome. And we're able to shift FDA's view of that marker. And now because of that, 5 programs for MPS brain disease that were going to be canceled or lost potentially are now getting accelerated and surviving. And so that's a step forward for the science. And we're very pleased that the FDA's change in view has put us in position now of treating not just MPS brain diseases, but so many other rare diseases that the new technology we have, gene therapy, gene editing, and other drugs can finally address. But if we didn't adopt that, we wouldn't be able to solve those problems. But I think now this first step, I'm hoping, will lead to more steps and a resurgence in the use of gene therapy, editing, other strategies to tackle some of these diseases that affect only a few thousand, where we know what's happening. Now you might think, that's only a few thousand people, what does it matter? Well, it feels too rare to matter, but I guarantee you, if it's your family, it doesn't feel so rare. And there's a whole lot of them, sitting out there waiting, and they're waiting for us to get out of our own way, cut through red tape, believe the science, and start pushing forward and getting drugs approved that need to be approved. And I think the FDA has made a great push forward, and we're real excited about the possibilities of changing things in the next few years.
All right. We're going to have to end there. But thank you very much, and thank you for sharing your story.
Well, thanks for having me.
To all of you.
By the way,
yep. I told you at the beginning of the day how amazing my colleagues at STAT are. Are we on? I told you at the beginning of the day how amazing my colleagues at STAT are. Now you're going to hear from a bunch of them, just some of the best reporters around. Please welcome STAT chronic disease reporter Isabella Cueto, STAT FDA reporter Lizzie Lawrence, STAT Washington correspondent Sarah Overmole, STAT West Coast Biotech and Life Sciences reporter Jonathan Wozen, and to moderate, STAT co-founder and executive editor Rick Burke.
Thank you for sticking it out, and everyone with the food, come on in. This is going to be really fun. So really, bring your food and come on in. So it won't be Bill Nye the Science Guy, but it's going to be good. So one of my favorite things is to show off our great STAT reporters, and we got a lot of them here today, but here are 4 of them, and I wanted to especially focus on invite 3 of our reporters from our DC office because they're right on the front lines of everything going on. But let me start with San Diego with Dr. Jonathan Wozen, who's a PhD in immunology from Stanford, who knows what he's talking about. And I thought it'd be great to start with Jonathan, who covers biotech life sciences on the West Coast. And I just want to ask you, you're writing story after story about the brain drain and what's going on in scientific research. Put yourself in your shoes from back your research days and sort of what's your perspective on all that's going on? And are you glad you, thank God, went into journalism?
I like the work I'm doing right now.
Well, good.
And there's a lot of it to do. Yeah, we don't often really get to talk about ourselves as reporters, spend a lot of time talking about institutions and the people who make them work. But yeah, I was a graduate student. I got my PhD at Stanford in immunology, and so I am acutely maybe painfully aware of how hard it is to do science just inherently. You know, you're literally creating knowledge. I don't know if there really are, if there's anything else professionally where you're creating knowledge. We spend a lot of time, excuse me, gathering, that's okay, synthesizing knowledge, not necessarily creating it. So, you know, it's hard to come up with good scientific questions, hard to design experiments, hard to collect data, hard to analyze it, hard to submit papers and go through peer review, hard to get grants, you know, funded. So science is inherently hard. It's hard to start a career in science, hard to sustain one for all sorts of reasons. I guess what I see right now is that science is hard for reasons that don't really have to do with the science itself, right? So reasons that have to do with, you know, people are now thinking about whether their grants are going to be abruptly terminated for using the wrong words, or whether they're going to lose funding because of who's on the swim team at their university. So a lot of the challenges I see right now and that we're writing about and tracking don't really have to do with the nuts and bolts of science. And I think that puts researchers in a tricky position because you're not really trained as a graduate student to prepare yourself for funding opportunities being abruptly taken away because they touch on diversity, equity, and inclusion, and the administration's taking a very broad view of that. Or, you know, postdocs who are looking for funding opportunities that can help get them to, you know, one of those very rare and very prized faculty jobs. So I see research under strain in ways that, you know, researchers aren't really that equipped to deal with. And, you know, I still know a fair amount of people in that world. I was literally just texting with one of my friends from undergrad who's currently a grad student in Southern California, and we became friends through a program, a summer program that existed or that exists to support minority students, students of color, and, you know, those are programs that are being phased out at a lot of places across the country. So yeah, it's hard not to, you know, see what's happening and remember that, you know, 5 or so years ago I was a graduate student, and to kind of think about how I deal with that. And I don't have a clear answer on that front, but it's a tough time.
Thanks, Jonathan. Let me jump to DC and Lizzie. Let me actually jump to the Maryland suburbs. You were just at the FDA. What was it this week, or like the days keep like blending into one another? You were just at the FDA when they called everyone back to work in person, and it sounded like a nightmare. Can you describe the scene that you saw?
Yeah, so it occurred to me, I was getting texts from FDA employees on the Sunday night, you know, that return to work was on Monday, and people were anticipating, you know, not being able to find parking, massive lines, chaos, all, you know, and I was like, I should just go and like see this for myself. It turned out that I was there a little bit later. I think everyone, I got there around 8:39, and people had left as early as 4:30 in the morning or 5 just to make sure that they could get to the campus. So by the time I was there, it was a lot calmer. There were still some lines, and folks were, you know, I heard just people saying, wow, it's been so long since we've been here. So on the surface, it looked, you know, pretty normal. Under the surface, I mean, I've been getting so many employees reaching out, you know, who normally would never talk to press, but are talking to the press now because they've just found working conditions to be so impossible. I think in Maryland at the main campus, things are a little bit better just because it's a bigger space. Although, okay, a few weeks ago, I had some employees say they were concerned about the water quality, that some tests had tested positive, some water had tested positive for Legionella, so they weren't sure if they were going to be able to drink water at the office. But and then there are folks all across the country that were hired remotely and are now being told to go to federal buildings, you know, courthouses. I was talking to someone who was put in a supply closet with jail cells directly upstairs. Someone texted me the other day in Utah, was in a closet and had her laptop on a garbage can. Like, it was just ridiculous. Very, very absurd working conditions. So I think it's tough over there right now.
Let me jump now to Sarah. You've covered all these new leaders that have been appointed in these health agencies, and we just heard on stage today, we just heard Scott Gottlieb, Tom Frieden, former federal officials talk about their concerns about measles and other and sort of what the government is doing right now. And you just wrote a piece, a big examination of Dave Weldon, the CDC choice. It was pulled the morning of the Senate hearing. And you remember Dave who now you're saying, because like it seems like that lasted for a second. But you've had the perspective of writing about all these different nominees and RFK Jr. Like, what do we make of them and their degree of being in or outside the mainstream? If you could quickly go through these people.
Yeah. So of course, we all know about Robert F. Kennedy Jr. And I think, like you said, a lot of people have talked about him today and how his commentary has evolved, or like the concerns they have over the measles messaging. Dave Weldon was the CDC nominee until about 2 hours before his confirmation hearing. And as you said, I went to investigate sort of his congressional archives. And when I was looking at there, ran a lot of parallels to RFK Jr. over the early aughts as well. I saw someone who, like many people in the early aughts, it was really striking looking through archives from 2001 to 2002, 2003, where it wasn't just someone like Dave Weldon who was asking questions about vaccines and autism. He had New York Congressman Carolyn Maloney was co-signed on some of those letters. Like, they were just Jim Greenwood, who became a bio chairman, was signed on some of those. So the early aughts, it was a different time, or at least it was a time where people were trying, I guess, in good faith to ask those questions, but then they got answered. And so as you went through the years, you saw people falling off of those letters, falling off of those investigations he was asking for, that legislation he was asking for, and him sort of standing alone saying, okay, you gave me data, but that wasn't the data that I wanted, or it's not good enough. And that's a lot like how Robert F. Kennedy Jr. has approached this. He's appeared before Senate committees and said, just show me good science. That's all I'm asking for, is good science. And then when people say, but there's this science and there's that science and there's this, he says, well, we're going to do it my way. And that's kind of the main thread that we've seen through all of the nominees. I know I just talked about the two of them, but when we listen to the FDA commissioner nominee, Marty McCary, or the NIH director nominee, Jay Bhattacharya, they both have also taken this tact of, look, I think vaccines are okay, but what does it hurt to get more data? And that's kind of the through line with all of them.
Do you feel so far that RFK Jr. is really calling the shots with all of them?
I think to a certain extent, yes, if not him directly, more that they know that to President Trump right now, it's really important to look in line and be like a true believer in these priorities, and that the Make America Healthy Again movement is very popular and it attracts a lot of voters as well. I don't know that necessarily Robert F. Kennedy Jr. is calling those shots with all these agencies, especially because, we haven't even talked about Medicare and Medicaid yet. I don't think that he has much. He's talked about Medicare and Medicaid very much. I think with Weldon getting withdrawn from the nomination, we're really going to see going forward how much influence Robert F. Kennedy actually does have, because if they go in the same vein as a sort of Weldon type who's been skeptical of vaccines, then yes, he still maintains authority and clearly has some influence and sway with the White House. But if they swing in another direction, choose someone safe, then I think that's going to be the first time we've seen him hit the MAHA wall a bit.
Speaking of MAHA, let me turn to Issa, who's more than 2 years ago, we made you our first chronic health reporter, chronic disease reporter, and talk about great timing, because look what's happened. And so Issa has been sort of totally on the front lines and ahead on the MAHA movement and who the players are and so forth. Can you, and you know a lot of these people, you've reported them on them. What do we need to know about what these, is there a way to, without generalizing too much, to say sort of what are these this cohort of people like, and what's their understanding of the science?
Well, I think the interesting thing about MAHA is nobody really knows what it is, and so you can kind of shape whatever you want to be MAHA, and we're kind of seeing that on the state level right now with a lot of different bills that are about everything ranging from mRNA to food labels and obviously food additives. And so I think the food and nutrition is obviously an area of focus. It's maybe the area that RFK Jr. is most outwardly conversational and passionate about, and I think that's why he would go for someone like Dr. McCary that maybe he can bring into that work. But we also have this presidential MAHA commission, which just met for the first time. It was not a public meeting, so we don't know everything that was discussed in there. But there were MAHA moms that were brought in, and they were talking about infant formula and the supply, and we've already brought that up today, and that's already becoming an action item. And so I think it will continue to evolve and pivot in a lot of different directions. The main question I have is, how do you actually pursue the quest of reducing chronic disease when the administration that you're in is taking all these steps that don't support that goal? And can you reconcile those things?
Let me ask any of the four of you, you're all kind of jumping on stories nonstop to try to make some sense for our readers of what's really going on and trying to get to the bottom of some of these decisions or changes. Are you finding that some of the sources you deal with at these agencies or institutions are scared? And how does that manifest itself? Sarah, yes. What would you say?
Yeah, it's, leaking from an agency at any time in any administration has its risks for people, and they know that. But the rhetoric, especially in the second Trump administration, has been that at minimum you'll lose your job. But he's talked about it being treasonous, et cetera. So a lot of people have changed the methods by which they communicate. So it'll be like Signal, it'll be something encrypted. And I think they'll take more precautionary steps. But then another way that things have changed a lot, and Lizzie touched on this a bit as well, during the first Trump administration, I think that a lot of the people that I was talking to on a more regular basis were what we call the politicals. So you have like the career staff who are there year after year after year, regardless of who is the president and who is the commissioner or the secretary. And then you have the political staff who come in with that one president. First Trump administration, a lot of my sources were actually the politicals. Now, like Lizzie's saying, a lot of people are coming out of the woodwork who have worked at these places for years, never saw themselves leaking something, never saw themselves being part of these narratives at all, but they feel moved to for certain reasons because of their job, because of anxiety over the work that they've done. So that's one way that's changed a lot too.
So even though people are more afraid to speak out, more people are speaking out because things are so bad? Lizzie, are you finding, what are you finding?
Yeah, no, I think that people are just so distressed by what's happening, and also their jobs. These are supposed to be, they expect to work in government for decades, and now that is actively being threatened. So there's a little bit less to lose, I guess. But also, they just feel so passionately that they want people to know what's going on, that they're finding the courage to share and to leak things.
Before we get to, I want to, anyone that has questions, but before we get to that, I just want to ask Jonathan and or Isa your sense of the impact of these cuts on progress in medicine and finding cures and research. Like, is it too early to, it's so early, but can you give it, what's your sense of that?
Sure, I can start, and not even exclusively drawing on things I've written, but things that my colleagues have written. Elaine Chen had a great piece about how a long-running diabetes study is now being stopped because of the fact that Columbia is involved, and how that jives with the chronic disease focus. It's hard to really square those things. But if you're canceling grants, then you're canceling the research that happens funded through those grants, and so whatever fruits would come out of that, that's being jeopardized unless researchers can quickly find alternate sources of funding, which isn't easy, and there are only so many other places to go. The federal government's clearly the #1 funder of biomedical research, and particularly when it comes to supporting facilities and administrative or indirect costs, a phrase people have probably heard a lot about recently, which sounds abstract, but includes things like keeping the lights on in lab and custodial fees and administrators who are involved in preparing grants, and these are payments that are negotiated on a regular basis with the federal government. So I think the expectation, I mean, I'll just, maybe one interesting point, when I was working on a story with Angus Chen, a good colleague, a cancer reporter, about concerns that there's going to be a major scientific brain drain, which you heard a bunch of people say today. I spoke with a couple researchers who had put together a study, I think in 2024, 2023, looking at the impact of past funding disruptions on labs that are dependent on R01s. And what they had found was that even a 1-month delay in eventually funding or sort of continuing funding on an R01 can lead to an eventual decrease can lead to researchers in those affected labs being less likely to stay in the U.S., less likely to be part of the U.S. workforce. And that's off of a 1-month delay in funding that's eventually brought back. So I think it's going to take time to actually see the data, but every expectation I have is that this is going to have a real impact.
Yeah, I think we're just beginning to understand the ramifications. And I wrote a story yesterday about a center at Columbia University for ME/CFS, which is can be an extremely severe and disabling condition. These are some of the sickest patients. It's been continuously underfunded for decades. There have been people who have been in their beds for decades, not able to leave the house with this condition. And this is a major research center, and they are shut down. And they had, Dr. Ian Lipkin, who's one of the main researchers there, told me, I have data that came in from 2 weeks ago on Gulf War illness that I cannot analyze because I am not allowed to work on it. And so that's a tangible impact that we're feeling. In many other ways, I think of like how we never know about the crises that are averted or prevented. It's similar. Like we won't know all of the findings and all the breakthroughs that we could have had during this time.
Yeah. Let me go to questions.
Hi, my name is Anita Kishore, and I'm a former scientist and coach and advocate to the scientific workforce. And I want to say thank you for your coverage of these topics. And how are you making editorial decisions, allocating resources to such a diverse range of topics on things like cutting edge technology from CRISPR through to the politics of the scientific workforce?
Well, let me answer that, because what I would, or start answering this. This is a challenge for us every day. I wish we had like 3 times the reporters we have, because there's so much to cover. So we have to make choices every day about what's the highest and best use of these reporters and all our reporters around the country every day, about what are stories and targets to report on that no one else is doing, that can really add to the knowledge base and the authority. So we just make those decisions every day. And let me just say, each of these reporters is just, like, there's less time for some of the maybe longer narrative, big, like spend 2 months researching this, because we've got to find out what happens at this agency at this point. So we're kind, I wouldn't say reactive, because I don't like that. We're very aggressive, but it's hard to sort of keep on top of what's going on. And I've seen these reporters, like, we'll work, there was one Sunday where we had 8 reporters on one story, and the story kind of can change by the hour. People lose their jobs and they get their jobs, or, does anyone else want to jump in and sort of how this moment has affected your job in terms of how you approach stories? Anyone want to?
Well, I think covering indirect costs directly cost me a whole weekend. I think the answer in terms of editorial decisions, Rick already gave a great answer. I think it's something we're all figuring out as we go in conversation with each other and our editors, and the answer has changed over the weeks. As I think in the beginning, probably anything we could do to cover any instance of a program that was being pulled back, we were more inclined to jump on. But frankly, there's just so much happening now that we do have to think about our shot selection and, are we telling readers something that's meaningfully different from what we've told them before and that advances the story in some way?
Let's go to another,
So it seems to me that that chronic diseases are so linked to equity and diversity. So are they just talking about chronic diseases in white people, or are they interested in talking about chronic diseases in all U.S. people?
I think we are awaiting a response on how they're thinking about chronic disease. I am watching very closely this commission to see what they actually decide to study, what data they decide to look at, because I think you're absolutely right that you can't address chronic disease without acknowledging that it's mostly affecting poor Americans, Americans of color, other minorities. So I think it's like a really important, undeniable contradiction within the administration.
Let's go to the next.
Hi, everyone. My name is Lippy Roy. I'm an internist and an addiction medicine physician. I'm also a national media medical commentator and speaker. First of all, Rick, Matt, the whole STAT team, thank you for putting on this really incredible event. Few things that a couple of things I'd like to know. First of all, let me just say, as somebody who's on the front lines, I work at a methadone clinic. I treat people, some of the most marginalized, vulnerable individuals. They're not vulnerable, but the situations around them make them vulnerable. That are at extremely high risk for serious illness, hospitalization, and death. Not just from their opiate use disorder, their substance use disorders, or behavioral addictions. And then on top of everything else that's happening, life will get considerably worse. Earlier, Rick asked, instead of the word disruption, what word would you use? I'll tell you, as a frontline doctor, medical professional, it's not anecdotal. I'm seeing it. It's devastation. So that's the word I would use, and it's only going to get worse. My question for all of you is, what would you want, or what more would you need from, like, say, medical professionals like me in terms of to really optimize doing your job, or like institutions, even like small clinics, community health centers, where, by the way, you would get really great stories and feedback. But what more can we do to help you? I'm also on both sides as media, but I'm also a physician too. So just, that'd be helpful. Thank you.
Go ahead.
I think, honestly, what you were just talking about, like saying what you see firsthand, and I know, of course, there's sensitivities around that because you're talking about your patients and you want to be sensitive to them and their experiences, but when we hear things like that, when we get it put in real terms by someone who sees it, and even just about how it made you feel, or like how you're thinking about where things go, I tend to ask people at the end of interviews, like, is there a question I should have asked you that I didn't? Or like, is there something you wish you saw in the media more? And I tend to get like the most interesting insights at that part of the conversation. So yeah, I think it's just being a resource like that. And even if it isn't an on-the-record interview, like getting on the phone and being like, can I ask you some stupid questions? Can you just explain this thing to me? And usually the person who knows it best can explain it the simplest.
We are out of time, but let me take one last question because there were a lot here. There's one right over here.
Yeah, tell us your stories.
Make it a really long one.
Hi, STAT team. You know I love you all. But my question for you to bring us home again is, where have you been surprised in who is stepping forward and who is being more transparent? And where have you been disappointed with who has recoiled and stepped back from some of these topics that we're discussing today?
That's a good one. Who wants to take that? Lizzie.
Well, yeah, like we've said, I've been surprised that people, career civil servants in government, have been willing to speak up. Surprised, even my panel with our really courageous researchers, that was, that's difficult to do. I think people in industry have been a lot more hesitant, and they've talked to me, but to actually get on the record statements from trade associations, and that has been challenging. And I think for me and for others at STAT.
That's honestly the answer for
Yeah.
it's industry.
Yeah.
It's just like this is going to impact you too eventually, and so it's like, when do you hit your line? Yeah.
Yeah, I mean, I was talking with somebody at a local research institute in San Diego who was complaining that their trade group, Biocom, hasn't been vocal about the indirect cost policy that NIH is trying to implement, and essentially saying, we're paying members of Biocom if they're not standing up for us. Why are we even part of this organization? So I think the industry silence isn't just difficult for us as reporters. It's been difficult for academics as well.
Thank you. I could ask. I have a million more questions, and I know you all do too. But we got to go. So thank you so much.
Thank you.
Thank you.
With that, we are at our second and final coffee break of the day. So back outside of this room, you're going to find the sponsor booths as well as afternoon snacks, coffee, and tea. Go check them out, and we will be back here at 4:15 p.m. Thank you all.
Please take your seats. Our presentation will begin in 5 minutes. Thank you. Please take your seats. Our presentation is about to begin. Thank you.
So is everybody interested in a session that might not be about the world possibly ending? Because this is a really great story you're about to hear, but it is not that one. Please welcome to the stage Andrew Baum, Pfizer's Chief Strategy Innovation Officer. And we're going to, this is your life, Andrew. But I do actually want to start for a very long time, Andrew was a European-based sell-side analyst. And during that time, I think you had a buy on Pfizer how many times?
Once, in 2010, briefly.
And other times it was a neutral or a sell, which most people know that analysts generally don't give very often. So what led you to decide to work for a company that you had so long been skeptical of?
So it's a very fair question, and it's good to be here. The main reason is the conversations I had with Albert when he...
That being Albert Bourla, the CEO of Pfizer.
When he approached me, which would have been the early part of last year. And understanding that he was frustrated with the way things had evolved and obviously the share price evolution. And I found him incredibly pragmatic and thoughtful and recognized the challenges both for the industry but also in Pfizer. And when I looked at not just the conversation I had with him, but all my now colleagues on the LT, I saw that, look, this is a company where it has a storied legacy, both in development of many first-in-class drugs. And actually, this is a key point. Pfizer's been remarkably successful in producing lots of drugs, including first-in-class drugs. The challenge is we've produced a lot of drugs, and unfortunately, many of them haven't generated the revenues we had hoped. Mm-hmm. And if I think about what would I rather have as a problem, a company that can't discover and develop drugs, or a company which discovers, develop drugs, but for a variety of reasons hasn't translated into revenue, I would much rather have the second, because that's much easier to fix. And so that's very much how I characterize Pfizer from the outside. An effective R&D machine, which had prosecuted, and obviously medicinal chemistry is incredibly strong, the first-in-class JAK, the first-in-class ALK inhibitor, the first-in-class CDK4/6, and on and on and on. First, they were almost first-in-class bispecific in myeloma. But there's been a lot of investment in other drugs which really haven't enjoyed success. So we recently deprioritized a couple of gene therapies, which were highly innovative, but the need wasn't there. We have a JAK, which was designed to compete against Rinvoq, but it hasn't delivered. So the key point was I could see a sausage machine that was very effective at making sausages. It's just they're not the right type of sausage at the right time in the right place. And that's something that I thought I could leverage my skills and really help deliver more drugs to patients and at the same time help create shareholder value.
But what makes you think that you can identify the sausages people want to eat? I mean, you were a very good sell-side analyst. You're very good at predicting, both forecasting but what makes you think that you'll be able to fix that taste problem? That taste problem of, it's an ALK inhibitor might have seemed like a much bigger thing. That's a cancer drug. It's an amazing cancer drug. It makes people's, if they have ALK or ROS1 cancer, they have non-small cell lung cancer, it makes tumors just shrink and people stay alive for years, right? Some of these medicines were amazing and not very big successes. So what's going wrong that?
Well, I correct the crizotinib, which was Xalkori and now Lorbrena are going to be very substantial drugs.
Right.
Now, is ALK positive non-small cell lung cancer as large as HER2 breast cancer or other patient populations? No, it's not.
Right.
But still, as a meaningful commercial opportunity, it's very, very significant. So I would differentiate that and obviously drugs like Ibrance, which have been tremendous revenue successes, against the molecules that I highlighted, so the gene therapies, for example. I think Sabenco is a really good example to answer the question that you posed me. Why do I think I can identify? So again, hindsight's wonderful, so I want to be humble because I'm going to refer back to things post hoc. But it was very clear from where I was sitting that with Hemlibra on the market, you had a very well-served patient population, and now you've got long-acting factor
We're talking about
VIII.
hemophilia just to keep everybody up.
And in particular, this patient population, hemophiliacs, have a history of very deep scars associated with the blood transfusions, with HIV and hepatitis from the 1980s. So if there is a very well-tolerated, very effective therapy, the willingness to adopt a highly experimental therapy,
this is a gene therapy.
Which is the gene therapy, as Matt says, for little gain, really didn't strike me as a very good idea. So in retrospect, and I would have argued ahead of time, why are they doing it? So to avoid making that kind of decision would be something that I would bring to the party. Similarly, again, with humility, because hindsight, I said, is easy, but if you are competing against an incumbent which has a $20 billion drug with the rebate attached to it, in this case I'm talking about AbbVie with Humira, you need to be very thoughtful about how you're going to get formulary access for your competitor if you don't have differentiation. Is that a fight you can win unless you're going into a particular indication that's unique or you have a differentiated asset? So those would be two examples.
And that's a very hard thing you just talked about, because you're saying you're going to stop development, not you're going to have a drug you think is effective and decide it's not effective enough to get formulary placement late in the game then.
No, I think we look at the profile of the drug and we find an indication, right, where we think this drug can fit in and differentiate versus the incumbent. Because there is always opportunities to go around and to deliver, in some cases it's going to be second line, some cases it's going to be other indications, because when you look at the JAKs, there's a plethora of indications. Mm-hmm. So I think one of the things which AbbVie did very well is they went very, very broad simultaneously. So they not only had the rebate wall, but they had a very comprehensive, very broad development plan. I think in retrospect, that's something that one would have liked to have done if you're going to embrace that challenge. So I don't want to over-index on a couple of examples, but I'm trying to give you two discrete examples where you may have come to a very different conclusion, given the unique dynamics related to those two different scenarios.
So let's talk about an example that's forward-looking that people are really interested in hearing about, which is danuglipron, Pfizer's oral GLP-1. You're still in the race. You certainly fell behind where some people were hoping you were going to be with the readout of the trial and the need to do more studies to figure out the profile before going forward. But talk about the strategy there. I mean, you're probably dealing with, if this oral GLP-1 works, I don't think most people think it's going to be the most effective. I don't know how to think about the side effects. Like, what's the reason to think about continuing to develop it?
So we will, again, we haven't disclosed.
You haven't disclosed doing danuglipron. You have said you're going to be in obesity. Albert said that very forcefully.
We have said we're going to be in obesity. We haven't disclosed exactly what we're doing. We've said we'll be updating the market around the first quarter, right? Mm-hmm. So that's kind of where we're at. And to make sure everyone's on the same page, danuglipron is our small molecule GLP-1 agonist. So if we were to take it forward, we would be the second to market small molecule oral GLP-1 behind orforglipron, which is obviously completing its phase 3 trials. And it's true, we would not be first to market. However, as I don't think I need to say to anyone in this room, this is a very, very substantial market, and it is a market which is open to potential commercial innovation. And what do I mean by that is, well, there's obviously potential coverage through PBMs, there's potential coverage through employer riders, there's potential coverage for self-pay. And we're very thoughtful about where we position Danu. However, because I suspect Matt's going to come back to me on that, before we just anchor it around Danu, I want everyone to remember that we will be starting, if we take Danu forward, combination trials with our oral GLP-1 plus a GIP agonist, where we are very confident that we'll be able to increase the efficacy. So to Matt's point,
it's another related mechanism.
You're building in clinical differentiation, which gives exactly the point that you then are able to compete both through clinical differentiation as well as through commercial innovation.
What's been the best moment of these past 7 months for you at Pfizer?
Look, I think every day I wake up.
Every day can't be the best moment.
No.
Nobody has that.
No, look, to be honest, I was doing research for more than 25 years since I left clinical medicine. And I loved that career, right? It was a great career. But after you've been doing quarter after quarter, you count your life in quarters. How many GSK quarters can you count in your career? To go into a company which is delivering novel drugs to patients, allocating capital as we are doing, and understanding the challenges, both from a legislative point of view as well as from an R&D, it's truly exhilarating. And the sense of purpose that you get by working with colleagues and knowing that you're accelerating bringing a novel ADC or drug for cachexia, it really does energize me because remember, my background is clinical medicine. So I've treated patients with GIST, I've treated patients with breast cancer. I remember sitting in front of each of these individual patients, the patient with myeloma before Revlimid was a thing, who was given months to live. So I've seen and experienced, both professionally and also through my personal life, the impact of drugs on patients. So a chance to have a hand in that and to shape it, particularly for a company which is frustrated because we tasted the glorious highs during COVID, and to help that company get back on track, that's what I find exhilarating.
If you're making an argument to someone who is sitting in your old seat,
yes,
some very skeptical analysts, what do you say to them without going through we don't have time to go through the entire balance sheet right now. It would be fun. But what basic arguments do you make to them that they're missing something? What areas do you think they're missing most?
So I think the onus is on us to showcase to investors the growth that we are going to deliver, both with our recently acquired products, which the likes of Nurtec, for example.
Migraine.
Thank you. But also for our pipeline of oncology drugs, both internally discovered and developed, which are now drugs like our CDK4 for breast cancer, which is a successor to Ibrance, which is now in first-line metastatic breast cancer. So that should be approved 2028 or so. But also our Seagen-acquired portfolio of novel ADCs in non-small cell lung cancer. And these are now in phase 3, again in front line. So look, it's going to take time, but 12 months goes by relatively fast, and then that timeline shrinks. I think the third thing, aside from accelerating performance of in-market drugs and then the oncology pipeline, is BD. And after Seagen, Dave, our CFO, has been very clear. Our primary goal is to deleverage. Albert recently shared that we're now back in the BD business. We're looking at opportunities. I think Dave quoted 10 to 15 billion. So this is important in terms of thinking about how we augment our near-term growth rate and build critical mass in those therapeutic areas where we want to scale.
Pfizer's bought a lot of things over the years that did not pay off, quite frankly. I could honestly name a litany of deals going back 20 years. There have been a few that have, but how do you keep that from happening again? How do you make sure that you buy things that actually will pay off and not the gene therapies you just mentioned or the Array Pharmaceuticals chemistry, which it sounds like is being spun off or has been spun off or closed down again, or any of those examples going back?
Yeah, I mean, look, I could point to the BioNTech collaboration in COVID.
As a fantastic collaboration, yeah.
It's a fantastic collaboration. So I think it's everything is a balance, right? I wasn't here at Pfizer when some of those decisions were made. I think one thing I would point to is, number one, I'm here, and as, if not more importantly, Chris, my counterpart, who is CSO and president of R&D, is very, very aligned on the importance not just of delivering cures for patients, but creating shareholder value, delivering drugs that translate into revenues. And so we partner very, very closely, and our organizations partner, to make sure that even very, very on in development, the trials that are being initiated, the design of the trials is very much viewed from, are we going to be able to get access? Will this translate to reimbursement? As well as, is it addressing an unmet medical need, and is it clinically differentiated? And I think that is a change from what went on before, where I think there was, in part, we've got this really cool scientific hypothesis. Let's just bring a drug to market without actually thinking about how we're going to get access and how we're going to sell it, given the competition.
Are there any questions out there? Okay, so I was debating which of these two things to ask, so maybe we can get to both of them. But one is, you mentioned one of the great Pfizer drugs, which is Ibrance for breast cancer, CDK4/6 inhibitor. This is a very good drug in adjuvant breast cancer, and Pfizer has a CDK4 that is one of the things you mentioned to me earlier as being a potential. How do you think that competes, though, in a world where there are there are now new CDK4/6 inhibitors, which are taking a lot of share from Ibrance, how confident are you in this molecule, and how can?
So we obviously have to run a head-to-head trial against investigator's choice, which is exactly what we're doing in the first-line setting, so that trial is open. What we know about the CDK4 is it is active in patients who are refractory to both Ibrance and other second-generation models like Kisqali. And we also know that it has a tolerability profile, particularly in relation to neutropenia, which is, as well as the GI effects and CV effects, which is superior to the two other molecules which are there. So on both efficacy as well as on safety, it looks to be differentiated. We're going to see how that pans out in terms of PFS, and watch this space.
You started with a discussion of one gene therapy for hemophilia as one of the mistakes. We've spoken a lot today, actually. But we've spoken a lot today about all the reasons gene therapy hasn't turned out the way a lot of people thought it would. Do you think gene therapy becomes investable for large pharmaceutical companies again at some point, or is this a niche area at this point?
I think in the right indication, where there is an absence of alternatives, for the right company who gets their hands around reimbursement and can work with payers, because obviously the price of these therapies is very high. I think there's a business model there that makes sense. It's definitely not in hemophilia, right? I mean, that's very clear.
Right.
But in other areas with life-threatening unmet medical disease, for example, you look at Zolgensma, right, which has been, intrathecal has taken time because the regulators have put a brake on it, but it's been, in infants, it's been commercially successful. I don't know what they're doing, 2 billion, something around that, or they were.
So I asked you what the best thing about being at Pfizer these past 7 months has been. What's been the worst?
Well, one of the things I really used to value about being an analyst is I used to have a lot of control over my agenda and ability to think deeply about things and to deprioritize meetings that I didn't want to. And one of the learnings of the transition to industry is there are a lot of meetings.
A lot of meetings?
A lot of meetings.
And I imagine if Albert wants you somewhere, you're there.
It's difficult to say no to Albert.
Yeah. Well, I mean, as you proved by being here.
It's true. It's a good point.
All right. Well, thank you very much, Andrew.
My pleasure, Matt. It's been a pleasure.
For this sponsored conversation, please welcome to the stage Evidation CEO Leslie Oley Wilberforce, and to moderate this sponsored conversation, Stat Managing Editor Gideon Gill.
Well, I'm really thrilled to have the chance to interview Leslie, the science gal. Leslie,
It's hard speaking before Bill Nye.
there are many companies collecting real-world data from patients for clinical research, but one of your company's, Evidation's, differentiators, as you put it in a recent interview, is that you have 5 million users you can tap to participate in studies for your clients. How did you get so many folks to join your platform, and what's in it for them?
Yeah, great question. So I'd say we bet on 2 really big things early on in the company. The first thing that we bet on is having a direct connection with individuals and their permissioned health data that they can share in really trusted and transparent ways. Could help to understand health in really different ways, and it could help to accelerate the pace of research. And so what that looks like in a practical sense is people can join, like anyone over 18 can join on the App Store or Google Play Store. They can connect wearables that they use to track their health, anything from a Fitbit or an Apple Watch to a Dexcom continuous glucose meter. And as they participate, they can take surveys, validated e-pros, submit genetic tests. And so that's one of the modes of participation of people on the platform. The second big thing that we really bet on early on in the company is that EHR data, claims data that really 10 years ago was kind of locked in the system would become more portable over time. And so it's really interesting to be sort of 10 years or plus into the course of the company, where now a user can actually mediate that data and choose where it's shared as well. So it allows you to create these really interesting data sets that are like multi-omic in nature that you can use to really measure and understand health in different ways. And so from a user perspective, couple reasons why people join the platform. One is because of that data, we can share really interesting insights back with them about their health that they might not get in other places, different correlations between sleep and mood and a variety of things. The second reason is we connect people with ways that they can use their data to participate in research. So we can join people to other research opportunities. And so we find that for some people that can be a big activator. The third thing that we do that's more extrinsic value is we actually reward people for their participation. So we actually compensate people for their data. Their data is valuable, and they should receive compensation from that as well.
What kind of compensation?
So people earn points for doing health-related activities, for submitting different types of data, and they can cash that out for money, a donation to a health charity they might care about, gift cards, a number of vehicles. But we find that gamification and really like recognition that their data is valuable and they're contributing to something actually means a lot.
So can you talk a little bit about what kind of data you're collecting and how you're using that data, and give us maybe some examples of studies you've been involved in?
Yeah, one great use case that's really relevant to some of the conversation today, even how we kicked off the day, is in the GLP-1 space. And so last summer we started a cohort that's now about 140,000 individuals in a weight management program. And the really interesting thing is, like, when you have a connection with 5 million people at a really like broad level, we enrolled the initial cohort of about 100,000 people in like 2 weeks, like very, very rapidly. You can get people involved and engaged and really primed for research to understand why it matters and how they can easily participate from anywhere. That helps from a representation and participation too. And within that cohort, we have about 10,000 people who are on GLP-1 drugs. And the really interesting thing, it even came up this morning, is that to understand a lot of the user journey around medication usage for weight management, a lot of that data isn't in traditional real-world data sets. It's not always in claims and EHR data, because many people are going to Roe. Many people are going to Hims and Hers. People are seeing telehealth providers. And so you can get some of that data by just asking people. And then when you start layering in things like wearable data or genetic testing, you can see really fascinating correlations. Like when someone switches a GLP-1, is the weight bounce back curve different between different types of it is actually between different types of GLP-1s. What does layering high frequency like PHQ-9 or GAD-7 scores for anxiety or depression look like when someone is before therapy, on therapy, and after they've discontinued therapy? And so you're seeing these like really interesting patterns of really important secondary endpoints that matter disproportionately in people's everyday life sort of shine through in that data and have the voice shine through that you don't see really in traditional data sources.
And we talked a little bit earlier about there's a lot of barriers to people participating in research, and you talked a little bit about the gamification and what you do to help encourage people to participate. But one of the challenges also that is diversity in clinical trials, and I wonder if you can talk about how successful this has been for that.
It's been a big aim of ours. I think, even in the conversations today, you can probably all tell it's like more important than ever right now. So we've really tried to build a technology and way that people can access the population, that they can do it from anywhere. You don't have to live near a big medical center to participate. People can really contribute to research from anywhere. We're in 97% of all zip codes in the United States, so even from just a geographic distribution, we've worked hard to make sure we're reaching people in all areas of the country. I think one of the things that even when we started the company, we started doing research on why people don't participate in studies. And there's like many historical reasons, there's many reasons for hesitancy in research. And so part of what we can do is help educate people about research, help educate people around scientifically valid things that they can learn on the platform. But another really interesting stat from back then when we started doing that research is when you ask people, like, why have you never participated in a clinical study? Can anyone guess the number one reason? Literally, nobody ever asked me. You're right on the money. Like, no one ever asked me. And so I think that's part of where we can help fill some gaps, is like in something fast moving like GLP-1s, like understanding how the system is evolving and how patient choice is evolving, and really in research, like how you can match people and surface research opportunities when they've never been asked before.
Interesting. So you've been with Evidation now for about 10 years, I think.
Yeah.
Don't want to age you.
Yeah.
So what's been your best moment in that time?
Yeah, you know, it's funny, like sometimes the simple things are what really sticks with you. And the way I'd answer this is, there was this one moment that I was actually reading App Store reviews, and a patient had commented, like, participating in this research study actually saved my life. And the reason was, and I think this is a really powerful thing about research, is it's not just like the data set that's generated on the other end. It's like if you can deliver value to someone while they're in that experience. And so we were the technology partner for a large-scale cardiovascular research study with Apple and Johnson & Johnson. And one of the aims of that study was to take people over 65 who had never been diagnosed with a cardiac arrhythmia, randomize them to receiving a watch, an engagement program on an app, and an alert flow. So if the watch went off, the Series 4 watch and beyond have had regular rhythm notifications. It actually walked them through what to do to go see their doctor. It rolled up an information sheet to have a much more productive conversation with their doctor. And then it followed them to see if they got on a DOAC afterwards. And so we had this really interesting data set of wearable data, claims data, survey and validated survey data over time to understand outcomes. But what actually ended up happening is we had many, many people write in and say, I would have never even, wouldn't have had an Apple Watch. I would have never even thought to think about having a cardiac arrhythmia. I had no risk factors. And so I think the magic in some of this work is if you can find things that are the intersection of great for individuals and great for the healthcare system. Like those people who discovered they had a cardiac arrhythmia early, like probably avoided a stroke down the line in some cases. And so I think that's the power of, connecting directly with individuals through research.
Well, thank you very much, Leslie.
Yeah, wonderful. Thanks so much.
Appreciate it. Thank you, everyone.
Please welcome to the stage science educator Bill Nye, National Ataxia Foundation CEO Andrew Rosen, and to moderate, Stat Sharon Begley Science Reporting Fellow Anil Oza.
Hello, everyone. So glad to see you all here. We're here to talk today about ataxia with two people, with Andrew Rosen, who is in charge of the Ataxia Foundation, and Bill Nye, who I think in this room needs hardly any introduction. You have inspired generations of people to go into science, I'm sure. Some of the people in the crowd, myself included. But a couple of years ago, you opened up about something that is a little more personal to you than gravitational waves or states of matter. Can you tell us about—
Which are pretty personal.
Yeah. Can you tell us a little bit about your family's relationship with ataxia?
So my family has spinocerebellar ataxia, SCA, and ours is, I don't want you to be jealous, but it's 27B, SCA 27B. And I can tell you that because 2 years ago, two sets of researchers found the gene repeat. My whole life, we just called it, the guy who apparently brought it to North America, the last name was Darby. And so my family always referred to this condition as the Darby glide. You're gliding around, falling on your face, smashing stuff. And so my father had it pretty strongly, his older brother had it, and my grandmother had it, and so on. So the Darby glide's been in my family for several centuries. But 2 years ago, the repeat was discovered, and so now I felt there was something I could do about it. So I'm raising awareness of ataxia using my D-level celebrityhood to raise awareness of ataxia. And I just emphasize to people who throw this word around, it's from the Greek word to taxi, to move, to walk. You can't taxi. There's no cab waiting for you. So anyway, it's good to see you all. Thanks for including me. Back to you.
Andrew, you work with the Ataxia Society. Can you tell us a little bit about how common this disease is, how common is Bill's family's version of this disease? Like, sort of zoom out for us.
Yeah.
So ataxia is considered a rare disease, and ataxia is a kind of a set of diseases. There's many different types. There are literally several hundred types of hereditary ataxia. That's what Bill's family has. Bill mentioned that he has SCA27B. We are now up to SCA, I think, 51 or 52. They just name them as they discover them. We continually discover more. So this is a rare disease. There's probably, we estimate, somewhere in the 15 to 20,000 in America that has SCA. Again...
But if there were 20,000 people in this room, I mean, you'd notice, crying out loud.
Right. So it's not ultra rare, I think is what Bill is saying. And there's many other types of acquired ataxia. It's a complex set of diseases. But yeah, so and again, they're still identifying more and more as science continues to build the research.
Right. To this point of scientific progress, we were talking earlier about progress both in sort of diagnosing this disease, but also in treating it. Bill, I know that you've been tested for the genetics behind this to see if you had it, as much of your family does. What did that mean to you?
It changed my life. So I just presumed I was going to get it. As a man, there was a family myth that the men got it, the women got it, could pass it on, but men couldn't. That turns out to not be true. I just presumed I was going to get it and so on. But 2 years ago, it was discovered, I guess I finally got tested last year, my repeat's too short. I almost certainly will not have significant symptoms, except I'm crazy. That's still an issue. So, but now having a chance to do something about it, we can evaluate my sister and her kids and then their kids. So these people would be my grandnephew, grandnieces. They'll be able to select, if they want to have kids, they'll be able to select the zygotes, the fused egg, for the gene repeat and not pass it on. It is very reasonable that this next generation, we could eliminate it, at least in the developed world. That's really extraordinary. And the thing I go on all the time about, SCA27B is in the mix of ataxias, but when you do research to find the repeats, you're just learning more about brains and what everybody's running around. I don't know if you guys watch cable news. There's a lot of skin stuff. There's a lot of there's depression, but the other big one is Alzheimer's. Man, that's all the rage. Every other ad is there's some Toyotas and then there's Alzheimer's ads and dementia writ large. That's what everybody's talking about. So the more you learn about brains, the closer one may be to this, to understanding whatever brings these conditions on. You know, the problem in evolution is you only have to be good enough. So if SCA27B doesn't kill you, but you can have kids and they can have kids, well, then that's cool. And the same is true with dementia. If it doesn't kill you, you have kids first, that's all good. But wouldn't it be great if we could eliminate all these things so that people could be more productive? And for you mean-spirited people out there, they can pay taxes longer. Just think of it that way. So it's an exciting time in this research. It's really something that when we got in a study at Johns Hopkins back in the 20th century, back in the 1900s, and people were saying, well, it's like trying to sort out the genetic sequence was like trying to find 1 in 80,000. And that was probably, I guess, pretty close. And so after 30 years of screwing around, 2 independent sets of researchers found the gene repeat for my family. It's really extraordinary.
Let me say one additional thing. Bill's type of ataxia, SCA27B, is a sort of a later onset, particularly slow progressing form of ataxia. And there's others that are much earlier onset and faster, but what that often does is, of course, people aren't diagnosed until their 30s, 40s, 50s, even 60s. And of course, by that time, they've perhaps had a family and unknowingly pass this on, right?
Well, in my family, it's just something that was going to happen.
Right.
It was not an option you were going to avoid. That was just something that's it'll be along. And why did it affect my father more strongly than his older brother? Why does it affect my sister more strongly than my brother? Well, nobody knows, man. But maybe we'll figure that out. So stay tuned.
Yeah. Andrew, tell us a little bit more about the scientific progress. You were telling me that there is some progress in developing things that can help treat this disease. What do those look like? What is the promise there?
Yeah. So it's exciting. I mean, even 10 years ago, I don't think we would have said that there was really pharmaceutical interest in developing treatments for ataxia, and that has changed. There's a number of drugs in the pipeline. The very first treatment for any type of ataxia, Friedreich's ataxia, which is a particularly nasty form, affects kids, and they often don't. They're gone by their mid-20s. But the very first treatment was approved in February of 2023. And 5 months later, that little pharma was bought for $7.3 billion.
Billion.
Billion with a B.
Which, of course, doesn't go as far as it used to.
But one of my messages would be, hey, don't forget about us in the ataxia space. So ataxia is a movement disorder, right? Think Parkinson's is kind of the one that everyone knows, but in the same field of ALS and Huntington's, maybe just not quite as well known. And we're obviously thrilled about our work with Bill because his ability to get the message out is exciting for us.
So you guys, on one day, we were in Susan Collins's office, or in a meeting with her, and then some progressive people. We didn't meet with Adam Schiff that day.
No.
Right, but we could have. And then Tom Cotton. I mean, these are people that don't get along about freaking anything. But when it comes to medical research, they are generally like-minded. But there has been, I will say, an odd choice for head of Health and Human Services lately.
Yeah. I mean, let's talk more about
Let's
that.
go with odd.
Well, now that you brought that up, Bill.
Well, he's a guy who doesn't believe in vaccines. Dude, pasteurized milk. I mean, come on. This is a couple centuries old. I went to elementary school with a guy who had polio. You don't want polio. Polio bad. And that there's a guy who doesn't understand that is really something. It's just, it used to be the United States. Those were the days. Yes.
Yeah.
Back to you.
Yeah. I mean, Andrew, what are you hearing from the researchers that the Ataxia Foundation partners with that are doing some of these things? Because all this hope is well and good, but as we've heard throughout the day, the sort of scientific process is not, what it was even 6 months ago.
We're very concerned.
Very
there is
concerned.
no doubt.
Let's go with that.
That our research community is under attack. We give away a couple $1 million a year in research grants, and we're proud of that. That's obviously a drop in the bucket compared to what the federal government can do. There's a large program.
But $1 million, man, if you're looking at any sort of genetic analysis, that's a chunk of change. If you're trying to make this mythic thing, the mouse model, that's a chunk of change. Yeah.
And you'd be hard pressed to find an ataxia researcher in the world that hasn't received a grant from NAF over the years, right? We've been around a long time. We're proud of that. But we can't match what's happening at a federal level. And programs are under attack. We hear it from the large academic research centers that you all know are under attack. And we are very concerned about what that's going to do to, frankly, the progress. I mean, we're excited. We're accelerating here. And we just are horrified to see that slow down.
So you guys, I have a day job. I'm the head of the Planetary Society. This is a 40,000 people around the world who just think space exploration is cool, and we're not into Earth science. We want to explore planets: Mars, Europa, Titan, moon of Saturn, Europa, moon of Jupiter, where there might be signs of life. All right. It was started by Carl Sagan, this famous astronomer many years ago, and Bruce Murray. And you may not have heard of Bruce Murray, but he was the head of the Jet Propulsion Lab, JPL, during the Viking missions landing on Mars and Voyager, the spacecraft that are still flying, going way out. And people would say to him, why do you want to build these spacecraft? Why do you want to send them out into the solar system? What are you going to find? And his thing was, we don't know what we're going to find. That's why we're sending them, Mr. Congressperson. And so that we have a situation where our own government doesn't understand or appreciate the value of fundamental research is very troubling. So we're in there advocating. Lobbying's a technical term, legal term rather. We're advocating for investment in medical research writ large, but especially into ataxia, because it is this identifiable set of symptoms, and it affects minor celebrity Bill's family. No, really. So that's why we're doing
Bill, I
it.
think you're B-list.
B-list. Woohoo!
I think we have a question.
List. We have a
No,
question
it's
from
a
the audience.
B-list celebrity. I can go into like Starbucks, and I can get a cup of coffee for $6. Whereas you people, I don't know what it costs you. I can't even think about it. Yes.
Hi, my name is Stephanie Henry. I'm a neuroscientist and freelance writer. My question is in regards to the pharmaceutical interest in ataxia drugs. So you said that's kind of new, maybe about 10 years ago. What stimulated that? Is it because of our A-list celebrity, Bill Nye? And if so, do we need champions for all of the other rare disorders? I don't imagine that this subtype of ataxia has become any less rare. So what are the factors?
It's a great question. I'd say a couple things. So, there is legislation that supports development of treatments for rare diseases. The Orphan Drug Act is probably a perfect example of that. I think the other thing that is added to that is the years of basic scientific research that's been done in the ataxias, partly because of organizations like mine and others that have funded those efforts, have led to the point of understanding kind of the basic mechanism and can then move to translational science and to the clinic, et cetera. So I think those are probably the two key reasons. And yeah, there is certainly a lot more interest now. The challenge, I think, for us at a regulatory level is helping the regulators understand that rare diseases need to be looked at differently than more common diseases. Our patient community simply cannot wait, and frankly, there aren't enough of them to do the classic, double-blind, placebo-controlled trials. And so they use things like natural history data that NAF, we sponsor the largest natural history study in the ataxias. They use things like that, and we need to continue to help educate our regulators about that, and that will help speed the development of these treatments along.
Yeah, you can't, double-blind thing takes 10 years sometimes. And my sister, like, I don't want to wait 10 years.
Right.
I'll try it. If it doesn't do anything, okay, I'm no worse off. But if it could, especially the big thing now is perhaps halting or virtually stopping the progression, because SCA27B, like many of these things, and Friedrich's, just gets worse and worse as you get older. Yes, and help. Yes. Oh, yeah.
Question?
Hi, Bill Nye. My name is Catherine Ames. I'm a young patient advocate, and a question I have is relating to caregiving. A lot of the times, people like my mom aren't necessarily recognized for the burden they bear.
What's your mom's deal?
She takes care of me.
Oh, and what do you have ataxia?
I don't have ataxia. I have, well, technically I'm undiagnosed, but for the purposes of insurance, my ICD code is systemic lupus.
Oh, wow.
And so, you're in the rare disease space. You caregive for someone with rare disease. You have a foundation to support you in that way. I'm wondering if you can marry that conversation with the brain drain that's happening in scientific research and where the undiagnosed population is going to fit into that conversation. And you used to call ataxia, what was it?
The Darby glide.
The Darby
We still
glide.
do,
You
yes.
still do. So I'm wondering if you can just speak to some of us young patients who are undiagnosed, and is there hope for us in this time of healthcare turmoil?
Well, I think the pendulum's going to swing, right? I mean, there's only so much incompetence we're going to put up with. No, really, it's really, no, I mean, you guys, there's only, like that guy Weldon didn't get the Centers for Disease Control. And I'm not changing the subject. We are talking about you. But this guy is so nutty that even our Congress couldn't hire him. And so people like you, I think, are going to get together, starting, I imagine, on social media, and then we'll get somebody like you to come with us to Congress, and we'll form, I'm making this up here on stage, having thought this through completely, we'll have a so-called rare disease advocacy day of action, so-called. I can imagine that easily. And so please stay in touch with Andrew at the National Ataxia Foundation, and let's see if we can get a coalition of rare diseases. And so we were in Tom Cotton's office. You know, if there's 180,000 people, I mean, that's a lot of people that could have a voice. You know, so I used to work with somebody, a Disney executive, had lupus. And then one of the guys, the editors on the Science Guy show's daughter had lupus. But it was diagnosed. Yours is a special something or other. Yeah. Yeah, ataxia is a catch-all, by analogy.
Yeah, but insurance won't process your claim unless you have, you know, a 5 or 6 digit number associated with
Yeah.
these. They
Yes.
know how to categorize and therefore sort of family tree all your potential treatments. But you can't submit to insurance for undiagnosed.
So are you involved in so-called natural history study? Because you just used the word.
What does that mean, Bill?
Oh, sorry. You just used. Yeah, Tyrannosaurus and yes, and the American Museum and who is running around? Who's the guy in the movie? No, Jeff Goldblum. That's the other movie. Night at the Museum. Who's that?
Ben Stiller.
Yeah. Thank you, Ben Stiller. Yes. Anyway.
Wow, we have digressed, Bill.
Just a little bit. So natural history study, everybody. This is the kind of thing we go into Congress to explain when I'm with him, is if you have a large patient population, you can sample everybody's DNA and take a meeting and I presume throw in AI and they'll, oh, wow, cool. And then you can correlate effectiveness of a treatment, of a drug, of a protocol. But when there's so few of you, or writ large us, I mean, I'm not in your league, but then you have to look at how your symptoms progressed with time.
Naturally.
The natural history of you. And then by correlating or keeping track of everybody's natural history, everybody in a family, literal family, hereditary people, then you can get data that are every bit as useful or effective or reasonable or scientifically robust as traditional double-blind running around 10-year-long longitudinal studies. So did I make natural history clear? It's how your symptoms progress can be tracked and then correlated with your family, people with the same genes, and we'll see what is what. And then you can also, people like you, in relatively small patient population, you can try a drug, like we'll get mice to show that it doesn't kill you very much, and then you'll try it. And instead of running around, well, we have to double blind, red dye number 3, and God, who is this freaking guy? It's just spooky. So thank you. Although you don't have ataxia, stay in touch with us, and let's see if there's some small patient. You know what I'm doing? I'm thinking out of the box. And if you're a young person, I don't know how old you are, what would I be doing? Yes, I'd be spitballing. That's what the kids do, spitballing.
And with
yes.
one more question
I want to get to, yeah.
Yeah.
So.
Hi, Julie Eagle. I'm an advisor to PPMD, Parent Project Muscular Dystrophy.
Oh, yes.
And yay.
So you guys, I got enough people in our muscular dystrophy carnival in Washington, D.C. I saw the Beatles live. Okay, check me out. I look like nobody. I was 9 years old. Yes, back to you. Hold the mic just a little closer to you. Yeah.
Sure. Quick question. We heard a little bit earlier from somebody who described how CRISPR is being used potentially in treating genetic diseases. When I talk to potential donors and patients and families, one of the questions that they always ask, especially if they have the means to make a significant donation, is what's the trade-off for me personally? You've described the sense of urgency that your sister has or that you have. How do you trade off getting some bespoke treatment for yourself and contributing to the science and the progress toward a treatment longer term that's more durable?
What do you mean, like getting a drug for my family?
Like getting a bespoke treatment, like a CRISPR-based therapy.
Oh, a CRISPR-based therapy.
If you have the funds to do that.
Oh, I see.
Right.
Well, we want a day where it's not so crazy expensive. I mean, and to throw everybody's favorite acronym around, this is where artificial intelligence, I think, is going to have a huge effect, where you'll be able to sample somebody's DNA and then customize a treatment in regular time. It's not, I'm claiming the following thing is analogous, not the same. You guys may remember this company Monsanto. It's been absorbed. Well, they sequence corn genes in 5 minutes because they got a sequencing machine and the buffering agents all dialed in. So it is very reasonable to me that you'll be able to sequence human genes very quickly and then customize a treatment at a reasonable cost. So, you know, God, it used to be a thing you could talk about, but electric vehicle manufacturers marketed to rich people first because they could screw around with it, you know, and so that sort of market-based investment leads to cheaper cars for everybody. So I don't think these things are incompatible, but I think advocacy is a big part of it. Getting appropriators, especially if we're in the US, in Congress, to correlate these things so that investment in a bespoke treatment becomes an investment for the greater good. I mean, am I hand-waving? But muscular dystrophy has been, in my family, confused with ataxia.
Often.
Yeah, very often, because the symptoms are very similar. I mean, I don't know how to tell them apart. So, in muscular dystrophy, do you guys have the saccades, where your eyes jump around? Yeah, that's in ataxia, that's a big thing. Your eyes do this. Anyway.
I think we are just out of time.
Neil, I want to say one more thing.
Oh, man.
Just in 30 seconds. Bill is wearing the small version of his Presidential Medal of Freedom that you may know he got. Bill.
Thank you. Thank you.
You got that because you are a man of science and you want to educate the world on science. And
That's
I
it.
don't know if there's ever been a time where we need that more. So keep up the good work.
So you guys, Article 1, Section 8, Clause 8 of the U.S. Constitution. Article 1, role of Congress. Do we still have Congress? Is that still in business? And then Section 8 is kind of the miscellany, letters of marque for pirates on the high seas. The post office is in the Constitution. Okay, we're not privatizing the post office. And then Section 8, Clause 8, they call it, is Congress shall promote the progress of science and useful arts. The word science is in the U.S. Constitution. And I have been in congressional offices, senatorial offices, where the staff and the guy or gal doesn't even know that that's in there. It's way down. It's on page 6. You know, you guys, science rules. Thank you all very much. Thank you. Way to go.
Great place to end
man.
it.
Big red.
Of course.
everyone. It is time for our last session of the day, which I promise is a very, very fun one. For those of you who do not know, Stat Madness is our annual bracket-style tournament in which we have universities and research institutions from all across the country compete to be crowned the winner with the top medical innovation of the year. This takes place on our website, but we have brought 4 of those teams from this year's lineup to the summit today, and they are going to pitch you on their work. You can kind of think of it like science's version of Shark Tank. And at the end of the presentations, you are going to be asked to vote for your favorite team, and that winner will be named this year's Stat Madness Summit Audience Pick. So here to kick things off, we do have a word from our Stat Madness sponsor. Please welcome to the stage Tyler Alchin, Managing Director of Healthcare at Jobs, Ohio.
Good afternoon, and it's a pleasure to be with you here at Stat Breakthrough Summit East and for Jobs Ohio to be the sponsor of Stat Madness. My name's Tyler Alchin, and I lead the healthcare and life sciences practice at Jobs Ohio, the state of Ohio's private economic development nonprofit corporation. Unfortunately for me in this job, Ohio is the best kept secret in life sciences. Even this audience, well informed as you are, may not know that we lead the nation in pediatric care and research. Led by #1 ranked Cincinnati Children's Hospital that garners much of the headlines. Just up the road in Columbus is Nationwide Children's Hospital, which has been a pioneer in gene therapy, having been responsible for 2 of the first 9 direct gene therapies approved by the FDA. Most recently, their work has come out of their lab and been furthered by Sarepta Therapeutics and approved by the FDA for the treatment of Duchenne's muscular dystrophy. The Sarepta story is an important one for Ohio and one we're very proud of, in that instead of pulling IP from a Nationwide Children's Hospital spin-out, Myonexus, that they acquired in Columbus, back to the coast, a model we're very familiar with in the Midwest, they decided to bet on and invest in the teams and partners in Greater Columbus. Fast forward a couple of years, they now have over 200 scientists and staff at a center of excellence for gene therapy in Columbus, their largest R&D center in the globe. If you weren't paying very close attention, you may not know that this leader in gene therapy and rare disease was placing their bets in Ohio, in the Midwest, and the people and talent coming out of our institutions and partners. But Sarepta is not the only corporation that's doing so, not the only life sciences leader. Amgen in 2021 announced their intention to build an aseptic fill finish, a state-of-the-art facility in New Albany, Ohio. 3 years later, it's the fastest facility for launch in the 50-year history of the company. Both the Sarepta story and the Amgen attraction win build upon long-standing relationships we have with global leaders like Abbott Labs, McKesson, Cardinal Health, and others, proving that Ohio wraps its arms around our life sciences innovators, is a catalyst for commercialization, and works and invests at the speed necessary to bring medicines to patients. So, with all this history of providing talent and infrastructure and support for leaders in life sciences, how are we still flying under the radar? With the #2 hospital system in the world at the Cleveland Clinic and The Ohio State University as part of our multi-billion-dollar research enterprise, how are we still relatively unknown? Our Midwest modesty is unfortunately to blame for this story. It's betraying the fact that now we are an ideal location for the innovators like those participating in Stat Madness with you this afternoon and evening. The Ohio Discovery Corridor, which we announced last year, is our effort to bring awareness to the research enterprises, staffing at CROs, CDMOs, as well as our clinical trials and preclinical trial networks across the state. The Ohio Discovery Corridor, all located within a 3.5-hour drive from Cleveland, Columbus, and Cincinnati, brings all of this value together in one single point of access. So whether you're needing to scale an R&D team, enroll a phase 2 clinical trial, or build and distribute your therapeutic for commercial launch, Ohio brings the collective strength of our network to your benefit. Now, we'd love you to take a look at Ohio Discovery Corridor online and follow us on LinkedIn. We believe that the Ohio Discovery Corridor is the future of statewide life sciences innovation ecosystems. Team Ohio will next be at Bio International in Boston in June in full force and ready to support your next endeavor on demand. It's been an honor to share the stage with the future of medicine today and this afternoon, and we look forward to showing you a little bit more about the people and the places that make Ohio Innovation the special place. So thank you so much, and on behalf of Jobs Ohio and the Ohio Discovery Corridor, best of luck to those competing this afternoon. Thanks so much.
Dive into this year's Stat Madness session. So as I mentioned, you will be able to vote for your favorite team following the presentations. If you have not already, please do use the QR code to download the Bizibo app and sign in, because that is where you are going to cast your vote. That code is on the back of your name tag. And to help you decide, we have pulled together an all-star panel of expert judges, and they are going to provide feedback on every pitch. But remember that it is you, the audience, who gets to pick the winner. So please join me in welcoming, from our Stat Madness presenting sponsor, Tyler Alchin, Managing Director of Healthcare at Jobs Ohio. From our Stat Madness supporting sponsor, Seema Kumar, Chief Executive Officer at Cure, Lonnie Levin, Professor of Pharmacology, Weill Cornell Medicine, and last year's 2024 Stat Madness Summit audience pick. And of course, Stat's very own Matt Herper, Senior Writer for Medicine and Editorial Director of Events at Stat. Thank you all for joining us today as judges. To start, we are going to have each of you sort of briefly introduce yourselves to the audience so that they know a little bit more about how you are. Tyler, would you start? And then we'll go down the line.
Sure, thank you so much. Again, Tyler Alchin, Managing Director of Healthcare and Life Sciences at Jobs Ohio, lobbyist for healthcare industry by background, and have been in economic development since, so it's a privilege to sit through these pitches today. Thanks.
Hi, everybody. I'm Seema Kumar. I'm the CEO of Cure, this beautiful facility that we're having the summit at. And we have many, many, many startup companies right here in this building, as well as companies that started with as a startup and then became public as recently as last year. So eager to be here, have worked at J&J and Johns Hopkins and the Broad Institute, so looking forward to judging.
Don't you all know? I'm Matt Herper. I'm the editorial director of events here at Stat and a senior writer for medicine. I've been writing about biotech and medicine for 25 years. Some of you who I quoted early in my career might be shocked by that number.
Lonnie Levin. I'm a professor at Weill Cornell Medicine, and I have technology that was fortunate enough to win the Stat Madness last year, so I also have a small company, biotech company, to commercialize that.
Thank you all. We're really thrilled to have you on our judges panel. With that, we're going to dive right in and get started. So I would like to welcome our first Stat Madness presenter to the stage, representing the Deep Sedation Disparities team. Please welcome Mary Armstrong Huff, Associate Professor at the NYU School of Global Public Health.
Thank you. Does anyone know how to make my slides come up? There we go. Thank you. I'm speaking today on behalf of a multidisciplinary team that is working to understand and intervene on risk factors for survival of respiratory failure. I am a medical sociologist and epidemiologist at NYU. My co-PI for this work is Tom Valley. He is a critical care physician and a health services researcher at the University of Michigan. And I'm going to tell you about a little study that we published earlier this year with some help from our friends, a few more colleagues with expertise in biostatistics, social sciences, and critical care delivery. Sorry, I've lost my slides. Oh, there's one. That's good. All right, I'm going to tell them to advance. So the first thing that you need to know about respiratory failure is that it is common and it is deadly. Can you advance? Advance again. About 750,000 Americans die of respiratory failure every year. Advance again. And nearly half of patients with the most severe form of respiratory failure, acute respiratory distress syndrome, or ARDS, will die within the first 3 months. The goal of my research with Tom is to try to get this number lower. And in order to do that, we need to understand which respiratory failure patients are at the highest risk of death and how they got there. Now, we know some things. Patient clinical characteristics like age, sex, severity, and type of illness, all of these things can influence the risk of death for a respiratory failure patient. Can you advance again? But that risk is not evenly distributed. And advance again, please. We know, for example, that Hispanic patients have twice the risk-adjusted odds of death from respiratory failure compared to other respiratory failure patients. Advance again. What we don't know is why. We've actually known that Hispanic patients have twice the risk-adjusted odds of death for over 10 years. And no one has ever been able to identify a mechanism that might explain it. We just knew that Hispanic patients died or had twice the likelihood of death compared to non-Hispanic patients. Our team thought that maybe we should do something about that. And we had an idea. We thought that Hispanic patients might be more likely to receive a low-value practice in the ICU, deep sedation. Now, ARDS patients typically receive mechanical ventilation and sedatives. But the level of sedation can have important implications for patient outcomes. Light sedation can help patients be more comfortable, more calm, and safer while on mechanical ventilation. But deep sedation is harmful. Please advance. Deep sedation is associated with a range of harmful outcomes. Advance. More time on ventilator. Advance. More time spent in the ICU. Advance. Higher risk of delirium, higher risk of long-term disability, and last one, the big one, higher risk of death. For all these reasons, the major critical care societies all recommend against the use of deep sedation for most patients. But we thought that it was possible that patients who are Hispanic might be exposed to deep sedation at higher rates than other patients, because of things like language differences and clinician perceptions of patient agitation. So we went about testing the hypothesis that Hispanic patients would have higher risk of exposure to deep sedation compared to non-Hispanic patients. And in order to do that, we took longitudinal data from patients who were enrolled in the control arm of a randomized controlled trial that took place at 48 hospitals spread all around the United States. We measured their exposure to deep sedation over a 5-day period where they were alive and mechanically ventilated, and we collected time-varying data on their clinical characteristics, including their severity of illness. We fit a mixed effects model to adjust for those patient clinical characteristics and to account for clustering at the level of the hospital, because we reasoned that it might be that some ICUs tend to sedate their patients more deeply than other ICUs. And we reasoned it might be possible that Hispanic serving institutions tended to sedate all their patients more deeply. But we wanted to partition the hospital level effects from the effects that were happening from the interaction between the clinician and the patient. Next slide, please. So this is what we found. Next slide. In our study, Hispanic patients had 5 times the odds of exposure to deep sedation compared to non-Hispanic patients. Next slide. This is adjusting for patient demographics, for time-varying severity of illness, and accounting for clustering at the hospital level. Next slide. This is equivalent to a 19 percentage point increase in the probability of deep sedation just for being Hispanic. So in other words, let's think about two clinically equivalent patients. If we look at one and based on their SOFA score, their age, their sex, their BMI, which day it is in the ICU, how long they've been vented, we guess that on this day they have a 60% probability of deep sedation. But then you blink and say that patient is Hispanic. Actually, that's about an 80% probability of deep sedation for that patient. Next. So we found that Hispanic patients have notably higher risk of exposure to a low-value practice in the ICU. Next slide. This is the first plausible mechanism that might explain persistently higher mortality rates among Hispanic patients with respiratory failure. But there's good news. This is actionable. Clinical guidelines already recommend light sedation targets. All we have to do to fix this is de-implement a practice that we have recognized for nearly 20 years to be of low value. To me, that makes this a really optimistic finding, because it's not often that you find out that you might be able to save lives by not doing something. Next slide, please. I just want to thank the many co-investigators on this at Michigan, OHSU, NYU, Johns Hopkins, and Columbia. Next slide. And also acknowledge our funders. This work was supported by an R01 from NHLBI, for which Tom Valley and I are co-PIs. Thank you, and I look forward to your questions.
Thank you so much, Mary. I'm going to turn it over to our judges. Does anyone have a question they would like to ask?
Yeah, I have two questions. Did you investigate or look into or have a hypothesis about something else that could be going on? Number one. Number two, so this was all U.S. hospitals.
Yes, all U.S. hospitals.
Did you compare it with hospitals outside of the United States, especially where there is a, equivalent Hispanic population?
So the equivalent Hispanic population, there's not really a place that has an equivalent Hispanic population outside of the United States where we would have a comparative population. So there's a lot of really great work that is done in Canada that looks at really granular clinical data on what care processes are delivered in the ICU and then what the long-term outcomes are for patients. But there are not large enough groups of Hispanic identified patients in those data sets. In fact, after doing this work, we looked around for lots of data sets, and we have found out that we just have to run our own trial if we're going to get sufficient numbers of Hispanic patients to make the next link in the chain, which is to mortality. Thank you.
Would it require a randomized trial? You said this is plausible. Would it require a randomized trial to prove this, where you took an intervention where I guess you told doctors not to? Maybe a cluster randomization? It sounds like a hard study.
It does sound like a hard study. The study section thought so too. They said, really, you want to randomize 20 ICUs? And we're saying, yeah, we can do 12. We can do 12, right? So two answers to that. First of all, we can actually do causal mediation analysis on observational data. We don't have observational data from other trials that have happened where we can do a secondary data analysis of the control arm like we did here. But increasingly, there are other sources of data like Truveta. I know Epic has a product as well where there are sort of harmonized measures from EHRs. And so we've actually been able to identify more than 500,000 people who experienced ARDS in the Truveta data set. And we've proposed that as a next step. So that would be observational. It would not be a trial. It would not be that expensive. We would essentially do something that would be like a target emulation trial with observational data. But I think that if we really want to move clinicians on this, we do need to have a cluster randomized trial that shows that units that greatly reduce the reliance on deep sedation actually have better long-term survival and hopefully better other patient-centered outcomes, right? If you have less ICU delirium, if you have less long-term functional disability, then you're going to have happier, healthier survivors of respiratory failure as well. And I do think that we would need a stepped wedge cluster randomized trial to show that.
We're almost at time, but we could do one
So you didn't really talk about why, but in your paper, I think you hypothesized something about communication and a lack of communication. Were you able to parse in your data if it was also true in hospitals that supplied people to help communicate and translators, or if there was a subpopulation that didn't have an English-speaking family to advocate for them?
Fantastic question. So unfortunately, they did not collect patient language in this trial, and they didn't collect proxy language either. And we tried to look at it through various creative ways, but we weren't able to do it. Every one of these hospitals actually did have a medical interpretation service. Whether or not it was used, I think, is quite questionable. We have gone on in this research to do a lot of actually direct face-to-face frontline work in ICUs to understand how sedation is delivered. And one of the things that we have seen is that a lot of it is about nurse staffing. And it may be that the nurse who is known to be bilingual, she gets like overstaffed, or she gets overburdened with patients compared to other nurses. If you have worse nurse staffing, there are many incentives to deeply sedate, because a patient who is lightly sedated and on mechanical ventilation, if they're moving around a lot and they're not being monitored, then they could self-extubate, right? So as a matter of patient safety, if you have insufficient nurse staffing, you're going to be more likely to be deeply sedating your patients.
We are at time. Mary, thank you so much for this presentation. Really appreciate it. I would like to welcome our next Stat Madness presenter to the stage, representing the Predicting Cancer Outcomes with Big Data team. Please welcome Dr. Justin G., medical oncologist at Memorial Sloan Kettering Cancer Center.
All right, hello, I'm Justin G. I'm a lung cancer specialist and also AI scientist at Memorial Sloan Kettering. So my talk is going to be about using real-world data to predict and hopefully improve cancer outcomes. We all know this slide. We all think about this all the time. This is like the audience that thinks about this stuff, right? This is too slow. This takes many years. If you want to get any kind of new technology to actually impact patient care, you've got to take it through cell lines, organoids, model systems, 3 phases of clinical trial, and then finally you can impact the real world. This is heinously expensive, many years. So really, we need something to get around and expedite this process, as we've been talking about today. And I think, as has been proposed, one potential solution to this is real-world data. We've collected a lot of data from a lot of patients thanks to electronic health records. And the beauty of any finding that you make with real-world data is that your demographics are real-world demographics. Your outcomes are real-world outcomes. So any finding that you make in real-world data has a much higher likelihood of success of actually then working in practice, unlike with mouse models and clinical trials. So the downside to real-world data, of course, is that it's not often created with research in mind. A lot of times you'll have free text notes like this, and as a lung cancer specialist, I have to read notes like this, and if I want to answer scientific questions like what anti-neoplastics are associated with a tumor mutation, I have to identify in this note, okay, yeah, this is a patient who is treated with carboplatin, paclitaxel, and bevacizumab. Usually is abbreviated. There can also be negations. There can be talk about potential hypothetical future treatments, a lot of linguistic complexity. And so that's why traditionally the system to analyze this in order to do analysis has been a person. But that doesn't scale if you have a cohort of 100,000 patients or even millions of patients, millions of records that you have to go through. So my colleagues and I hypothesized that a lot of this could be replaced by AI natural language processing. And we hypothesized this for a couple of reasons. So one is that transformers, we started this before ChatGPT was public, a couple of years before then. But even before there was this public awareness of the power of neural networks, the attention is all you need paper had already been published. There were already some really good NLP algorithms out there in the literature that showed a lot of promise. And the second reason was that our institution had participated in this large, well-funded study to manually curate a large amount of patient data, based on free text notes. And so we had that data, those labels, and we could use that to train and validate natural language processing algorithms. So we did this. We trained a bunch of transformers to annotate things like metastatic sites of disease, treatments, immunohistochemistry, smoking status, other clinical features from free text notes. And then we took those NLP algorithms. You can see they perform pretty well. Those are the precision and recall are pretty good. But actually, when we went back and looked at the quote unquote model mistakes, the false positives and false negatives, a lot of them were actually errors in the manually curated gold standard data set, not NLP errors. So that we think this is really performing quite well. So we deployed these NLP algorithms on all of the patients at our institution with tumor genomic profiling. It's almost 100,000 patients. We combined it with a bunch of other siloed data modalities to create a unified data product called MSK Cord, which can be used for analysis. I'm just going to highlight very quickly 2 examples of how we've used MSK Cord to better improve patient care. So one, as a lung cancer doctor, I'm interested in knowing what genes that are mutated on a tumor might predispose to better or worse survival for my patients. If you wanted to do a systematic analysis of all of the genes that can be mutated in a resource like the Cancer Genome Atlas, this is kind of a small, it's like about 1,000 patients. You actually don't see anything because there's too many genes, there's not enough patients. But if you do this in our large automatically curated MSK Cord data set, you see there's a number of genes that are associated with better or worse survival in non-small cell lung cancer. One of them, SETD2, when it's altered, it's about 3% of lung adenocarcinoma cases actually associated with better overall survival. It's not just prognostic, it's actually predictive of better response to immune checkpoint blockade in particular. And so this is an example of a predictive biomarker that can actually impact patient care that was discovered entirely using real-world data. One more quick example is another complication that our patients face very frequently are blood clots, venous thromboemboli, major source of cancer morbidity and mortality. There are no good mouse models for this condition, but we hypothesized that liquid biopsy data and circulating tumor DNA might be prognostic for VTE. So we tested this in a cohort of several thousand patients at our institution with liquid biopsy sequencing. It turns out, yes, ctDNA, very predictive for VTE, dose-dependent manner, and we actually now have a clinical trial that's about to launch that actually is using this in an interventional prospective setting. So all of this is to say, this is selling not just a specific finding, but a data platform. Come partner with us. We want to expand this to other institutions. We have a lot of, I could go on about other examples of how we've used this, but I won't for the sake of time. But please reach out, partner with us. That's what we want. Thanks.
Thank you, G. Do we have questions from the judges? Lonnie?
So it seems to me the strength is your ability to put the notes into English language, into usable standardized language. Do you think do you think it was easier because you did it at one institution where there's sort of a finite number of MDs and trainees that are learning the same way? I mean, there's sort of famous examples about how there's such different medical care across different communities. If you went to a different hospital, it might not have worked as well because they use totally different language.
Yeah, it's a fantastic question. In this paper, we didn't get into it. We have cross-validated some of these models at other institutions that use even totally different electronic health record systems. We've mostly tested across like us, Dana-Farber, like other cancer centers. So we haven't tested as thoroughly as I'd like in like community settings, for example. Partly because the AI infrastructure is just to pull the notes and run the models is not there yet. But it's definitely something we think a lot about. We have a paper last year that shows that you can do this. We actually even found a way to de-identify the neural network algorithms, because if they're trained on protected health information, you can't share them. But we have versions of these that are HIPAA safe, and so anyone in this room can download them from PhysioNet. You can actually run them on your own hospitals. Let me know how they work.
Justin, I hear a lot about physicians and systems being bombarded with AI tools and sort of not being able to get to applicability or a system to make it work, or even choosing between technologies such as yours. If you sort of dream out on the commercialization front, your thoughts or concerns on that front for the usability of your technology at this stage?
Yeah, I think definitely for the NLP side of things, there are a lot of companies doing this. I think that we've come up with a library that's very particular for cancer, and so I would say that if you're interested in curating data in cancer patients, that's very important. I will say I think that even though there is a lot of very exciting retrospective AI biomarker work, there are very, very few clinical trials actually testing AI biomarkers for concrete medical decisions that need to be made. I think this is a huge hole. I think that we all need to, this VTE trial that I'm showing, like this is our institution's first ever AI trial like this that we've ever tried to launch. And I think that no one is selling that, right? And I think that we need more of those kinds of biomarkers.
So are you using it regularly in clinical practice now, or is it still a technology that is not fully developed?
The NLP is not for clinical practice, it's just for curation. So the VTE finding, like I said, it's a clinical trial, right? It's just past Research Council. It's like being reviewed by IRB right now. I think that there are probably some, like the SETD2 finding, like, if I get a SETD2, patient, am I more likely to say, like, you really should be on immunotherapy? Yeah, probably so. But, I think that, again, really, we need more interventional trials to really, rigorously validate this, and that's just missing. But I think that we're, finally starting to make progress.
With that, we are at time. Thank you so much, Justin. I really appreciate it. I would like to welcome our third Stat Madness presenter to the stage, representing the Is Cancer Metastasis Hereditary team. Please welcome Wenbin Mei, Graduate Fellow at Rockefeller University.
Thank you for the kind introduction. I'm Wenbin from Rockefeller University. Today, I want to share with you a story about if cancer metastasis is actually hereditary. We actually find an example of a human germline mutation that could drive breast cancer metastasis. If you look at the textbook example of how the tumor develops in our body, during the course of human life, our normal cells in our body continues to accumulate mutations because of environmental damage. And then these mutations, when they hit oncogenes and tumor suppressor genes, will cause the transformation from a normal cell to a cancer cell, and then we eventually form a tumor. It has been quite successful for people to identify mutations in the tumor called somatic mutations that can drive the formation of a tumor. But when it comes to metastasis that kills 90% of the cancer patients, the field has actually not found any causal driver mutations that is in the tumor. So the field has been wondering maybe metastasis is a random process. It's purely due to bad luck. Today, I want to view the question a little differently. We want to look at if inheritable factors or germline genetics, human genetics, play a role in this picture. And the mutation I want to talk about today is a mutation that's affecting the gene called PCSK9, very famous in the cardiovascular disease field because of its cholesterol regulatory role. And the mutation, if you look at this mutation, PCSK9, it's common in the population. 70% of the people are affected by this mutation. That means 70% of people in this room will be AA homozygous of this mutation. And what does this mutation do to cancer patients? Today, I want to talk about breast cancer. And in this breast cancer cohorts from US and Netherlands and the UK, we consistently identify that people, breast cancer patients with the AA homozygous status of this mutation will experience a shorter overall survival after they are diagnosed with a breast cancer. And this will mean that this mutation of PCSK9 may be linked to patient survival, and the most impactful factor of breast cancer survival will be breast cancer metastasis. So we went on to study if this PCSK9 gene, the gene in the cardiovascular disease field, also affect cancer development. In this cancer model, we can create a PCSK9 knockout animal model, and then we implant the breast cancer cell into the bloodstream of these mice and observe the formation of lung metastasis. As you can see here, in PCSK9 knockout animal, we have way less breast cancer metastasis. That means this protein is a promoter of breast cancer metastasis. And when we also directly model the effect of this mutation observed in human population by introducing this mutation into animals, you can see that animals carrying this AA version of the PCSK9 gene also have higher metastasis burden. So this is the first causal evidence that we have identified a germline driver of breast cancer metastasis. And this is very exciting finding to us because PCSK9 is a famous gene in the cardiovascular disease field. there are many commercially available safe and effective therapy targeting this protein. And we tested one of the antibody therapy developed by Amgen called evolocumab. We didn't tell Amgen about it, no conflict of interest at all. But we found that when we test this antibody therapy in the animal models, we could achieve a suppression of metastasis formation in these animals. As you can see, the typical mouse lung histology will be like this, and the dark purple would be the metastasis nodule. So we tested the efficacy of this therapy in a variety of models and observed similar phenotypes. So pinpointing a new potential therapy for breast cancer patients. And what's even more exciting, because it's an inherited factor, we may use it as a biomarker to predict the risk of metastasis. In this Swedish early-stage breast cancer cohort, we can see that patients of the AA genotype will have a higher risk of developing metastasis. Years after a diagnosis is actually, after 15 years after diagnosis, 22% of the AA people, that's 70% of people, will have metastasis, while compared to the 2% risk in the G carrier group. So in summary, through this kind of combination of human genetics, experimental study, and translational work, we could identify an inherited factor that could promote breast cancer metastasis. That basically means that the disease outcome of a future malignancy is probably in part predetermined at birth, decades before the diagnosis of cancer. So believe this is a universal mechanism. This will help, if we apply this pipeline to other cancer types, we will uncover new biology, new drug targets, and new biomarkers that will truly benefit the patient population. And with that, I want to thank people who contributed to this work, and I want to thank you for your attention.
Fantastic. All right, who has a question? Matt?
Are there enough real-world data of people taking evolocumab who have metastases to get a read on whether this really is recapitulated in people?
Yes, a very good question. We are working with our Swedish collaborator to see if they, in their patient population, they have this kind of record. One challenge in this kind of research is that evolocumab or this kind of PCR inhibitory therapy is mainly used when statin is not effective or the patient cannot tolerate statin. So, and also this drug was approved 10 years ago, so it's not has been in the clinic for very long. We are trying to gather enough patients to answer this kind of question. Yeah.
And have you approached Amgen about doing any kind of, I mean, how big would a clinical trial have to be to show this? How long does it take for metastasis to happen in this population?
Yes, one problem of this kind of research is especially for early stage breast cancer patients, it takes very long time for patients to have a metastasis relapse. So it's quite difficult to have a group of patients taking this therapy on a monthly basis. So I think in the future, going forward, if we really want to make this access useful, we need to combine this with biomarkers to pick people really at risk of developing metastasis. For example, of the people of AA genotype, we have higher risk of developing metastasis. So this therapy may work better for them.
Are there any other questions? All right. Thank you so much. For our final Stat Madness presenter representing the new food allergy treatment team, please welcome Kim Mudd, Nurse Manager at Johns Hopkins Division of Pediatric Allergy, Immunology, and Rheumatology.
Thank you. So we're going to talk about food allergy today. So food allergy affects between 8 and 10% of the population in the US. The food allergy is an interaction between the immune system and a food protein. In response for certain susceptible individuals, their bodies make an antibody called IgE. And it's this IgE that really drives that allergic reaction. Symptoms of food allergic reactions can be mild or severe. Mild symptoms include, hives and swelling. We have GI symptoms. And then we start getting into some respiratory symptoms, looking at airway swelling, stridor, coughing, wheezing, cyanosis, decreased blood pressure, thready pulse, lightheadedness, fainting, cardiovascular collapse, and death. Now, we call all those symptoms in red, we call those anaphylaxis, right? So multi-system severe allergic reaction, anaphylaxis. Food allergy is the #1 cause of anaphylaxis in children. Food allergy management. If you eat this food, you're going to have a reaction. Don't eat that food. Turns out to be way harder said than done. And accidental ingestions are extremely common. And we're talking about a sip of milk. We're talking about a bite of a cookie. We're talking about a little nibble of nut. These are the amounts of foods that can result in significant symptoms and cause medical emergencies, right? So now, is every single allergic reaction going to be anaphylactic? No. Is this reaction going to be anaphylaxis? We don't know. We don't know. And that constant having to be constantly vigilant and not knowing what that next reaction is going to be like really impacts on the quality of life for people that have food allergies. If you ask a food allergic mom what she's worried about, she'll look you right in the eye and tell you, I'm afraid my baby's going to go to preschool, my child's going to go to elementary school, my son's going to leave for college, he's going to eat something he shouldn't eat, he's going to anaphylax, and he's going to die. There's limited treatment for food allergy. Before we discuss treatment, I need to know that most people, certainly the patients in our clinic, are multi-sensitized, right? They have multiple food allergies, allergic to peanut, milk, and egg, allergic to sesame, cashew, and wheat, right? Multi-sensitized. There's an FDA-approved oral immunotherapy agent. It treats peanut allergy, right? Which is great if you just have a peanut allergy. Most people don't, all right? And then we have omalizumab. So omalizumab prevents allergic reactions by blocking IgE. We talked about IgE being the thing that really drives that allergic reaction. Omalizumab blocks IgE. And because it blocks the reaction at that IgE level, it can be used for multiple foods. It doesn't matter what the food is triggering the allergic reaction. It works on all of them. Omalizumab was approved in 2024 to reduce allergic reactions that may occur to one or more foods. The FDA application was supported by the OUtMatch study. The OUtMatch study is a phase 3 multi-center clinical trial being done across 10 sites in the US. The entry criteria for OUtMatch, everybody had to be between 1 and 55 years old. They all had to be multi-allergic, right? Multi-food allergic. And not only did they have to be multi-food allergic, they had to be exquisitely sensitive to peanut, plus 2 additional foods from this list: milk, egg, wheat, cashew, hazelnut, or walnut. We screened 479 participants across these 10 sites using a double-blind, placebo-controlled food challenge, which is the gold standard when you're doing food allergy research. 180 patients met our inclusion criteria. They were randomized 2-to-1 to receive omalizumab or placebo. 120 people went on omalizumab, 60 people got placebo. We went through 16 weeks of treatment. At the end of that 16 weeks, we did another round of those double-blind, placebo-controlled food challenges. And these are the outcomes of those challenges. It's a busy slide, I apologize, but let's look at the far left. The far left area is looking at peanut, okay? The orange bar are the people that were treated with omalizumab. The blue bar, people that were on placebo. These people coming into the trial had to react to less than 1/3 of a peanut. That's how exquisitely sensitive these people were. After 16 weeks of omalizumab, those same people could tolerate 4.5 peanuts. If we look down at milk, again, somebody who reacted to less than a teaspoon of milk. 66% of them, after 16 weeks of omalizumab, could handle 1/4 cup of milk. If you look at egg, again, somebody that could handle less than 1/10 of an egg. After 16 weeks of omalizumab, they could handle 1/2 an egg. And you look across the tree nuts, it's similar except for cashew. So what does this mean for our patients, all right? What this means is that treatment with omalizumab provides significant protection across multiple food allergens, right? There was improvement in every single one of those categories. That protection far exceeds accidental exposures, right? We're not talking about a sip of milk. We're not talking about a bite of a cookie. We're not talking about a nibble of a nut, all right? Omalizumab is going to be life-changing for our patients. Thank you.
Thank you so much, Kim. And now to pass things over to our judges. Does anyone have a question? Lonnie?
So very striking data, very cool. One question, is the proposal then that you're going to take a 2-year-old and have them have an injection every 2 weeks for the rest of their life?
So, good question, great question. This is a treatment, not a cure, right? If you stopped omalizumab treatment, you're going to go back to being just as allergic as you were. Could you do it for long-term? Yes. We have long-term safety data on omalizumab. We've been using it for 25 years to treat allergic asthma, right? What we're probably going to see how this is going to be used is at transition times, right? So, you know, for some toddlers, a toddler that's going to eat anything you hand to them, that's a toddler that needs to be on omalizumab, all right? School-age kids may be a little more careful. College-age boys, absolutely allowed to go on omalizumab. They just should, because it's a transition time when they're not prepared to either recognize symptoms, treat symptoms, they're not prepared. So we see this as being something that's probably going to be used during those transition periods. But there's going to be people that are going to choose to stay on it for lifetime. There are.
So can I just ask a follow-up then?
Yeah.
How many injections before the protection kicks in? So how long do you have to prepare?
Hard to say. We did 16 weeks. That was way longer than we needed, except for cashew, because there was an open label extension here that I didn't have nearly enough time to talk about. And so 60 of these people went on to another 24 weeks of omalizumab, and we actually saw that cashew did improve over that 24 weeks. So we say at least 6 weeks before we get good coverage. But yeah, this was a 16-week plus another follow-up with 24 weeks.
Seema?
No, so it's FDA approved for?
It is FDA approved for food allergies.
It is FDA approved
as of February 2024.
So what are the next steps, and what's the uptake, and how are you?
So the uptake is that we have a clinic nurse that spends her entire day doing prior authorizations to get our patients on these drugs, right? And what this looks like 6 months from now, a year from now, 3 years from now, we don't know. I mean, this is not a cure. It certainly doesn't stop us from doing the next study and the study after that, but it's going to help. It's something we can do proactively for our kids now.
Is there a final question? All right, thank you very much.
Thank you.
You can actually stay on the stage.
Okay.
So now, thank you all to all of our presenters for sharing their research. I would like to invite all of the presenters back onto the stage. And then we are going to have the judges share any final questions or comments on the 4 presenting teams. Do you have any final questions or comments? If not, I'll point to Matt.
Yeah, you'll point to me. I have some comments. They're less questions, but I thought that the quality of all of these was really amazing. Real clinical implications for all of them, near term in either research or in actually doing something in the clinic. Dr. Kim Mudd, I was really impressed by the immediate utility here. I think that that's really important. And similarly, Dr. Armstrong-Hugh, the applicability there and the ability to actually potentially, although the trial sounds very hard, it's exactly why we need an NIH to do trials like this, and why before my comment was the NIH needs to do more trials. So I was really impressed with those two. I think in terms of blowing my mind, Dr. G, Wenbin May, I'm sorry. G, you blew my mind too, but the craziest idea up here is that a PCSK9 inhibitor could prevent cancer metastasis. Yeah. I do think we just need a lot more. I've seen so many attempts at preventing cancer metastasis that worked in mice, I feel like. So or appeared to work in mice. This looks really promising, but I think it needs more runway. And I want to know more on the on the big data. I would want more comfort that it's really different from all the other NLP approaches that I'm seeing out there.
And can I add one more thing?
You can add whatever you want, Seema.
No. I agree with you. And the biomarker piece is actually even more important, because if you can detect, if you have a biomarker that can detect who is predisposed to their cancer metastasizing, then I think, you have something there. Because otherwise, you have to wait for a long time to figure it out.
Well, especially if you could enrich the trials too.
Yeah.
I mean, you could cut the size in half, right? And
Exactly.
I just wanted to congratulate you all on great presentations. Having been there, I know how nerve-wracking it can be to get your story across in 5 minutes. Well done.
Go ahead. Yeah. Well, I'm glad we're not scoring, right? The audience is scoring.
The audience is scoring.
It's going to be very difficult.
Equally glad I'm not scoring on the deep end of the science pool this afternoon, but thank you in getting through really dense and complex challenges, and the applicability really shone through in each of your presentations. So congratulations and thank you.
Congratulations, everyone. And now it is the hard part. It is time to vote. So everybody, please take out your cell phones and open the Bizzabo app that you should have downloaded. If not, you can download it super quickly. There is a QR code on the back of your name tag to download it from the App Store and then make a profile. You sign in with the same email you used to register for the event. In the app, on the event homepage, you're going to click on the 3 dots at the bottom right-hand corner of the screen. It should say More, and then from there you are going to select Polls, and then you should see a poll for this session on the slide. It might populate as an example on the slide. And then you're going to vote for who you think should win Stat Madness 2025. I'm going to pull up my own poll here and cast a vote so that I can see for myself how we're doing. This is a very tough decision, you guys. We've got our Jeopardy music going. I don't know if anyone follows the actual March Madness competition, but I grew up in a basketball town, so while I don't follow basketball, I am following the intensity back home in Durham, North Carolina between UNC and Duke is always very, very intense, as you can imagine. All right, I'm looking at some results. I see they're still coming in. If everyone can cast your vote. It is a tight race. We'll wait and get a couple more votes in. Sorry guys, I know I'm like drawing out the suspense. Wow, these numbers are moving. You guys are all still casting votes. All right, I think I do see that we have a winner. I am very pleased to present this trophy to Mary Armstrong Huff. And then, we can get a picture with everybody. Let's get pictures.
Justin, NYU grad.
Very nice. Congrats. Very good.
Congratulations. Thank you all so much for participating.
For closing remarks from our Stat Madness sponsor, please welcome to the stage Josh Hoffman, Director of Healthcare at Jobs Ohio.
Awesome. Well, I know better than to stay between a group of people and drinks at 6 o'clock, so I'll try to keep this short. Wow, just what a great event today at STAT 2025 Breakthrough Summit East, as well as the conclusion of Stat Madness 2025. It was great to hear from industry leaders like Scott Gottlieb and James Wilson as we face really unprecedented challenges and opportunities in regards to access to care, as well as R&D and commercialization in life sciences and healthcare. We were very privileged at Jobs Ohio, as well as the Ohio Discovery Corridor, to be a part of the event today, and I'd love to thank STAT and their incredible team for hosting us. My name's Josh Hoffman. As you can see, I am Director of Healthcare at Jobs Ohio. My colleague Tyler Alchin mentioned we head up the life science investment as well as economic development for the state of Ohio. In 2020, Jobs Ohio and the state of Ohio invested a little bit over $400 million in 3 innovation districts across our 3 largest cities to catalyze life sciences research and commercialization, as well as talent development. This has resulted in us going across the state to connect our top clinical care providers, our research institutions, our scaling biotechs, as well as our industry leaders across this Ohio Discovery Corridor. We believe this provides a one-stop shop for all your biotech and pharma needs, and we truly do think it's the best kept secret in healthcare and life sciences, and we encourage you to join us on our journey as we grow. Excuse me, sorry, should have got some water. If you are interested in joining us in the Ohio Discovery Corridor, please find somebody from Team Ohio. We'll be upstairs at the event. We were happy to talk about how we can help support your growth. We would also love for you to follow us on LinkedIn at Ohio Discovery Corridor. Once again, I would like to thank the team at Stat News for this incredible event and opportunity, and I hope everybody has a great evening. Thank you.
And you can go, but I have a couple things to say first, but that's basically it. So you saw on screens outside the room here, up on the rooftop for lunch, we're promoting a few exciting things going on at STAT. Scan the QR codes to learn more, and in no particular order. Sign up for our newest subscriber-only newsletter, AI Prognosis. Each Wednesday, STAT's health tech reporter, Brittany Trang, brings the Hitchhiker's Guide to the Healthcare AI Galaxy to your inbox. The answer is 42. We hope that you're all Stat Plus subscribers, but maybe a few of your colleagues aren't. We have enterprise subscriptions. Andre at the Stat booth right at the back can share more details. Purple glasses. Email alerts. They are available exclusively to Stat Plus subscribers. You can be alerted to get an email when your favorite Stat reporters post a story, or subscribe to specific keywords like biotech, AI, CRISPR, and more. Now just subscribe to me. This puts the news in your inbox faster than waiting for an end-of-day recap email. And lastly, events. We're doing a lot of them. We'll be in San Diego for the American Academy of Neurology hosting an evening event on April 6th. Free and open to anyone to register. Our West Coast Summit, focused on AI, the Stat Breakthrough Summit West, is in San Francisco on May 14th. Featured speakers include Reed Jobs of Yosemite, Aviv Regev of Genentech, and Karen Knudsen, former president of the American Cancer Society. A bunch of announcements coming there. We'll head out to Chicago for ASCO and host an evening event on May 30th, open to everyone that will be in town for the weekend. That event is amazing. And finally, a big heads up that our flagship Stat Summit will be taking place on October 15th and 16th in Boston. The theme is the next 10 years. More information on that will be available on our website soon, but you won't want to miss it. Already, we've confirmed Elise Dan Skowronski, Michelle Ziavakeso, Biopharma, and more. Thank you very much for being here. I hope to see you again soon, and also at the cocktail hour. Thanks.